Hello, am I audible, visible? Good afternoon all of you, am I audible, visible? Hope everything is clear. Yes. So, welcome to Unacademy, a one-stop destination for all English medium civil service aspirants. Welcome to the final revision series. In today's class, I'll be taking a marathon on entire science and technology, focusing on most important areas of the subject. You can download the annotated PDF of the session in my Telegram channel, the link for which is provided here. Today's session will be held in 4 parts with 3 breaks in between. Please note the schedule. And before we start the session, a very important announcement. If you are preparing for 2026 and beyond, you have a great opportunity to get a beyond offline experience through our dream project Unacademy Platinum, where you can interact with the teachers live with your cameras on, and we can provide continuous guidance, mentorship and answer writing evaluation. The first batch of Platinum started recently. Till May 16th, evening at 7 p.m, you can attend the classes for free. Click on the link provided in the video description below. Let us start the session. And as you all know, in science and technology, we have several subjects like biotechnology, nuclear, nano, defense, space, ICT, etc. We are starting the session with biotechnology, the most important area from where considerably more number of questions are coming. In the preliminary examination, you can expect around 15 questions from science and technology, where around 4 to 5 questions are coming from biotechnology itself. And most of the biotechnology revolves around one hero molecule, that is DNA. So before we actually proceed with the techniques, okay, let us try to understand some basics of what is a cell and what is DNA. I hope everything is clear. Yes. Right, I am at my maximum volume. You can try to increase volume in your devices, please. Right. Next. So to start with, our body is constructed with cells. Cells are the building blocks of the life. And our body is constructed with the cells, just like the wall is constructed with the bricks. Every cell will have a cell membrane and a fluid inside called as cytoplasm. Cytoplasm. And at the center, we have a structure called nucleus. And in today's class, what all we are going to discuss are located in the nucleus, right? And in the nucleus, we have some chemicals. Inside the nucleus, we have some chemicals. Okay, these chemicals are called as nitrogenous bases. Nitrogenous bases. Why they are called bases? Because they are basic in nature. You know, acids and bases, they are basic in nature. Why are they called nitrogenous bases? Because they contain nitrogen element in their structure. Okay, these are the nitrogenous bases. Where do we have the nitrogenous bases? Inside the nucleus. What are they? Okay, they are adenine, thymine, guanine, cytosine and uracil. I repeat, adenine, thymine, guanine, cytosine and uracil. What are they? They are all nitrogenous bases, nitrogen containing bases. Now listen carefully, complete focus please. So, any one nitrogenous base, like for example adenine, any one nitrogenous base, to that if I add a sugar, this complex is called as nucleoside. Why the name? Because it is present inside the nucleus, nucleoside. Then to this nucleoside, if I add one phosphate, okay, nucleoside plus phosphate, then it is called as nucleotide. Again, why all these words are starting with the prefix nucleo? Because they are all located inside the nucleus, nucleotide. So what is a nucleotide? It is nucleoside plus phosphate, or a base plus sugar plus phosphate, a complex of 3 chemicals. A nitrogenous base, a sugar plus one phosphate is called as nucleotide. Okay, like for example, look at here. Okay, what is this? This is adenine, adenine. And what is this R here called as? That is a sugar called ribose sugar. Plus what is this P here? Phosphate, phosphate. So you can see 3 chemicals, adenine, okay, that is one nitrogenous base, okay, a sugar and a phosphate. The 3 together, I mean this complete block is called as one nucleotide. Then this is another nucleotide, one more nucleotide, one more nucleotide. Then all the nucleotides together will form a chain. Such a chain of nucleotides is called as nucleic acid. A chain of nucleotides is called as nucleic acid. Okay. And we have 2 types of nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Let us see the differences between the 2. The first difference is in the bases. DNA contain adenine, thymine, guanine, cytosine. Can you tell me which nucleotide is missing in the DNA? There is one nucleotide which is not there in the DNA. What is that? Yes, uracil. Okay. Whereas RNA, ribonucleic acid, contains AUGC, adenine, uracil, guanine, cytosine, thymine is missing. The second difference is in the sugar. Okay. DNA contains a sugar called as deoxyribose. Name of the sugar, nothing to understand in that, name of the sugar, deoxyribose. Whereas RNA contains a sugar called ribose. Actually, the nucleic acids are named after the sugar they contain. Okay. Ribose containing nucleic acid, ribonucleic acid. Deoxyribose containing nucleic acid, deoxyribonucleic acid. Okay, in this diagram also you can see the R stands for ribose, whereas here this is DNA. In this I have drawn DR. What is this DR? That is deoxyribose. Deoxyribose. That is the second difference. The third difference is DNA is a double stranded structure, whereas RNA is a single stranded structure. Double stranded means what? It will have 2 chains of nucleotides, whereas RNA will have a single chain of nucleotides. Okay. So these are the differences between DNA and RNA. The difference between DNA and RNA. Now let us see the same difference in a little more detail. Look at here. This first one is RNA. In this RNA you can notice, okay, adenine, uracil, guanine, cytosine. You cannot see the thymines. Second observation, which sugar you have here? Ribose sugar you have. Third observation, you have how many chains of nucleotides? You have a single chain of nucleotide. So this is RNA. Okay. Now coming to this one. This is DNA, deoxyribonucleic acid. Okay. You can notice adenine, thymine, guanine, cytosine. You cannot see the uracils here. First point to be noted, Your Honor. Okay. Second difference. Okay. If you notice, this is not the ribose sugar. You have deoxyribose. DR stands for deoxyribose. Okay. Third difference is you can see 2 chains of nucleotides. This is one chain of nucleotide. This is a second chain of nucleotides. Both of them are connected with some bonds called as hydrogen bonds. Okay. And we call this mechanism as base pairing. Base pairing means what? Pairing of the bases. Adenine will always pair with thymine and guanine will always pair with cytosine because they are like lock and key. If adenine is like lock, thymine is like its key. They both are complementary to each other. Adenine cannot bind with guanine or cytosine or another adenine. Okay, AT will base pair, GC will base pair. Between AT you have 2 hydrogen bonds and between GC you have 3 hydrogen bonds. AT also means a TA. I mean, in one strand if you have A, the other strand will have T, and in one strand if you have T, the other strand will have A. Okay, in one strand if you have G, then C. If there is C, then G will bind. Okay, this is about the DNA and RNA. Okay. After understanding the DNA and RNA, now let us try to understand the chromosomes. What are the chromosomes? Okay. See, we have seen about the DNA, double-stranded DNA like this. But this double-stranded DNA is very long, okay, like even kilometers together. It's a very long thread-like structure. But whereas our nucleus is very small, so how can you fit such a long DNA into such a small nucleus? So this thread-like structure will get tightly folded. Just like, for example, you are flying a kite, the thread, the manza is very long. Then how do you maintain it? By folding it onto some wooden piece. Okay, so you are coiling it, tightly folding it. Okay, such tightly condensed DNA or tightly packed DNA is called as chromatin. Use the word once, all of you. The tightly condensed DNA or tightly packed DNA. Where is it getting packed? It is getting packed inside the nucleus. That is called chromatin. Okay. Then if the chromatin is more tightly packed, maximum condensed, if the chromatin is more tightly packed, then it is called as one chromatid. There is only one letter difference. Chromatin, condensed DNA, tightly packed DNA. But this tightness is not enough. So more tightly condensed DNA is called as chromatid. And two chromatids together is called as one chromosome. Two chromatids together is called one chromosome. Okay, like look at here. This is one chromatid. Chromatid is 8 shaped like this. I mean, don't count this as 1, 2, 3, 4. This is wrong. Okay, you have only 2 chromatids here. 1, 2. So chromatid is 8 shaped like this. Then this is 2 chromatids. 2 chromatids together is called as one chromosome. 2 chromatids. Okay. And now look at here. And one small disclaimer. In today's class, every point here is needed to understand the next part of the class. So since beginning, okay, keep understanding and also keep remembering the points very carefully. Okay. Now coming to the number of chromosomes. In each cell of our body, okay, in every cell of our body we have 23 pairs of chromosomes. What do you mean by a pair of chromosome? Look at here. Is this one chromosome or a pair of chromosomes? Yes, is that one chromosome or a pair of chromosomes? This is one chromosome. Okay. So a pair of chromosome looks like this. This is another chromosome. This is one pair of chromosomes. Right. If this is one pair of chromosome, we have total 23 pairs of chromosomes in each cell of our body. In this 23 pairs of chromosomes, the first 22 pairs are called as autosomes. What are autosomes? They are chromosomes only. The first 22 pairs of chromosomes are called as autosomes. Okay. What is their duty? They control all the characters in our body which have nothing to do with the sex of the person, like digestive system, nervous system, circulatory system, skin, nose, ear, everything other than the reproductive aspects is taken care by these autosomes. These autosomes are same for a man and a woman. A man also will have 22 pairs of autosomes and a woman also will have 22 pairs of autosomes. The difference between male and female lies only in the last pair. Okay, try to understand using this diagram. Look at here. Okay, this is first pair of the chromosomes. I hope you are able to see this first pair of the chromosomes, second pair, and as I cannot draw 23 pairs, just dot 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 dot, imagine this is a 21st pair and this is 22nd pair, right? Okay, and here I have drawn, this is female and this is male chromosomes. Chromosomes in a female cell, chromosomes in a male cell. Okay, if you compare a male and a female, you can notice that they both have same number of autosomes. There is no difference in autosomes. The actual difference lies only in the 23rd pair. Okay, the 23rd pair is called as allosomes or sex chromosomes. Only 23rd pair is called as allosomes or sex chromosomes. Why are they called sex chromosomes? Because they control sex characters, I mean reproductive system, okay, etc. Okay, so the 23rd pair in the female is XX, only 23rd pair, and only 23rd pair in the male is XY. Okay, so if the 23rd pair is XX, it is female, and if the 23rd pair is XY, then it is male. So male and female differ only with respect to the sex chromosomes. Both have 22 pairs of autosomes. Okay, and autosomes control the characters that are common for both the sexes. Okay, there is no difference between both the sexes. 23rd pair are called as allosomes. They are also called as sex chromosomes. Okay. Why they are called sex chromosomes? Because they control sex characters. Because we have two sexes, male and female, we have two types of sex chromosomes, X and Y. X is female sex chromosome. What is X chromosome? Female sex chromosome. Why this is called female sex chromosome? Because it controls female sex characters. Okay, I mean ovaries, egg production, uterus, female reproductive system. Okay, whereas Y chromosome is called male sex chromosome. Why this is called male sex chromosome? Because it controls male reproductive system or male sex characters, like for example testis, sperm production, etc. And you can also notice that the X chromosome is larger and the Y chromosome is smaller. Look at here. Okay, I have drawn the X chromosome larger and Y chromosome little smaller. Okay, X chromosome is larger, therefore it can control more characteristics. So that is why what happens is, okay, X chromosome control female sex characters plus, okay, important point please, X chromosome control female sex characters plus it also controls some common characters, like which have nothing to do with the sex of the person, like color vision, blood clotting, brain development, etc. So Y is only for male characters, whereas X is for female characters plus, okay, plus some common characters. What are those common characters? Like blood clotting, color vision, brain development, etc. So based on this point, you all tell me, can a human being survive without X chromosome? Because it controls female's characters. Not only that, brain development, blood clotting, such vital common characteristics also. So can a human being survive without X chromosome? No. That is the reason why even a male will have X chromosome. You may get a doubt that if X is a female sex chromosome, what is it doing in the male body? Because it is also controlling some common characters. And can a human being survive without Y chromosome? Of course, yes, because it is only for the male characters. Females do not need the male characters. So females do not have a Y chromosome and we can happily survive without Y chromosome. So X is compulsory. At least one X chromosome is compulsory for the survival. 23rd pair in females is XX. 23rd pair in the males is XY. And you can see one word here, that is somatic cell. Somatic cell. Use the word once, all of you. Okay, somatic cell means body cell. What is body cell? Any cell in our body. Skin cell, hair cell, tooth cell, bone cell, liver cell, heart cell. Any cell in our body except the reproductive cells like egg, sperm, zygote, etc. Okay. So such cells are called as somatic cells, right? Okay. Right. Then we have seen the difference between male and female. Now let us see some chromosomal aberrations. So we are now discussing a topic called as chromosomal aberrations. Use the word once. Chromosomal aberrations. Chromosomal aberrations means errors in the chromosomes. Errors in the chromosomes. Okay. And let us see here. Answer these questions. XX means normal female or is there any aberration? First, XX is a normal female or is there any aberration? This is a normal female. But if you have an extra X chromosome, this is called as super female. Simple logic. There is one extra X chromosome, so it is called super female. But super female is not super, rather actually this is a disorder. It is not a plus point, not even a normal condition. It is a disorder. Super female syndrome. Next, XY is a normal male. Okay, XY is a normal male. Okay, but if there is one extra Y chromosome, a normal male will have only one X, one Y. But if one male by mistake have two Y chromosomes, okay, what is it called as? Not Superman, super male syndrome. Super male syndrome. Again, this is not a plus point, it is a disorder. Remember, we are discussing about aberrations. Is it clear? Next, X0, I mean, okay, a normal female should have 46 chromosomes with XX and male should have 46 chromosomes with XY. But this individual will have only 45 chromosomes. Look at here, okay, 22 pair, first pair, second pair, 22nd pair, 23rd pair, one is missing. Okay, that condition is called as, okay, X0, X0, one chromosome here is missing. Okay, that condition is called as Turner's syndrome. Okay, why the name Turner's syndrome? Because the scientist who identified that particular syndrome is Turner. Okay, Turner syndrome, it is monosomy of X chromosome. Monosomy, quite simple. Mono means what? Mono means one. Somy is a suffix of chromosome. You can see only one chromosome here. Therefore, it is called as monosomy. I hope the word monosomy is clear. Okay. Next you have XXY, XXY. This condition is called as Klinefelter's syndrome. This condition is called Klinefelter's syndrome. Why the name Klinefelter's syndrome? Because a scientist called Klinefelter identified this syndrome for the first time. Okay, you can see the Klinefelter's syndrome condition here. Look at here. This is Klinefelter's. First pair, second pair, 21st pair, 22nd pair, normal. But 23rd pair, if you observe, you can notice XXY. Two large chromosomes and one small Y chromosome. Klinefelter's syndrome. This is trisomy. Okay. In this word trisomy, tri stands for 3 and the word somy stands for, this is a suffix of the word chromosome. So 3 chromosomes in this slot, trisomy. Overall gender, okay, I will ask you to predict after some time. Okay, so that is Klinefelter's syndrome. If you have XXXY, okay, XXXY, that is called as extreme Klinefelter's syndrome. Extreme Klinefelter's syndrome. I hope it is clear. Same like Klinefelter, but okay, the symptoms will be more severe. Okay, then answer a few questions here. Everybody please. Okay, tell me the total number of chromosomes here. XX, normal female. What is the total number of chromosomes? 46 or 45 or 47? XX. No, not 47, 46. Very good. Okay. Super female. Okay, total number of chromosomes. Everybody, super female total number of chromosomes. 47. Okay, because you will have 22 pairs, 44 till here plus 3 chromosomes extra. There is one extra chromosome. So the overall number is 47. XY is 46. Super male syndrome. Look at here, this is super male condition. Okay, look at super male syndrome. 22 pairs of autosomes normal, but okay, XYY. Okay, notice the difference between okay super male and Klinefelter. Super male is okay XYY. Klinefelter is XXY. I hope the difference is clear for all of you, but both are trisomies, right? Yes, correct. Okay, so here also total number of chromosomes 47. Turner's syndrome, here one chromosome is missing, I said, X0. Okay, what is the total number of chromosomes in Turner's syndrome? Total number of chromosomes, very good, 45. Okay, Klinefelter's 47, that is also trisomy. Extreme Klinefelter's total number of chromosomes will be 48. Okay, 3 X chromosomes and 1 Y chromosome. Fine, very good. Thank you so much. Then give me a few more answers. Okay, guess the sex in each case, male or female or intersex. These are the 3 choices I am giving to you. Okay, is it male or female or intersex? Everybody? This is of course female, normal female. Super female, disorder but male or female, abnormality but male abnormal or female abnormal? Female, because there is no male sex chromosome. All the 3 are female sex chromosomes only. So female. Okay, XY, normal male. So of course male. Okay, XYY. If a male contain an extra male chromosome, the gender obviously will be, if a male contain an extra male chromosome, the gender will be male only. Okay, X0, male or female? Male or female? X0. Okay, X0 female. Then you may get one doubt. Why female? Because, okay, see which chromosome gives male characteristics? Fast everybody. Which chromosome gives male characteristics? Which chromosome gives male characteristics? Y. So without Y, how can she get male characteristics? So no male characteristics. So therefore the sex will be not a normal female. Okay, but overall the sex will be female only. Okay, obviously simple, like clouds gives rain. When there is no clouds, there is no rain. Y chromosome gives male characteristics. When there is no male Y chromosome, there are no male characteristics. I hope it is clear. Now comes Klinefelter. Okay, XXY. This is intersex. This is intersex. Okay, now all of you answer this question. Okay, a normal male will have one X and one Y. I mean female chromosome is also there, male chromosome is also there. But in a normal male, which one is expressing? Everybody? In a normal male, female chromosome is also there, male chromosome is also there, but which one is expressing Y? It means Y will suppress male domination here also. Y will suppress the X chromosome. Okay, so Y will suppress even if there are two X chromosomes. I mean, even if you have XXY, the Y will dominate both X chromosomes. But when there are two females, okay, unity matters. So the two female chromosomes can also show a little bit of expression. They are not completely suppressed by the male chromosome. When there is a single X chromosome, it is completely suppressed by the Y chromosome. Okay, therefore, it is purely like male. But when there is a Klinefelter, Y chromosome can suppress both X chromosomes also, but not completely. So overall sex will be male. Okay, in the case of Klinefelter, okay, overall sex is male, but will exhibit certain feminine characteristics. Look at here, Klinefelter syndrome, trisomy, overall gender male, but will express certain feminine characteristics. I hope it is clear. Yes. So we can use the word intersex here, male but female, breast development, voice changes, like that. So we can use the word intersex here. Intersex means between the two. Super male syndrome symptoms, trisomy, overall gender male. Symptoms are they are taller than average, and interestingly they have criminal bent of mind. But of course it does not mean that all criminals are super males or all super males are criminals. Okay, just that they have a little aggressive nature. That is also not a proven fact, it is observed in some studies. Okay, that is about the super male syndrome. I hope everything about the super male syndrome is clear. Okay, super female, trisomy of X chromosome, gender female, taller than average, they have some learning difficulties, lesser cognitive skills. Okay, Turner's syndrome, monosomy of X chromosome. Okay, gender is female, symptoms infertile, short stature, stature means height. Okay, short height, and they also have learning difficulties. Okay, I hope all these are clear for all of you. Super female, normal male, super male, Turner's, Klinefelter's, extreme Klinefelter's. Okay, right. Next coming Down's syndrome. Why this is called Down's syndrome? Because discovered by the scientist called Langdon Down. Langdon Down. Right? Okay. This is trisomy of 21st chromosome. You can see the 21st chromosome is present in 3 copies. Okay. Till now what all we have seen, this complete list is sex chromosomal aberrations, whereas this one is an autosomal aberration. Okay, and it is the most commonly observed aberration. Because it is an autosomal aberration, it can occur in a male and also can occur in a female. You can also have a male Down's patient or you can also have a female Down's patient. I hope it is clear. It has nothing to do with the sex of the person. Here also delayed development, growth retardation also we can call, learning disabilities, autism. Autism means behavioral problems and learning difficulties. Okay, so this is about the Down's syndrome. Okay, so after we understood what is DNA, chromosomes, chromosomal aberrations, the next word we are trying to understand is ploid. Okay, use the word once and memorize the word. All of you memorize the word. Right, what do you mean by the word ploid? Okay, ploid means, okay, look at here, one set of all chromosomes in a cell, one set. Now look at here, through the diagram let us try to understand. This is 23 pairs of the chromosomes, first pair, second pair, dot 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 dot, 23rd pair, right? 23 pairs of chromosomes, right? Then in this, take one chromosome from each pair. take one chromosome from each pair. Okay, like this if you take. This is one set, not both please. This is only one set and not pair. Set. Pair means only two chromosomes. One set of all chromosomes in a cell. Okay, it means you are taking one from each pair. Okay, how many chromosomes will be there in one ploid? Everybody please. How many chromosomes will be there in one ploid? And feel as if this is an offline class and feel as if you are sitting right in front of me and feel like LKG students and you can shout out your answers. Okay, and do it even if you are sitting in some library, no problem, everybody will realize that you are properly revising it. Okay, now, so one ploid is equal to 23 chromosomes and we represent it with the letter N. N is equal to 23. I hope it is clear. Right, okay, N is equal to 20, that is one ploid. Then, so one ploid is equal to 23 chromosomes, okay, right. Then, how many ploids you can have in one cell? Yes, all of you, how many ploids, I mean, how many sets of chromosomes you have in each cell? 2 sets, okay, 2 ploids. Okay, look at here, this complete package of 20, like ploid is like a package. This complete package of 23 chromosomes is coming from the father. This complete package of 23 chromosomes is coming from the mother. This package is one ploid, this package is another ploid. We have one ploid coming from the mother, one ploid coming from the father, so therefore we are diploids, 2 ploids. Okay, we all are diploids. All animals and a good number of plants are diploid. Okay, they have chromosomes in pairs. Okay, but the number of chromosomes can change. Like for example, you take pea plant. It will have 7 chromosomes, but it is diploid. So what does it mean? 7 pairs of chromosomes will be there. I hope it is clear. You have Drosophila. What is Drosophila? Fruit fly. It will have 4 chromosomes, but it is diploid. Okay, means what? It will have 4 pairs. Is it clear? Same way you have maize, that is corn. Hope you enjoy sweet corn occasionally. That corn, okay, is okay, having 10 chromosomes and it is also diploid. Okay, means it will have 10 pairs. Whereas human beings are having 23 chromosomes, humans. Okay, but we are also diploid. That means 23 pairs. Is it clear? So the chromosome number can differ, but we all are diploids. It means whatever number of chromosomes we have them in pairs. From each pair, one comes from the mother, one comes from the father. For example, okay, from the first pair, one chromosome comes from the mother, one chromosome comes from the father. Actually, this is the reason why, okay, we should not say that we have 46 chromosomes. We have to say that we have 23 pairs of chromosomes. Okay, why we should not say that we have 46 chromosomes? Because look at here, this is not 24th chromosome, it is another copy of the first chromosome. Is it clear? It is not we don't have 46 different chromosomes, we have only 23 different chromosomes, but we have them in pairs. So that is the reason why we are diploid. I hope it is clear. Okay, right. Okay, then now, is ploid having sets like in one ploid having? No, it's not. Okay, don't confuse between pair and ploid. Look at here. Okay, this is, okay, pair. Okay, I'll draw like this. Okay, look at this. This is pair. Okay. Whereas this is ploid. Ploid will have 23 chromosomes, whereas a pair will have 2 chromosomes. Okay, we have 23 pairs and we have only 2 ploids. I hope it is clear for all of you. 2 ploids. Okay, this red color is one ploid. Okay, and this purple color is another ploid. Ploid 1. Okay, whereas that red color one is ploid 2. Clear? What is the difference between ploid and genome? Similar, Tejaswini. Okay, similar words. Okay, you're able to hear me? Sound, is the audio clear? When I am checking in my devices, the audio is clear and it is DJI mic, the best company mic ever, and I am in the maximum volume. And majority people are saying that the voice is clear, so please focus on the class, please. Okay, I am on full volume with the best company mic. Okay, and okay, please focus on the class. Right. Okay, so that is ploid, diploid, right? Okay. Most eukaryotes are diploid, means that they have 2 sets of chromosomes, 1 set inherited from each parent. However, some organisms are polyploids. Okay, look at here. Okay, why do we make polyploids? Okay, how do we make polyploids? Look, why do we make polyploids also? Look at this. This is a strawberry. Okay. This is diploid strawberry. You can see that it is small sized. But look at here, this is also strawberry. But this is octaploid strawberry. Octaploid means what? You will have 8 sets of chromosomes. It means, okay, whatever number of chromosomes you have, 1 set, 2 set. I hope you understood. Each chromosome will be there in 8 copies. Each chromosome is there in 8. Look at here, first chromosome 1, 2, 3, 4, 5, 6, 7. That is the meaning of octaploid strawberries. Is it clear? Yes. What happens if the ploidy increases? I mean, what happens if you get more copies of the chromosomes? In that situation, what happens is, polyploids cannot have new characteristics, but whatever characteristics are there, they are more enhanced. Like for example, if the strawberry is already in red color, the polyploid strawberry will be bright red color. If it is already sweet, the polyploid strawberry will be sweeter. If the diploid strawberry is small, the polyploid strawberry will be bigger. Whatever quality is there, it will quantitatively increase. So that is the reason why we make polyploids in the plants. I hope it is clear. We make polyploids in the plants. Is it clear? Not 8 parents chromosomes, you need not say like that, Varsha, but you can say 8 sets of chromosomes. Okay, because in the case of plants, we also have vegetative reproduction. Plants always do not mate with each other, okay, and give birth to the young one as in the case of animals. We can artificially add more ploids. You need not consider it as like 8 parents are involved. It is not like that. Plants also have vegetative reproduction. From a single plant, by grafting, you can grow another entire plant. Okay. These octaploids can also be made artificially. Okay. That is about the polyploid. Okay. Done. So now, next we are proceeding about, okay, the childbirth process. Okay. The childbirth process. Complete focus, please. Okay. Complete focus on the class. Okay. Look at here, this is female somatic cell. This is female somatic cell, right? Somatic cell means what? Somatic cell means body cell, right? Somatic cell means body cell. Any cell like liver cell, kidney cell, I already explained. Okay, and one small request, in my knowledge there are no technical issues, we have taken care of everything, let it be mic or streaming or whatever it is. And even if something is there, I am not a superhuman being to solve it instantaneously. So ignore those small inconveniences and completely focus on the topic. We have a limited time where we have to complete as many topics as possible. So please kindly understand that point, right? Okay. So this is a female somatic cell and this is a male somatic cell. Female somatic cell will have 22 pairs of autosomes plus 2 X chromosomes. All of you answer. How many chromosomes you can see in this cell? Everybody, how many chromosomes you can see in this cell? 46 chromosomes, 22 pairs of autosomes plus 2 X chromosomes, 22 pairs of autosomes plus 2 X chromosomes, right? Okay. How many chromosomes you can see in this cell? This is male somatic cell. Here also, okay, this is also 2N, means diploid. Total number of chromosomes, 46 chromosomes, okay, right. But it is 22 pairs of autosomes plus XY. That is the difference. Male somatic cell, female somatic cell. Okay. Now look at here. What are these? These are egg cells. Egg cells. Okay. Eggs are the female reproductive cells. Means in the female body, the cells that can grow into the baby. In the female body, the cells that can make a baby are called as female reproductive cells. Is it clear? Okay. Answer this question, everybody. One egg cell, this one egg cell will have how many chromosomes? You can see that one egg cell will have only, okay, 23 chromosomes. One egg cell will have only 23 chromosomes, not 23 plus 23. Only 23 chromosomes you have, okay. Whereas the sperm cell also contain, okay, this is also N, that is 23 chromosomes, not 46, okay. Means eggs and sperms have only half the number of chromosomes when compared to a somatic cell, only half the number of chromosomes when compared to a somatic cell. Is it clear? Okay. Then now tell me how many types of, not about the number, I am not asking you the number. How many types of egg cells you can see here? Look at this. How many types of egg cells you can see here? Only one type of egg cell. Both are X type. You can see only one type of egg cell. Both egg cells are X type only. How many types of sperm cells you can see here? How many types of sperms you can see here? Two types. One is X type, the other one is Y type. Ignore the number. You can have any number of eggs and any number of sperms. That is a different story. Okay. But you have only one type of egg cell and two types of sperms. I hope it is clear. Then, if the egg fuse with X type sperm, look at here. If the egg fuse with X type sperm, 22, XX, that is a female zygote will be formed. What is a zygote? If this is egg cell, this is sperm cell, these two fuse to form a single cell called as zygote. Okay, that fusion process is called as, okay, fertilization. Fertilization, right? Then, okay, if this particular egg fuse with, okay, Y type sperm like this, if the egg fuse with Y type sperm like this, then 22, XY, okay, that is a male zygote will be formed. 22, XY, male zygote will be formed. So who will decide whether a male baby will be born or a female baby will be born? It depends on the mother or it depends on the father. Only father can decide whether a male or a female baby will be born. But socially, even today, females are blamed for giving birth to the female children. That is a different story. Now coming back to this. Okay. After zygote, what is the next stage? Okay. Zygote will undergo cell division. Cell division means what? One cell will break into two. Then two will become 4, 8, 16, 32. So, like that, the cell keeps on dividing and the cell number keeps on increasing. And it will form a group of cells. Such a group of cells is called as embryo. Use the word and also memorize it. Okay, that is called embryo. Okay, and what is embryo? Embryo is undifferentiated mass of cells. Okay, don't imagine complexity, understand the word. Undifferentiated, it means what? Within the embryo, the cells are not different. All cells in the embryo are similar to each other. All cells in the embryo are similar to each other. Okay, unlike our body, for example, if you take our body, we have hair cell, skin cell, liver cell, brain cell. All of them are structurally and functionally different. But in the embryo, it is not like that. All the cells in the embryo are similar. Undifferentiated group of cells, that is called embryo. And then embryo will undergo a process called as differentiation. Differentiation. Use the word once. Differentiation. Okay, means what? The cells will start becoming different from each other. The cells will start becoming different from each other. Okay, finally forms liver, kidney, pancreas. With all organs formed, the embryo finally takes a human shape, like a small baby, that is called fetus. Look at the word fetus. Okay, fetus is the baby which is still inside the womb. Human shape looks like a baby, whereas embryo looks like a bunch of grapes. Fetus looks like a baby. All organs are there, but still inside the womb, not yet born. That is called as fetus. And then neonate. The word neo means new and natal means birth. So neonate means newborn baby. Okay. So these are the stages of development. I hope it is clear. Now let us see about the twins. Okay. About the twins. Complete focus please. We have two types of the twins. First type are called as dizygotic twins. Why are they called dizygotic twins? Here, instead of one egg cell, two egg cells will be released. The female releases two egg cells. Is it clear? Okay, two eggs are released. And they will fuse with two sperms. Okay, they will fuse with two sperms. And they will form two zygotes. I hope it is clear. Quite simple. A female releases two eggs. They will fuse with two sperms. Okay. And the two sperms and after fusion they will form two zygotes. The two zygotes will grow into two babies. Look at this flow chart, you will understand. Two eggs will fuse with two sperms to form two zygotes. They will grow into two babies. They are called dizygotic twins because they are born from two zygotes. Di means two zygotes. And because they are formed from two different zygotes, they do not look identical. They may look similar. Overall, on the whole, they can be similar. But they are not 100% identical. So, therefore, they are also called as non-identical twins. Is it clear? Right? Okay, non-identical twins. Okay. Then why are they called fraternal twins? You know, fraternity means what? Brotherhood. Fraternal. Fraternity means brotherhood. Okay. So, why they are called fraternal twins? They are not identical, okay. But they are not opposite also, like black and white, lambu, jambu. They are not like that. They can be similar. Okay. How much similar they can be? They can be as similar as normal brother-sister. Therefore, okay, they are called brotherly, that is fraternal twins. Okay. They are just as similar as normal siblings. Not more similar than normal siblings, not less similar than normal siblings. Therefore, they are called fraternal twins. Fine. Answer these questions. Use the most appropriate word please. Okay, what are they called as? Because they are born from two zygotes. Use one of the three words, most suitable word. What are they called? Because they are born from two zygotes. Very good. They do not look identical, though they can be similar. Use the best word among the three. Non-identical. Very good. Okay, they don't look identical, but they are as similar as siblings. Use the best word among the three. Very good. Wonderful. Thank you so much. That is about the dizygotic twins. Okay. And dizygotic twins are also born just like the same diagram will explain dizygotic twins also. The only point is this zygote, the both zygotes are formed in the same day. That's it. If these two zygotes are formed one after the other with 1 or 2 years gap, then they will become two siblings. If these two zygotes are formed on the same day, the babies develop together and born on the same day, they are called dizygotic twins. So the same diagram will explain the dizygotic twins also. And the dizygotic twins can be two females, two sisters or two brothers or a brother and a sister. Because if both eggs fuse with X-type sperms only, then two sisters will be born. Keep completing my sentences. If both eggs fuse with Y-type sperms, then two, complete my sentence, then two brothers will be born. Very good. If the two eggs fuse, one with the X-type sperm and one with the Y-type sperm, then one brother, one male and one female child will be born. Okay. So the dizygotic twins can be, okay, either two males or two females, okay, or a male and a female. Now coming to, okay, the second type of the twins, identical twins. Here what happens? Okay, they are also called as monozygotic twins. Identical twins are also called as monozygotic twins. Okay. Why they are called monozygotic twins? Because they are born from the single zygote. Okay, one egg is released, it will fuse with only one sperm, it will form only one zygote. That one zygote grows into only one embryo. Okay, you can see one egg cell, one sperm fused to form only one zygote. That will grow into only one embryo, but at one point of time that embryo splits into two. Okay, so this embryo splits into two, you can notice here. Okay, so you got two embryos. Okay, but they are not two embryos from two zygotes, they are the two embryos from a single zygote because the embryo just got broken down. Okay, these two embryos will form two babies. These two babies are called as, okay, dash, because they are born from a single zygote. Use most appropriate word between the two. They are called dash because they are born from a single zygote. Monozygotic twins. Monozygotic twins. Okay. Next. They are genetically identical. They are genetically identical. Okay. Therefore, they are called as, use the most appropriate word please. They are genetically identical. Therefore, they are called identical twins. Is it clear? Okay. Then why am I stressing that they are genetically identical? Because look at here, they are only genetically identical. But phenotype means what? Appearance. Can differ, okay, due to environmental or external influences. Okay, for example, two people are identical genetically, but one is a software engineer and the other one is a marketing executive. Then the marketing executive can have a little darker skin color, okay, or one uses a better shampoo, can have more hair on the scalp. Okay, so like that, okay, the phenotype, appearance can be slightly different. Why are they called identical twins? Because they are genetically identical. Okay, I hope it is clear. Done. Okay, Next. Now, we are discussing the first technique, that is cloning. Cloning is also called as somatic cell nuclear transfer. Somatic cell nuclear transfer. Okay. So, what is cloning? Okay, complete focus please. Let us suppose we all will get 50% of the characteristics from the mother and 50% of the characteristics from the father. Am I correct? Okay. We get 50% similar to mother, 50% similar to father. Not exactly, but roughly. We get characteristics of both the parents. But I don't want like that. I want a copy of Einstein. But of course, I am using a hypothetical example. We cannot clone Einstein because, okay, he is no more, and we also do not have his somatic cells preserved. So, I am just taking a hypothetical example because all our favorite scientists. Okay. So, let us suppose that, okay. So, let us suppose that, okay, Sangam PDF, regarding PDF I already told you, okay, and it looks like someone will not leave me till I speak about the PDF. Okay, I have given the Telegram link in the beginning. There you can download the PDF. First understand the class, after that PDF. Is it clear? Okay. If it is like this, I cannot even interact with you. If you spam the chat box like this, I will have to stop interacting with you people. So kindly focus on the topic. Okay. So we are discussing about the cloning, right? Okay. So what is the purpose of cloning? Okay. In general, a normal child will get some characteristics from the mother, some characteristics from the father, okay, and but I don't want like that. I want a copy of Einstein. I want 100% characteristics from only one person. That is called as clone, okay. The word clone means copy, and we can define clone as the child of single genetic parent. Okay, the clone is, okay, the child of single genetic parent. Genetic parent means what? Okay, will receive chromosomes from where? Okay, like biological parent is the one who gives birth to the baby. Genetic parent is the one who gives genetic material to the baby. I hope the difference is clear. Biological parent is the one who gives birth to the baby. Genetic parent is the one who, okay, gives genetic material to the baby. The clone is a child of a single genetic parent. It means a clone will get, okay, all 46 chromosomes from only one parent. Okay, how is it possible? I will explain this particular topic in 3 simple steps. Complete focus please. For example, I want Einstein's clone. Then what should I do? Take one egg cell from a donor. Okay? Take one egg cell from a donor. Right. But second step, remove the egg nucleus. Okay? Look at here. Egg cell taken from the donor. Second step, what you have to do? Remove the egg nucleus. Why am I removing the egg nucleus? Because I want Einstein's clone. I don't want this donor's chromosomes in that. If the donor's chromosomes are there, the baby looks like the donor. But I want the child to look like Einstein and want him to be as intelligent as Einstein. So remove the egg nucleus. It means you are taking only the egg cytoplasm. Is it clear? And egg cytoplasm is compulsory to make a zygote because it will have all the nutrients for the fetal development. Egg cytoplasm, like the egg yolk, will have all the nutrients for the fetal development. Not only that, you know, mitochondria, the powerhouse of the cell. Okay, a normal cell, for example, my skin cell will have 1500 mitochondria, but my egg cell will have 1,50,000 mitochondria. Okay, it means, okay, the egg cytoplasm is special. It is not like the cytoplasm of the other cells. So I cannot make zygote in a somatic cell. I cannot make zygote in a sperm cell. Even artificially, if I want to make a zygote, I have to take the egg cell. So egg cell taken from the donor, but remove the egg nucleus. It means you have taken only the egg cytoplasm, which is having all the mitochondria, all the nutrients, that is roughly all the material required for the fetal development. Then now coming to the third step. Third step is simple. Okay. Now a zygote should have how many chromosomes in that? Everybody please. A zygote should have how many chromosomes? 23 or 46? Look at here if you want. Okay. A zygote should have 23 or 46? 46. Then tell me, in Einstein's body, which cell will have 46 chromosomes? In Einstein's body, which cells will have 46 chromosomes? Sperm cell or somatic cell? Somatic cell. So, okay, in the egg cytoplasm, transfer the somatic cell nucleus from the person to be cloned. Okay, you want to clone Einstein? So take his somatic cell nucleus. Why only somatic cell nucleus? Because only that one will have all 46 chromosomes. Okay, then the resulting cell is called as a cloned zygote, not a natural zygote. Remember, we are making this artificially in a laboratory. Is it clear? I have taken the egg cell from a donor, removed the egg nucleus from that, and I have transferred the somatic cell nucleus from Einstein, and the resulting cell I got is called as the cloned zygote, not the natural zygote. Okay, then the cloned zygote, okay, is transplanted into the womb of a woman. So here you may get a doubt. Is a clone totally manufactured in the laboratory? No. Even a clone is born to a woman only. Cloning is artificial. Only till forming this cloned zygote, and then we implant it into the womb of a woman. Okay. So therefore, after 9 months, the clone will also be born. Okay. Just like a normal baby. The resulting baby is called as cloned baby. Okay. So simple, take the egg nucleus. From whom? You can take the egg nucleus from any lady. Einstein's sister, wife, mother, neighbor, anybody, or a random woman, because we are anyway removing the egg nucleus. So take the egg nucleus, take the egg cell, remove the egg nucleus, keep the somatic cell nucleus. The resulting cell is called as a cloned zygote, and implant it into the womb, and the resulting baby is called as the cloned baby. Okay. Hypothetically, okay, this cloned baby looks like Einstein, but okay, in practical reality it is not like that, because even external factors will influence. Okay, for example, intelligence also depends on your neighborhood, the company you get, your friends, the training you get, or the coaching you get, all these parameters. Even appearance slightly differs, skin color, height and weight of the person based on the food he consumes and all those things. So Einstein's clone need not look identical to him. Plus Einstein's clone need not be as intelligent as him. So therefore, cloning might not be as effective as we expect. So this is about the cloning. Is it clear? Okay, right. Now you can compare, this is a normal zygote, this is a cloned zygote. Okay, this is a cloned zygote. A normal zygote will also have egg cytoplasm plus 46 chromosomes. Egg cytoplasm plus 46 chromosomes. Cloned zygote also will have egg cytoplasm plus 46 chromosomes. But here all 46 chromosomes are coming from one person, whereas here the 46 chromosomes are coming from two people. 23 chromosomes from egg cell, 23 chromosomes from sperm cell. That is the difference between a normal zygote and a cloned zygote. I hope it is clear for all of you. Okay. Then cloning is of two types. Okay. One is reproductive cloning. Okay. Reproductive cloning. Okay. And what is reproductive cloning? Actually, this is called reproductive cloning only. Use the word, all of you. Reproductive cloning. Why this is called reproductive cloning? Okay, because here we reproduce a baby. Here we reproduce a baby. Therefore, it is called reproductive cloning. Okay, but if you don't implant it into the womb, okay, if you use the cloned zygote, look at here, if you use the cloned zygote and you grow that in the laboratory into cloned embryo, grow it into a cloned embryo in the laboratory, use it to manufacture organs like kidney, liver, heart, etc. That is called as therapeutic cloning. It means we are doing cloning for therapy, treatment purpose. Therapeutic cloning. I hope it is clear. Okay. So simple. You get the cloned zygote. Okay. After you get the cloned zygote, you keep it in the womb and get the baby. That is called reproductive cloning. But okay, if you grow it into embryo in the laboratory and make organs, these organs will be transplanted. For example, if my kidney fails, okay, I can transplant this kidney. I can manufacture kidney in the laboratory and keep it in my body. Okay, transplantation. Then this is called as, okay, therapeutic cloning. Therapeutic cloning. I hope it is clear for all of you. Okay, then, okay, now, why are we using this particular cloning? Is human cloning permitted? One very important point. Human cloning is not permitted. In any country, human cloning is not permitted. I hope it is clear. Yes. Okay. Then why human cloning is not permitted? We have several reasons. One is, okay, let us suppose, given a chance, every human being want to clone at least one person. For example, I love my mother. Okay, when my mother passes away, okay, I can't bear the pain. So I want to have another copy of my mother. Okay, then I want to clone my mother, or I may wish to clone my father. Okay, or somebody may wish to clone their wife or husband. Is it clear? So in such cases, if each of us decide to clone at least one, then imagine about the population. Plus, cloning can be misused. It can lead to a dirty business of selling the organs in the market. Okay, so cloning can be misused. Okay, so moreover, the clone, as I told you, need not look 100% like the parent. Einstein's clone need not look like him. He need not be as intelligent as him, because external factors also influence. It is not always genetic. So it is even meaningless also. Moreover, what if the cloned babies are born with some disorders, because they are artificial? You are partly making the babies in the laboratories. Okay, partly of course. Okay, so what if they are born with disorders? So it is against human rights. What if the families do not accept such babies? What about their emotional well-being? So for all these reasons, and law and order issues, for example, there can be a terrorist, you may try to catch hold of that person. Okay, you caught him and you killed him, but okay, that person is having 2-3 clones, it will lead to confusion. So due to all these reasons, human cloning is not permitted in any country. But we can use cloning, okay, to clone endangered species. For example, black-footed ferret, okay, is an endangered species that is cloned recently. Okay, so as a ex situ form of conservation, you know, in situ conservation, ex situ conservation. In situ conservation, conservation in its natural habitat. Ex situ conservation, outside its natural habitat. This is cloning. We are doing it in the laboratory. So therefore, as an ex situ conservation, endangered species can be cloned. I hope it is clear. Okay. So that is the story of cloning. Now moving on to the next topic, that is 3-parent baby. Okay. Cloning is child of a single genetic parent. This is child of 3 genetic parents. This is child of 3 genetic parents. Is it clear? Okay. To understand this, let us also try to understand what are genes. This is some background information. Okay. Directly I am not explaining the 3-parent baby. Okay. We are discussing some background information. First, what is a gene? Gene is any functional sequence of nucleotides on the DNA. Okay. See, DNA is a long chain of nucleotides, right? Look at here, if this completely is DNA, then one part of it is called as gene. Okay. So we have genes on the DNA just like we have words in a sentence. Is it clear? Okay. So that is how we have genes. Any functional sequence of nucleotides. Okay. Just like what is a word? Any meaningful sequence of alphabets. Same way gene is a functional sequence of nucleotides. Is it clear? Okay. So I compared gene with a word and I can compare mutation with a spelling mistake. Mutation is like a spelling mistake. For example, ATGCCTA produces insulin. Let us suppose, okay, that produce insulin. Is it clear? Then by chance if the A changes into T, then it cannot produce insulin anymore. Can't produce insulin anymore. In that case, you can get diabetes. Is it clear? So that is called mutation. Are the mutations good or bad? They are both. Mutation is a change in the gene sequence. Change can be a positive change or a negative change. Is it clear? What is a positive effect of the mutation? Evolution. Okay, starting from the bacteria, today we are at the stage of human beings. Bacteria evolved into human beings. Okay, monkeys evolved into human beings. How the monkeys evolved into human beings? Genetic changes, mutations. We look different from the monkeys. Okay, of course our mind is sometimes like monkeys, that is a different story. Okay, but okay, what all differences we have from the monkeys is because of the mutations. So mutations are also called as the raw material for evolution. Mutations are called as raw material for evolution. But okay, mutations can also cause diseases. I mean, if the mutation occurs in an important gene, like insulin gene is mutated, I cannot produce insulin. So then I will suffer from blood sugar levels, diabetes. Okay, mutations can also cause diseases. I hope it is clear. Okay, right. So, okay, gene, DNA is a chain of nucleotides, long chain, it is like a sentence. Gene is also a sequence of nucleotides, a small sequence like words in a sentence. We have genes in the DNA. And mutation is like a spelling mistake, okay, change in the gene sequence. If the gene sequence changes, that is called as mutation. Mutations are just a change, it's a neutral word, they can be positive or negative. Positive effect is evolution, okay, negative effect is diseases. This is some background information, okay. Next, okay, most of our DNA is in the nucleus, isn't it? 99.9% of our DNA is in the nucleus, okay, but you know, mitochondria will have 0.1% of DNA, okay. We also have a little DNA in the mitochondria as well, remember this point, okay. Now let us suppose, now complete focus please, okay, and one small disclaimer, okay, I cannot repeat much in this particular class because Usually this particular content, if I teach slowly at my pace, it is total 90 hours class. But today my target is to complete this in 12 hours with 1 hour overall break in between. It means 11 hours I have to complete it. It means I have to proceed 9 times faster in this particular class. Okay. So, only to motivate you, let me tell you one thing, that is when an old lady like this is standing here and focusing so much, okay, I think you have to focus more than I do because you are young, right? Okay, and energetic. So, complete focus please. Okay, so this is like as I told you, 99.9% DNA is in the nucleus, but we have 0.1% DNA, okay, in the mitochondria. I hope it is clear. Okay, right. Now, let us suppose there is a woman and her mitochondrial DNA is mutated. Her mitochondrial DNA is mutated. Mutations in mitochondrial DNA cause diseases like Alpers syndrome, Leigh syndrome, etc. What are they? Nothing to understand. Okay, nothing to understand. They are the names of the diseases. Alpers syndrome, Leigh syndrome, what are they? They occur because of the mutations in mitochondrial DNA. Done? Okay. Now, let me tell you another small background information. In general, what we think about fertilization, we think that in fertilization, egg and sperm will fuse with each other. Egg and sperm will fuse with each other, but no. Okay, what happens here is, look at here, this is egg cell. This is egg cell with egg nucleus. Okay, and this is sperm cell. This is sperm cell, okay, with the sperm nucleus, right? Okay. Now, what happens is during fertilization, only the sperm nucleus goes into egg cell. Okay. It means sperm cytoplasm is not coming into the egg cell. Is it clear? During fertilization, which part of the sperm cell goes into the egg cell, all of you? Now the egg cell became zygote. During fertilization, which part of the sperm nucleus goes into the egg cell? Only the nucleus. Sperm cytoplasm is not going into the egg nucleus. Okay. So total cytoplasm of the zygote is only from the total cytoplasm of the zygote is only from which cell? Egg cell or sperm cell or both of them? Very good. Is only from the egg cell. Next, where are the mitochondria located? Mitochondria are in the cytoplasm. Is it clear? Okay, mitochondria are located in the cytoplasm. Okay. Then club all these points and tell me, okay, if the father's mitochondria are defective, if the father's mitochondrial DNA is mutated, or if the father is having this Alpers syndrome, Leigh syndrome, can the father inherit this disease to the child? If the father's mitochondria are mutated, okay, can the father inherit these mitochondrial diseases to the child? No. Why? Because sperm cytoplasm is not going into the zygote. From the sperm cytoplasm is not going into zygote. It means I never got my father's cytoplasm. I got only my father's sperm nucleus. So if my father's mitochondria are defective, it will no way influence my health. Is it clear? So, okay, from this what you have to understand is mitochondrial diseases, okay, are inherited only from the mother. We call it as maternal inheritance. I repeat, during fertilization, only the sperm nucleus goes into the egg cell. Sperm cytoplasm is not going into the egg cell. Okay, So therefore the child will never get father's sperm cytoplasm, means a child will never get father's mitochondria. The child will get all the mitochondria only from the mother. Okay, so mitochondrial diseases like Alport syndrome, Leigh syndrome are inherited only from the mother, maternal inheritance. I hope this point is clear. Okay, now, okay, let us suppose there is a woman. Okay, this woman is having Leigh syndrome, Alport syndrome or Alport syndrome. Okay, now she want to have a baby. Okay, she want, okay, complete focus and answer this please. She want the baby to have her features but not her diseases. Okay, the baby should have mother's features. For example, let us suppose I have Alport's disease. Okay, I want to have a baby and I want the baby to look like me, but I don't want the baby to suffer like me with the Alport syndrome. Then I want to take something from a donor. Suggest me what should I take from a donor? Everybody please. I want to take something from a donor. What I should take from the donor? Okay. Cytoplasm. So what should I do is, I'll tell you in 3 steps, listen carefully. Step number 1. Okay. Take the egg cell. I'll tell you the total scenario once. I have Alport syndrome. I want to have a baby. I want the baby to look like me but should not have Alport syndrome. Then what I should do? Take the donor's egg cell, okay, and remove the egg nucleus. Okay, remove the egg nucleus. Means you have taken only the egg cytoplasm. Egg cytoplasm. That is, you have taken the mitochondria from the donor. Is it clear? Then you may ask me, why can't you take my own egg cell and remove the mitochondria? Remember, we have 1,50,000 mitochondria. So if I take my egg cell, then I have to go on remove 1,50,000 mitochondria, which is easy. So leave my egg cell, take the donor's egg cell, remove the nucleus, single nucleus you have to remove. So you will get mitochondria from the donor. Is the step clear for all of you? Yes. Okay. Pricking each mitochondria 1,50,000 times is like showing where is your nose, means I have my nose here. But I can simply show my nose is here. Is it clear? So that is the reason why simply what you can do is take the donor's egg cell, remove the nucleus. Okay, why do I want to remove the nucleus? Because I want my baby to look like me. I just don't want the baby to have Alport syndrome, that's all. Remaining all, I want my baby to look like me. So remove the egg nucleus. And then transfer, okay, the egg nucleus from my body, okay, my egg nucleus, my husband's sperm nucleus. Is it clear? So total 3 people giving genetic material to this baby. Is it clear? 3 people giving genetic material to this baby. How? Okay, count the 3, all of you. Okay, number 1. Mitochondrial DNA is coming from the donor, right? 23 chromosomes are coming from the mother's egg cell. 23 chromosomes are coming from the father's egg cell. Is it clear? So 3 people contributing to this baby. So the child of 3 genetic parents is called 3-parent baby. Okay. What if I transfer this zygote into a 4th lady and the 4th person gives birth? Can I call it as 4-parent baby in that case? Take donor's egg cell, my egg nucleus, my husband's sperm nucleus, the resulting zygote into another lady, surrogate mother. 4th lady will give birth. Can I call that baby as 4-parent baby? No, we cannot call that as 4-parent baby because the 4th one is biological parent, not genetic parent. Genetic parent is the one who gives DNA to the baby. How many people are giving DNA to this baby? 3 people. So therefore this is called as 3-parent baby. I hope it is clear. Okay. What is the purpose of this technique? The purpose of this technique is only to avoid the mitochondrial diseases. What you can do with this? Only avoid mitochondrial diseases, nothing else, only to avoid the mitochondrial diseases. Is it clear? Okay, next. Therefore, this is also called as mitochondrial replacement therapy. Okay, 3-parent baby is also called as mitochondrial replacement therapy. Okay, and we can also call it as pronuclear transfer. We can also call it as look at here, it is written here. Okay, and if you notice, I have made all these drawings yesterday to save today's class timing. I mean, if I write all these in today's class, it will consume another hour extra. So I had to make all these drawings yesterday. Okay, so also called as pronuclear transfer. Why this is called pronuclear transfer? What do you mean by pronuclei? Okay, pronuclei means, okay, the nucleus of egg and sperm during the process of fusion. Okay, egg nucleus, sperm nucleus, while they are still fusing. I mean, when the fertilization is still going on, when the fertilization is still in the process, in that stage, the egg and sperm nuclei are called as pronuclei. I hope it is clear. Okay. Therefore, this is also called as pronuclear transfer. Pronuclear transfer. What is pronuclei? Okay, the sperm and egg nucleus during the process of fertilization. During the process of fertilization, right? So, what are the three names? 3-parent baby, mitochondrial replacement therapy, pronuclear transfer. Purpose is to avoid the mitochondrial diseases. Okay, there are 2 previous questions based on this topic. Okay, look at here. In the context of hereditary diseases, consider the following. Passing on mitochondrial disease from parent to child can be prevented by mitochondrial replacement therapy. Okay, either before or after the in vitro fertilization. Yes, correct. Okay, a child inherits mitochondrial disease entirely from the mother, not from the father. Correct. Okay, both 1 and 2 is the right answer. Okay, next question. In the and I cannot ask you to solve the question because PYQ, by chance if you have missed it, a few days ago, okay, as the 4th phase of Conquer Prelims, we have done 10-year complete marathon on 10-year previous year questions. Okay, so in that we slightly tried to solve those questions, but in today the main target is revision. So I am just showing you the question, but I cannot give you a few seconds of time for you to solve them, right? Okay, next. So there is another question here. In the context of recent advances in human reproductive technique, pronuclear technology is used for prevention of mitochondrial diseases. I hope it is clear. Next. Okay. After that, we are moving on to the next topic, that is assisted reproductive techniques, also called as artificial fertility techniques. I mean, okay, let us suppose if a couple is infertile, the wife or husband is infertile, okay, it's been a few years after their marriage but they are unable to have a child. Then we have some medical procedures to help them have a child. They are called as, I mean, we give medical assistance to help them have a child. That is called as assisted reproductive techniques. In that, okay, the first one is called as cell banks. Cell bank, is it clear? Okay, what do you mean by a cell bank? Just like you know blood bank, you know, eye bank, you know, like that you have cell bank also. Okay, here the cells are preserved in extremely cold conditions, okay, in the liquid nitrogen. Okay, liquid nitrogen temperature is -190 degrees Celsius. At such a temperature, when you preserve the eggs and sperm cells, okay, completely they are frozen. Is it clear? They can be stored up to 100 years also. After 100 years, the eggs and sperms will be as fresh as they were 100 years ago, because we are just freezing the life processes. Okay, nothing proceeding inside that particular cell. Okay, so these are called as cell banks. I hope it is clear. Okay, now using these sperm banks or egg banks, what we can do? First one, in vitro fertilization. Use the word once, in vitro fertilization. Okay, look at here. I'll explain all the procedures using this diagram. Okay, look at here. This is the female uterus. Look at this part. This is the uterus. Clear? Then, okay, this is the opening of the uterus. We call this as cervix. Cervix, opening of the uterus. Quick responses please. Use the word once. Cervix, uterus. And what are these? This one, this is ovary. Ovary will release the eggs, isn't it? Ovary will release the eggs. Okay, this is ovary and these are the eggs. Okay. Usually a woman releases only one egg per month. In the complete month, a woman releases only one egg cell. But here what you are doing is, we are giving hormones to that woman to make her release more egg cells. To make her release more egg cells. Is it clear? I repeat, naturally a woman gives only one egg cell per month, but we are giving some hormone treatment here, okay, so that the woman will release more egg cells. And this tube is called as, can you see a tube between the ovary and the uterus? That is called fallopian tube. Okay, the egg will travel through this fallopian tube into the uterus. Is it clear? Egg travels into the uterus through fallopian tubes. Okay, and from where do the sperms enter? Sperm naturally enters from the cervix. Is it clear? Sperm naturally enters from the cervix. Okay, and where do the fertilization occur? The site for natural fertilization. The site for natural fertilization is fallopian tubes. Understand all the topics and keep remembering them well, please. Okay, fallopian tubes. Okay, naturally fertilization occurs in the fallopian tubes. Okay. Sperm enters into the cervix and this is how the sperm will travel. I hope you understood the route through which the sperm travels. Sperm, cervix, uterus. From the uterus it will go into the fallopian tube. And how the egg will come here? From the ovary. They both will fuse in the fallopian tube. That is where the natural fertilization occurs. Is it clear? Done? Okay. Now, so here, okay, let us continue with this. First step what we have done, given the, remember step by step. Step 1, hormone injections and we stimulated the egg production. The woman released more egg cells. Step 2, look at here. Multiple eggs are taken from the woman's ovaries. Look at here, multiple egg cells. Did you follow the step 2? Multiple egg cells are taken into, okay, a petri dish, okay, or a test tube, like a test tube, okay. Okay. Then step 3. In the laboratory, the eggs are mixed with the sperms in a culture dish. What is step 3? Look at here. Okay, sperms are added into this culture dish or the test tube. Okay, I hope the steps are clear for all of you. Okay, then next, what is the 4th step? Okay, then all of you tell me, normally where do the fertilization occur? What is the site for natural fertilization? Fallopian tube. This is called as in vivo fertilization. In vivo means inside the living system. Inside the living system. Okay. Whereas in the test tube baby, In the test tube baby technique, what happens? Fertilization occurs in the test tube or the culture dish. Therefore, that is called as in vitro. In vitro means in the laboratory or in the glassware. Okay, like test tube or culture dish, like that. Therefore, test tube baby is also called as in vitro fertilization. Okay, fertilization will occur. Okay, for that we have to keep it in an incubator for 48 hours. Okay, in this 48 hours, the zygote will also start dividing and will form the embryo. What is the next stage? Embryo. Then we have two options. Either, okay, implant the embryo into the woman's uterus. Implant the embryo into the woman's uterus. Embryo. Or else, okay, freeze the embryo for future use. I made the embryo, but I don't want the baby now. I want the baby 10 years later. Okay, 10 years I can keep it in the liquid nitrogen. Is it clear? Okay, so now, so I repeat, step 1, okay, give hormone injection. Okay, step 1, give hormone injection. Stimulate the egg release. More eggs are released, multiple eggs. Step 2, take multiple egg cells into a culture dish. Step 3, add sperms to it. Okay. Step 4, keep it in the incubator, you will get the embryo. Step 5, either implant it into the uterus so that you can have a baby now, or freeze it in the liquid nitrogen so that you can have a baby in future whenever you want. Okay. So, this is like the in vitro fertilization. Okay. Then after in vitro fertilization, the second one is called as, okay, embryo transfer. Both are roughly the same. Okay, I'll just explain it. Look at here. Okay, you have taken the egg cell and then you fused it with the sperm and then you got the zygote. If you keep this, okay, into the womb, that is called in vitro fertilization. But if you grow this and then you transfer this embryo into the womb, that is called embryo transfer. So the only difference is at what stage you are transferring it into the womb. Okay. If you transfer at the zygote level, IVF. If you transfer it at the embryo level, it is called as embryo transfer. You can also call frozen embryo transfer also. By chance, if you are not like implanting a fresh embryo, you are implanting a frozen embryo, okay, then it is called FET, frozen embryo transfer. I hope it is clear. Okay, then embryo cryopreservation, frozen embryo transfer. Done. Next, artificial insemination. Okay, sometimes what happens is, okay, let us suppose the wife is normal, able to release the egg cell. Husband is also healthy, able to release the sperm cell. Okay, so no need of an egg donor or a sperm donor. But due to some reason, let us suppose the man is suffering from a condition called as oligospermia. Use the word once, all of you. Oligospermia. Oligo, the word oligo means few. So oligospermia means what? Lesser sperm count. Okay. In this condition, okay, is natural childbirth possible? Possible. But the pregnancy may be delayed. So it's been already 2-3 years after the marriage. Now they want to have a baby, but they are not getting the pregnancy naturally. Then what we can do is, you need not take a sperm from a donor. You need not mix it in this culture dish. But what you can do is, okay, you can just, okay, look at here. what you can do is you can just collect the semen from the husband. Okay, no need of a donor. The event is between the wife and husband only. You are not taking the sperm from another person. Just collect the sperms from the man, inject them at the cervix. Okay, we call that condition as ICI, intracervical insemination. Use the word once, intracervical insemination. Okay, intracervical insemination, that is inject sperm into cervix. Okay, next, intrauterine insemination. I mean, if you inject the sperm here, directly if you inject the sperm here, intrauterine insemination, okay, means what? Inject sperm into uterus, okay, or okay, give them a gift. Give them a gift. So what do you mean by gift? Okay, gamete intrafallopian transfer. I mean, if you give them here, okay, egg plus sperm, if the sperm injected here, okay, where fallopian tubes, that is called gift. Is it clear? All the three together is called as, okay, artificial insemination. Okay, I repeat, artificial insemination means the sperm is not naturally reaching the egg cell. So you have to inject the sperm. Then if you inject the sperm at the cervix, it is called intracervical insemination. If you inject it at the uterus, that is called intrauterine insemination. If you inject it in the fallopian tubes, it is called as gift. Gamete intrafallopian transfer. I hope it is clear. Right. Sperms and eggs are mixed and injected into the fallopian tubes. Then what do you mean by this word? Gamete. Gamete means, okay, egg or sperm. Female gamete is egg cell. Male gamete is sperm cell. So gametes means reproductive cells. I hope it is clear. Okay. Right. Next. Okay. After that, the next technique is called as surrogacy. Okay. In surrogacy, let us suppose the wife is able to release the egg cell. Husband is able to release the sperm cell. So they do not need an egg donor or a sperm donor. But due to some reason, she do not want to give birth to a baby. Like for example, you can take the case of Priyanka Chopra, okay, or any such cases. Is it clear? Somehow, okay, you do not want to carry the pregnancy. Either you don't want to carry the pregnancy or you have a medical condition in which your uterus is not healthy. You are able to release egg cell, but uterus is not healthy. In that case, what you can do, take the sperm cell from the husband, egg cell from the wife, okay, then fuse them in this laboratory petri dish. Is it clear? Form the, all the procedure is the same. Just if you inject this into the womb of another woman, is it clear? Into the womb of another woman, that is called surrogacy. Okay, same here. Take the egg cell, take the sperm cell, mix them in the culture dish or test tube. In vitro fertilization, incubate it, you will get embryo. Okay, same like IVF or embryo transfer, but the embryo or the zygote is not transferred into my body. It will be transferred into another lady's womb. She will carry the baby and give that baby to me finally. Okay. So, if a woman carries a baby for another couple, it means if I have a child, not for myself, if I have a child for somebody else, if I am carrying another couple's baby for them, after the birth I should give the baby to them. In that condition, I will be called as surrogate mother, and the technique is called as surrogacy. I hope surrogacy is clear. Yes. Okay. Next, you have ART Act, Assisted Reproductive Technologies Act. okay, and Surrogacy Act. Both the acts are recently passed, both the bills are recently passed. Very important, go through, but I will not explain it because there is no technical element in that. Okay, I have given the content in the news, you can go through that. I can use the time to explain some other technical content, right? Can we proceed? Surrogacy Act 2021 and Assisted Reproductive Technologies Act 2021. Both, okay, acts came into being in the year 2021. Clear? Okay. Next one, next topic, we are proceeding with artificial womb. Artificial womb. Okay. Artificial womb means what? Okay. Till now what all we have seen is, okay, only making up to embryo in the laboratory, isn't it? Till here what all we have seen is all ART techniques like IVF, okay, or embryo transfer technique or frozen embryo transfer or ICI or IUI, okay, or like whatever you take, all these are artificial only till embryo. Finally, you need a female to carry the pregnancy, right? Okay, but here, okay, no need of a woman to carry the pregnancy, okay. It is something like the total baby made in the laboratory. This baby is not born, this baby is made. Is it clear? Not born but made, like that. You don't need a female to carry this. That is called as artificial womb. Okay, I'll explain the artificial womb, okay, with a small disclaimer. Now we are preparing for prelims, so you don't have to explain the process in the exam. So you don't need this step by step, everything must be clear, okay. Remember that point so that, okay, you can precisely work for the prelims. Is it clear? Okay. Now, so let us see about the, okay, this artificial wombs, how it will work. Okay, complete focus here please. Okay, right. What is this? This is a premature lamb fetus. Okay, till now did we make any human baby in the artificial womb? Till now did we make any human baby in the artificial womb? No. Okay, so the only animal which is made in the artificial womb is a lamb fetus. The only animal which is grown in the artificial womb is lamb fetus. Okay, why? Just as an experiment. Is it clear? You don't need an artificial womb to give birth to a lamb. We have so many sheep over there. Okay, so like the goat, okay, the mother will give birth to the baby. You don't need artificial womb. It's not an endangered species. Okay, so here, okay, we are doing this only for an experiment, so that if this works, in future we can try. In future we can try with, okay, endangered or even the extinct species, even the endangered or extinct species. Is it clear? Okay, so I hope this first part is clear. Okay, premature lamb fetus in a temperature controlled biobag. What is biobag? Okay, this is also called as biobag. Artificial womb is also called as biobag. Yes, clear for all of you? Right. Then, now what is biobag? In a way, what all is available to the baby in a natural womb, We should provide all of them in this bag, isn't it? Okay. Whatever a fetus will get in the natural womb, we just have to provide all of them here. The first and important thing here is amniotic fluid. I think you must have heard, during the pregnancy, the fetus is not in the dry conditions. It is immersed in a liquid called as amniotic fluid. Okay. So we should also arrange amniotic fluid in this bag, because we have to keep everything which is available to the normal fetus in a natural womb in this bag. And this amniotic fluid flows out of the bag, okay, enabling the fetus to breathe as it would do in the natural womb. It means we keep this amniotic fluid circulating. Why? Okay, because over a period of time, toxins will be accumulated in this and the nutrients will be depleted. Is it clear? So the amniotic fluid comes out. We remove the toxins and add the nutrients. Again it will go. Again it comes out. We remove the toxins, add nutrients. Again it goes. So like that, the amniotic fluid is kept flowing through this particular bio bag. I hope it is clear for all of you. Okay. Then next one, the fetus lungs are not yet developed. Is it clear? The fetus lungs are not yet developed. So what we do? Okay, we keep an oxygenator. Look at here, oxygenator. Okay, and also what we do here is, look at this red color tube here. Okay, and observe the arrow mark, please. Observe the arrow mark. Okay. Where the tube is connected? The tube is connected to the umbilical cord. The tube is connected to the umbilical cord. Is that clear? The tube is connected to the umbilical cord. What is going in that tube? All of you tell me. Blood. Blood is going into the fetus through that umbilical cord. Is it clear? Then, now what happens? Look at this arrow mark. Okay. The blood is coming out. Okay, the blood is coming out. Which blood is coming out? Deoxygenated blood plus impure blood. Which blood is going here? Oxygenated blood, pure blood. Okay, oxygenated pure blood is going into the fetus. Fetus will utilize that blood, and the deoxygenated blood is coming out, and the oxygenator, what it will do? Oxygenator, what it will do? It will oxygenate or purify the blood, just like lungs. Just like lungs. I hope it is clear. Okay. Then next, another part you have to observe is a drip bag, like saline, a drip bag. In that we are keeping the nutrients for the baby to grow. Nutrients are provided through this drip bag. Please note that it is connected, okay, to this blood circulation. Is it clear? The drip bag is connected to the blood. Is it clear? Okay. So we send the blood, nutrients and oxygen will be completed. Such blood will come out. We add the nutrients and we add the oxygen and send it back. Again nutrients and oxygen will be utilized by the fetus. Again such blood comes out. We add nutrients here. We add oxygen here, and then we again send it back. Is it clear? So digestive system, respiratory system, circulatory system duty the machines are doing here. Is it clear? So the baby can grow despite the fact that the baby's circulatory, digestive and respiratory systems are not working yet. I hope it is clear. Yes. Lastly, excretory system is also not developed in this baby. So what we do? The waste is disposed through dialysis. You know, kidney patients, we do dialysis. Same way the wastes from the blood are removed through dialysis. Okay, dialysis, I think it is also given in your 9th class book. Okay, in the 9th class NCERT also you have dialysis. Is it clear? Okay, I repeat. Okay, point number 1. Okay, white color, written in white color, observe it, premature lamb fetus. Point number 2 in yellow color, amniotic fluid. Okay, this is kept circulating so that it will be fresh. The third one, an oxygenator. Okay, we add oxygen here and a drip bag that will add nutrients. From the drip bag, nutrients are added to the blood. From the oxygenator, oxygen is added to the blood. Such blood goes into the lamb fetus. Okay, the oxygen and nutrients will be utilized by the lamb fetus. The blood comes out. Again nutrients and oxygen is added to it. Again the blood goes in. And then toxins must be removed from the blood. That is done by dialysis. It means digestive, circulatory, respiratory, okay, and excretory systems are still not working in the baby. We are doing all of them with the help of the machines. So this is called an artificial womb. Is it clear? Okay. Then why do we need an artificial womb? Okay. Remember surrogacy, I will give you one important point. Okay. Do you remember recently there is one news called dire wolf? Dire wolf. Okay. Kind of de-extinction. Okay. De-extinction. Okay. You might have heard the news. I mean, dire wolf became extinct. We brought it back. How? We used a dog as a surrogate mother. We used a dog as a surrogate mother. I hope it is clear. Okay. But usually the success rate is less. Success rate is less. Can you imagine a human baby being born to a donkey? Human baby born to a monkey? Okay. It means one species cannot give birth to the other species young one, isn't it? Is it clear? One species cannot give birth to the other species. Here, this is the first case like this in the history of biotechnology. This is the first time such a de-extinction was achieved. We are also trying, okay, to use southern white rhino as a surrogate for northern white rhino, but not yet successful. Not yet successful. I hope it is clear. Okay. So, it is very difficult. So, just like a human cannot be born to a donkey or a monkey, dire wolf being born to a dog is not an easy task. After decades of effort, this is the first successful case, which is difficult. But why we had to inject the zygote into the dog womb? Why not into the dire wolf womb only? Because the dire wolf already became extinct. You don't have a dire wolf female to carry the pregnancy. Is it clear? So that is the reason why we had to choose the dog. Is it clear? But what if the artificial womb comes out? Okay, we can use them for de-extinction. You don't need a female. Is it clear? Okay, directly in the artificial womb you can bring back the extinct species. I mean, more such experiments like dire wolf are possible. Is it clear? Okay, clear for all of you? Right, that is the story of artificial wombs. Okay, and I told you already, using frozen sperm of northern white rhino. Okay, see, northern white rhino is a species. In this southern white rhino, white rhino is one species. In this southern white rhino, northern white rhino are two subspecies. Is it clear? this is, okay, last male died. Only 2 females are there, but they became old. Is it clear? Okay. So what they have done? Okay, what they have done is the frozen sperm of northern white rhino, the egg of southern white rhino, okay, and implanted it into the southern white rhino's womb. Okay, I'll show you here. Okay. Egg of southern white rhino, sperm of northern white rhino. You got southern white rhino, northern white rhino, hybrid, zygote. This is kept into the womb of southern white rhino, but not yet successful. Clear till now? Okay. Right. So this is how we can use artificial fertility techniques in the biodiversity conservation. So with this assisted reproductive techniques, okay, or artificial fertility techniques completed, moving on to the next topic called as stem cell technology. Moving on to the next topic, that is stem cell technology. Okay. What are the stem cells? Stem cells are like, okay, raw material. Okay, stem cells are like raw material. Okay, like for example, if you have stem cells, okay, stem cells can be converted into any other cell like kidney cell or liver cell or heart cell, okay, or lung cell, okay, or skin cell, like that. Is it clear? The cells that can be converted into other cells are called stem cells. Done? The cells that can be converted, undifferentiated cells, okay, or partially differentiated cells, okay. It means what? They are nothing. And I mean like a white wall, and can be made into anything. I hope you got the point. It is not a liver cell, it is not a kidney cell, it is not a skin cell, it is not a brain cell, but it can be made into any of those cells. It is nothing but can be made into anything. It is like a white wall on which any color you can apply. Okay, those are the stem cells. Clear for all of you? Right. Undifferentiated or partially differentiated cells that can differentiate into various types of cells plus proliferate indefinitely. Stem cells have two properties. First property, convert to other cells. Second property, okay, proliferate, okay, indefinitely. I mean they can increase their number by cell division. Is it clear? They can increase their number by cell division. I hope it is clear. Right? So stem cells are the raw material. We can also call them as precursor cells. Precursor means what? Raw material. Okay, before. Okay, they can be made into product. Stem cells are precursor. Product is different organ cells. Clear till now? And these are the stem cells have two properties. What are they? First, they can be converted into other cells. Second, they can grow unlimitedly. Their number can be increased unlimitedly. They can keep on increasing their number. How do they increase their number? By cell division. Is it clear? Okay. Then why do we use stem cell technology? To manufacture copies of one's own organs. For example, let us suppose a person's kidney fails, he need to take a kidney from a donor. First thing, getting a kidney can be difficult, costly, okay, or sometimes a matching donor will not be available at all. Okay, still kidney means to some extent it is okay, possible, because we have 2 kidneys, maybe somebody will agree to give me a kidney. But what if heart fails? Okay, people have only one heart and they don't want to give it to anybody. Is it clear? Giving heart is possible only in poetry and movies, but in medical terms nobody will be ready to give the heart. So from where do we get? Yes, you may tell me that in the case of head injuries, when the person is brain dead, the heart can be transplanted to the other person, but very rare. Okay, wherever I get heart attack, at that person another person should die with a head injury, or in a nearby place at least, and that heart should match with my body. On the whole, what I am trying to explain is organ donation is difficult. Agreed? Is it clear? Difficult to get a donor. Even if you get a donor, it may not match. Even if you get a donor and match, it can be very costly. So what if, and most of the organs are in a single copy, so nobody will be ready to give the organs. So on the whole, organ donations are difficult. So what if I can make more copies of my for example, an accident, okay, hand is amputated, no worry, new hand, okay, brain head injury, no problem, new brain, fresh memories also. Is it clear? All your psychological trauma that you experienced, you can forget. A new heart, a new kidney, a new liver, that is a target of the stem cell technology. I hope it is clear. Okay, now we understood what are the stem cells and we understood the purpose or the aim of the stem cell technology. Now we are discussing the types of the stem cells. Okay, types of the yes, Pragnya, definitely you can forget the X if you get a fresh brain, right? Okay, now types of the stem cells. In this, first one is called as totipotent. Okay, totipotent precursor stem cell. Array, how lengthy the name is, very difficult to remember, is it? Okay, but no, just observe the word toti. Toti means what? Total. Potency means what? Capacity. Precursor means what? Raw material. Stem cell means you know. Okay. The only example for this is zygote. Why this is called totipotent? Because it can make the complete organism. That is, it can convert it into all cells without exception. I hope it is clear. The only cell which is totipotent in our body is zygote. Zygote can grow into the complete human being. It means it can get converted into all the cells. It can get converted into all the cells, any cell. Okay. Now the second word is pluripotent precursor stem cell. Okay. Just look at these words. Okay. They are quite easy to remember. Okay. Now same thing. If you observe, the words potent, precursor, stem cell are repeating. Same words here also. Here toti, here pluri. That is the only difference. Toti means they can be converted into any cell of the body, whereas here they can be differentiated into most of the other cell types. Not all. Most of the other cell types. Maybe you can make a heart, but you cannot make a brain, like that. I hope it is clear. Example, embryonic stem cells. Embryonic cells are pluripotent. If you have embryonic stem cells, from that you can make most of the organs of our body. Is it clear? Done? Next. Okay. Next one also, potent precursor stem cell word repeated. Just multi. Okay, observe the words, you can easily remember this. Okay, pluri means most of, multi means many. Toti means all. Toti means all. Clear till now? Right. Example, umbilical cord stem cells. Okay, what is umbilical cord? Okay, imagine this is the uterus. This is the fetus. Okay, and this is placenta. Okay, from the placenta, okay, a tube will connect to the baby at the navel. Is it clear? That is called umbilical cord. Okay, when the baby is born, the umbilical cord will be, okay, cut. Then you will get some blood. This blood contain, okay, multipotent stem cells. I hope it is clear. When a baby is born, you might have seen in some movies, the umbilical cord will be cut and then they will put a clip. Okay, so that umbilical cord, the blood which is coming out of the umbilical cord is rich in multipotent precursor stem cells. They can be converted into many other cell types. I hope it is clear. Okay, lastly, oligopotent. Okay, few. They can be converted into few other cells. Earlier also I told you, oligospermia, few sperms, fewer number of the sperms. Wherever the word oligo comes, it means lesser number, fewer. Okay, so we'll get converted into only few other cell types. Okay, what are those few? Remember, okay, they cannot be used to produce organs. They cannot get converted into liver, kidney, pancreas, heart, brain. But they can only get converted into different varieties of blood cells. Therefore, they can be used in the treatment of blood cancer. Okay, see, totipotent, pluripotent, multipotent precursor stem cells can be used to manufacture the organs for transplantation. But oligopotent cells cannot be used for organ manufacture because they cannot get converted into organ cells. They can only get converted into different varieties of blood cells. Therefore, they can be used in the treatment of blood cancer. I hope it is clear. Okay. Lastly, let us suppose today I want a kidney. Do I have my zygote? No. The zygote only became Bindu. Do I have my embryo? No. That embryo only became this Bindu. Is it clear? Okay. Do I have my umbilical cord? No. It was thrown when I was born. Is it clear? Okay. Means I do not have any of those. Then what to do? Then we have a solution called as IPSCs, induced pluripotent precursor stem cells. Induced pluripotent precursor stem cells. Complete focus here, please. So what we do here is you take somatic cell. Understood? I don't have zygote, I don't have embryo, I don't have umbilical cord cells. No worries. Take my skin cell. Got it? Everybody, take my skin cell. Done? And then what you have to do is inject okay, the embryonic gene transcription factors, some embryonic genes transcription factors. What are they? They are called, okay, LIN28, nanog, SOX2, etc. Okay. Then you may say that I did not understand LIN28, nanog, SOX2, nothing. They are the names to remember. Okay. I did not tell you the meaning of them. They are the names. Names of what? embryonic genes transcription factors. Is it clear? Then you are injecting them to what kind of a cell? Yes, everybody? Okay. And here once again I just wanted to remind you one thing. This is a complete revision series, the 5th phase of our Conquer Prelims and the last phase of our Conquer Prelims. It will be primarily used for the students who already attended all the classes and revising it. Is it clear? And also very useful for the people who never attended any science and technology class, so that something is better than nothing. And obviously we have to remember that, okay, you cannot eat some 10 kg sweet in one day, okay. The best thing is actually daily 1 hour class, 2 hours, that is the best thing, okay. But okay, here I am trying to give you everything from the basics, whatever I am teaching in the comprehensive course, but it cannot be as slow and as clear as we do it in the comprehensive course. So I want you all to realize that and completely focus on the class so that you can make the best use of this class. Is it clear? Okay. Now, so what are we taking? Somatic cell. What are we injecting into that? Why am I taking somatic cell? Because I don't have my zygote, I don't have my embryo, I don't have my umbilical cord. It was thrown in the dustbin. I was saying my mother only, but she was not listening, throwing it in the dustbin. Is it clear? So I don't have all of them. So take my somatic cell. Inject the embryonic genes transcription factors. Then what happens? Okay, the embryonic genes are activated. The embryonic genes are activated in what? The somatic cell. In the somatic cell, embryonic genes are activated. Is it clear? Okay. Then what happens? The somatic cell became like an embryonic cell. Is it clear? Okay. Then yes. Okay, look at here. Answer this question, all of you. Okay, if the embryo forms different somatic cells, we call that process as differentiation. Do you agree? We call that process as differentiation, because if you remember, okay, the embryo is defined as undifferentiated mass of cells. Means all the embryonic cells are similar to each other. Now the embryo is getting differentiated into different varieties of cells like blood cells, kidney cells, liver cells, heart cells, etc. That is called differentiation. But what will you call, okay, if the somatic cells are converted like embryonic cells? We call that process as, okay, de-differentiation. We call that process as de-differentiation, the reverse of differentiation. Is it clear? Or genetic reprogramming. Is it clear? You are hypnotizing the somatic cell. Hey somatic cell, you are not a somatic cell anymore. You are not a somatic cell anymore. Now you are an embryonic cell. So that is what we are doing, genetic reprogramming. You are reprogramming the somatic cell to behave like an embryonic cell. How you are reprogramming? You are activating the embryonic genes in that. Okay. Now this is like, okay, a converted pluripotent cell. Agreed? I mean a converted embryonic cell. Not original embryonic cell. Somatic cell converted into embryonic cell. Or a duplicate embryonic cell, isn't it? It's not an original embryonic cell. Somatic cell converted to embryonic cell. Duplicate embryonic cell. Okay? Or induced. What we call this as? Induced. Okay? Remaining word as it is. Just look at here, you will easily understand. Okay, tell me the example for pluripotent precursor stem cells. Embryonic cells, isn't it? What is the example for pluripotent? This is not a new word. Just you are adding another word called induced. Means you are inducing the somatic cell to become the pluripotent. Induced pluripotent means converted pluripotent. A somatic cell converted into pluripotent. Is it clear? So simple. I don't have pluripotent cells. No worries. Take a somatic cell, convert it into pluripotent. That is called iPSCs. Induced pluripotent precursor stem cells. For this technique, in the year 2012, Shinya Yamanaka won the Nobel Prize for this technique. Okay, of course, this Nobel Prize point is not important because it is from 2012, but iPSCs are important. They are always important. Okay, regularly we will have news articles on iPSCs and their applications. Is it clear? Okay, so that's it. Next one, printable organs. Okay, next topic is printable organs. Okay, also called as 3D organ printing. 3D organ printing. Is it clear? Okay, so here we have seen, the raw material is stem cell. Okay, raw material for what? To make organs. Okay, so we understood that we can make organs from stem cells. Fine, very good. But how to make the organs? From where means stem cells. Okay, but how? Okay, that is 3D organ printing. That is 3D organ printing. Okay, see, there is one previous question. Okay, 3D printing. Please note, not 3D organ printing. 3D printing has applications in which of the following? Okay, all the above is the right answer. I am putting this without seeing even the statements, because in such questions most of the times it is all the above will be the right answer, but don't do that blindly like I am doing it. Is it clear? I am in a consequence free stage of life now, but your mistakes will have huge consequences, so carefully read all the sentences and answer. Is it clear? Okay, now what is 3D printing? Let us suppose you have a printer in your office. Every printer will have a cartridge. Like your pen will have a refill, printer will have a cartridge. In that, what you will do? You will fill the cartridge with ink, black color ink or colored ink. That is a normal 2D print. Is it clear? But sometimes what you can do? You have 3D printers. In the 3D printers, confectionery items, it means you know something? Okay, cartridge filled with cake or cookie batter. In such situations, cakes and cookies should not be reminded, right? Okay, so the cartridge is filled with cakes or cookies batter so that we can print out real cookies that you can eat. Is it clear? You can literally print out a cake in the shape, design, color you want. I hope it is clear. Okay, manufacture of bionic ears. Is it clear? Means this is hearing aid for deaf people. Okay, automotive industry. So what you have to do is fill the cartridge, okay, and print out a car model. Did you understand? A car, new car I designed, I can print out a model. Okay, reconstructive surgery. Okay, for example, I can print out, I can literally print out, okay, a steel rod for surgery. My femur bone fractured, I want a rod, so that rod can be printed out. Okay, data processing technologies, I mean like a hard disk, can be printed out. Not only that, in the space technology we are going to discuss, okay, an entire, okay, engine, rocket engine completely printed out. Did you hear that? Okay, right? rocket engine printed out. Also concrete, you can fill the cartridge with concrete. Okay, concrete filled in the cartridge. Okay, and you can print out a building. I hope you understood. Complete building can be printed out. Okay, so in the 3D printing, we are not printing the images, we are printing the real structures. I hope it is clear. I am not taking the printout of the rocket engine's image on the paper. No, real engine that can carry the rocket. Real steel rod that I can keep in my after surgery. A real building inside which I can go and live. Okay, that is called 3D printing. Okay, not only in this, okay, you can also fill now kidney cells in the cartridge and print out real kidney, then arrange it in the body. Is it clear? This is how we manufacture the organs using 3D printing technology. So in addition to the 5 statements that they have given, I am adding a rocket, a building and organs. All these can be printed out using 3D printing technology. Okay, so now printable organs. For this 3D data about the organ is needed. For example, I want to listen very carefully, please, complete focus on the class. Okay, so I want to print out kidney. What is the procedure? Step 1, I need a CT scan or MRI. Why? Because I need the dimensions of that, isn't it? Length, breadth, depth. I need dimensions of that organ to print it out. Agreed? CT scan. CT means computer tomography. Computer tomography. Not city, not village, not state. Okay. Computer tomography. Or magnetic resonance imaging. Is it clear? Okay. Why are we doing that? To get the dimensions and the 3D structure of the organ to be printed. Whatever organ you want to print, that organ's structure, 3D structure you want. Next, step 2. Okay, step 2, what we have to do? Using computer aided design, you know, AutoCAD software. Using such softwares, get a digital model of that, I mean get a virtual kidney. Is it clear? 3D model, like you make using AI, you create even the 3D structures like that. Okay, create a digital model of that. Okay, kidney, whatever organ you want to print out. Are the 2 steps clear? First step, 3D image. Second step, a 3D digital model of the organ using computer aided designing softwares. Step 2. Step 3, okay, take a hydrogel. Hydrogel means what? Water based gel. A gel in which base material is water. Okay. In that you are mixing alginate and fibrin. Is it clear? Water, alginate, fibrin. Is it clear? Together it will form a gel. This is called bio ink. It is not really ink. Okay, but normally in the cartridge we fill ink, so symbolically we are calling it as ink, bio ink. Okay, what is this? This is like base material. Is it clear? Base material. In this you should add, okay, kidney cells. I hope you understood that. This is like matrix. like cementing material. I hope you understood the point. Okay, what is the purpose of this? It will help in cell division, cell differentiation and physical attachment of the cells. Kidney is not just a group of kidney cells, isn't it? The kidney cells should divide, they should get differentiated and they should attach with each other to form the kidney. For that you need some cementing material, that is called this bioink. What is bioink? Hydrogel made up of alginate and fibrin. Why are we taking this alginate and fibrin only? Because they are biocompatible, biodegradable. Is it clear? Biocompatible, biodegradable, because once you transplant the kidney, then this alginate fibrin will get automatically disintegrated in our body. They will not cause any problem in our body. Is it clear? Okay, right. To make kidney, I need only kidney cells. I am repeating, to make kidney, I need only kidney cells. Then why am I taking this hydrogel in which alginate and fibrin are present? That is a base material. Okay, like in the salt water, salt is actually what you want to take, but water is like base you need, like that. Okay, why am I taking alginate fibrin only? Because they are biocompatible, not biohazardous. Not only that, biodegradable also, because it should get disintegrated, degraded once the organ is transplanted into my body. Then why am I using it? Okay, it is needed for the kidney cells to grow, divide, differentiate and attach to each other so that it can form like a kidney. Okay, so in that we add, is it clear? Plus kidney cells, what we do? We incubate it for some time. Okay, take water, mix alginate and fibrin, you will get a hydrogel. Into that mix kidney cells and incubate it for some time. Okay, then you will get, this is what we are putting in the cartridge. Okay, then what happens? Kidney printed out. You can surgically arrange it into your body. I hope it is clear. That is printable organs. Okay. Organovo company manufactured human liver. Already human heart is also manufactured. Okay, but they are not yet, okay, implanted into the human body. They are not yet implanted into the human body. Is it clear? Heart, liver already manufactured, but they are not transplanted into human body yet. Okay, but Chinese have created ears. Okay, and there are some kids with a defect called microtia. What is a microtia? They will not have the external ear, or even if the external ear is present, it is very small. I mean literally a baby born without ears. So a Chinese company manufactured ears using 3D printing and even surgically arranged them to them. Is it clear? So ears, heart, liver, many organs are manufactured, but the only organ which is successfully transplanted is ears. Real ears, not hearing aid, real ears with skin, circulatory system, nervous system. Is it clear? Okay, where the cells can divide, like that. Is it clear? Okay, which company? A Chinese company manufactured the ears and arranged into the patients who are born with microtia. Microtia is a condition where the child is born without the ears. Is it clear? Okay, then. So this is one way. Okay, this is one way to manufacture the organs. There is another way to manufacture the organs. Okay, this 3D printing is, okay, this 3D printing is in vitro organ production. Do you agree? This is in vitro organ production. Okay. But Japan recently approved in vivo organ production. In vivo organ production. Is it clear? Okay. So here what they have done is, look at here. In this, the black colored cells you can see, what is that? Pig embryo. Okay, keep using the words. What is that black color? group of cells. What are those cells? Pig embryo. In that, what am I keeping? Some red color cells you can see here. What are they? Human iPSCs, induced pluripotent precursor stem cells, which we just understood. Human iPSCs. Okay. Then here you may get one doubt that iPSCs can form into different organs. But how do they know? For example, I want liver. But how do the iPSCs know that they should form liver only? By chance, what if they make heart? I have a liver failure, but heart is developed. I don't want it, right? Okay. So I should communicate with the iPSCs that, hey, iPSCs, you should grow into liver only. Okay. Don't grow into heart or kidney. Is it clear? How do I do that? Okay. Treat them with liver specific hormones. Treat them with liver specific hormones. Is it clear? Step 1, take pig embryo. Step 2, okay, add human iPSCs which are treated with liver specific hormones. Means now they know that they should grow only into a liver. Now they know that they have to grow into liver cells only. They should not get converted into heart or brain cells. That is, okay, a kind of a chimera. Chimera word means mixture. Okay. Why it is a mixture? Pig plus human liver, isn't it? Pig embryo, human liver cells or human iPSCs, isn't it? Pig embryo, human iPSC, combination, mixture. Okay, you got that. Then what you have to do? Step 3, okay, implant such embryo into the pig womb. Okay, implant such embryo into the pig womb. Step 1, take pig embryo, make it in the laboratory through IVF. Make the pig embryo in the laboratory. Add human iPSCs to it which are treated with the liver specific hormones. And then you will get a combination embryo. Implant it into the pig womb. Then the piglet, that is the animal young one, is born with human liver. Is it clear? Now the pig is born. But the pig is not born with pig liver. The pig is born with the human liver. And then what you have to do? Okay, the liver the animal slaughtered. It means, will the animal die? Yes. That is the reason why it is not permitted till date, because even animals have animal rights. It was permitted only in the year 2019. That too only in Japan. Why it was not permitted till 2019? Because it will lead to the death of that animal. Because you take that liver out, na? Pig born with human liver. Okay, you kill that pig, take that liver. Now without liver, how can the pig survive? It will die. So that is the reason why it is not permitted in the other countries and in Japan also, not till 2019. Recently it is permitted. Then you may ask me, in that case, why it is permitted? Arey Baba, for consumption of pork or meat also we are killing the animals. Why can't I kill them for manufacturing a heart or a liver? Which is at least reasonable. Tell me, killing an animal for one day meal or killing an animal for an entire lifespan of a human being. When we are not hesitating to like kill the animals for meat, okay, I think it is even okay better than that. Is it clear? So okay, it is permitted and other countries are also moving in that direction. I hope it is clear. Okay. Then okay, you may get one doubt. What about the size? Okay, no problem. See, we are capable of in vitro organ manufacture. So why can't you just grow the size of the organ if you need? That is not a big task. If the liver from this animal young one is little smaller, we can grow it in the size. Next, you may get another doubt. Okay, don't the pig produce antibodies for it? Pig immune system do not destroy these human iPSCs? No, because they are injected at embryo level. The innocent pig immune system will think that it is my cells only. So the pig immune system will not reject it. Okay, if I transplant such a liver into my body, will my immune system reject it? No, Because my immune system innocently think that it is human liver only because it is made from human iPSCs. So human immune system also will not reject it. So this process is possible, okay, but organs are not yet manufactured using this procedure, only theoretically it is possible. Approved in Japan, other countries are moving in that direction. I hope it is clear. Okay, next topic. A man gets pig's heart. Don't get confused, both are not the same. Earlier, man heart made in the pig. This is not man's heart made in the pig. Pig heart only kept in the man. Is it clear? Is it possible? Yes. Okay. But unfortunately, okay, an American company did this called Reviviker. Is it clear? Okay. But, okay, not directly. Okay, not directly. They made 10 modifications. Simple. Take the pig heart. Make some 10 modifications. Why 10 modifications? So that it becomes similar to human heart. You are cheating the immune system. Okay. So you are trying to cheat the immune system so that the immune system should think that it is human heart only. I hope it is clear. Okay. Take the pig heart. Make some 10 modifications so that it will become similar to the human heart and then transplant it. That is another possibility. Is it clear? Okay. A doctor in USA did this experiment, but unfortunately that man died. Okay. But it was not a careless experiment. Before that, he made many experiments on monkeys. It was successful. Moreover, that patient had no other option to survive. Is it clear? So, anyway about to die, so at least, okay, do this experiment. By chance, if you are lucky enough, you can survive with a pig's heart. Otherwise also no problem, because anyhow he was about to die. So in that situations he did this. It was not a careless experiment but unsuccessful experiment. But in future it can be successful. That is another topic. Is it clear? Okay. Next topic, organoids. Organoids means what? Okay. Small organs. Small organs or miniature organs. Miniature organs. Is it clear? Okay. Like brain, kidney, lung. This much brain, this much lung, this much kidney, this much liver. Okay. Why? Do we use them for transplantation? Of course not. Okay. We use them for models. For, okay, like, see, what are the uses? Okay. For example, medicos can learn how the organs will develop. Live they can see the kidney growing. Live they can see the pancreas, not dissections. Is it clear? Okay. Next, they provide new insights on the diseases. I mean, for example, if you get COVID-19, how the virus is affecting the lung. You can in the laboratory you can see this on this miniature lungs. Take that miniature lung, add coronavirus, see what it will do. I hope it is clear like that. Okay. Then study how the drugs interact. Simple. Take that miniature organ, add that drug, see how the drug works. Okay. Like that. Drug discovery. Scientists have gained insights. One example, scientists understood the mechanism of COVID-19 using lung organoids. That is the story of organoids. Is it clear? Next. Next topic is Human Genome Project. Okay. To understand Human Genome Project, first of all we have to understand what is genome. Okay. Genome means the haploid genetic content of a cell, or simply put, one copy of the total genetic content of a cell. Earlier one student was asking me when I was telling you about the ploid. Okay, she asked whether ploid and genome are the same. I said yes, mostly they are the same. Yes. Okay. This is also one copy of the total genetic content of a cell. It means one genome is equal to 23 chromosomes in human beings. Is it clear? In the human beings it is 23. If it is Drosophila 4, if it is pea plant 7, if it is maize plant 10. I mean whatever chromosomes you have, we have one copy of it is genome. I hope it is clear. Next, okay, complete focus please, okay, and actually you are already focusing, okay, and I appreciate your concentration levels. Listening to a class is as difficult or even sometimes more difficult than actually teaching the class. Like for example, as a student, I could never focus on the class. So you are definitely, okay, your effort is quite appreciable. Just continue the same, right? Okay. Then, okay, one genome is equal to 23 chromosomes, right? Next, what is Human Genome Project? Okay, Human Genome Project. And that is where, Abhishek, I will have to remind you that your target is great. Your target is not small target that I have to clear LKG exam, I have to clear 10th class exam. When your target is huge, huge efforts are also needed. Okay, so complete focus please, right? Next, so if you have a target of clearing LKG exam, then enjoy outside, right? So one genome is equal to 23 chromosomes. Then what is Human Genome Project? Simple. It is to read, I mean know, okay, the sequence, I mean order of nucleotides, okay, in the genome. Is that clear? Okay, so what is a Human Genome Project? The task of this project is to know the order of nucleotides in the genome. I mean, is it ATG, CCC, TAT, or is it TAC, GGG, ATG, or is it CTC, TAGC? Which order? Okay, you have nucleotides in which order, which sequence in the genome. Knowing that is called as the Human Genome Project. Is it clear? Right. What is the instrument used? DNA sequencer. This is like a scanner, okay, that will identify the nucleotides. It's like a scanner that will identify the nucleotides. I mean, like look at here, I'll show you using my laser pen. Okay, look at this. First scanned and identified as C. Next letter scanned, identified as T. Next letter scanned, identified as C. Next letter scanned, identified as C. Next letter scanned, identified as T. This is how the DNA sequencer works. I hope it is clear. It is a scanner. It will identify the nucleotides. If it identifies each nucleotide, we will understand the order of nucleotides. Yes. Okay. Fine. Done. We understood the order of nucleotides in our complete genome. It means in all our 23 chromosomes, what is the order of nucleotides? We know that. Okay. After knowing the order of nucleotides, what are our observations? More than 95% of the genome is junk DNA, means not producing proteins. It means not genes. Not genes. Remember how did we define gene? Functional sequence of nucleotides on the DNA, but they are non-functional, junk DNA, not genes. Also called as selfish DNA because they are not producing proteins for you but still existing in your cells, means making selfish existence in your cells. Therefore, that is also called selfish DNA. Also called non-coding DNA. I mean, it is not coding for proteins. Is it clear? 99% of our DNA is like that. It means all our genes will put together will form only 0.1%. Okay, not 0.1%, around 1% of the total genome. Is it clear? Right. In that 1%, we have 30,000 genes. We have 30,000 genes in complete genome. It means we have, okay, 30,000 genes on 23 chromosomes. It means we have around 1300 genes on one chromosome, roughly on an average. Okay, 1300 genes will be there on one chromosome, but that is not fixed number. First chromosome may have 2000 genes, second chromosome may have 1800 genes, third chromosome may have some 1700 genes, like that. On an average, you have 1300 genes on each chromosome. So 23 chromosomes put together, you have 30,000 genes. Clear till this point? Next, those are the observations. Okay, now let us see the human genome projects in news. Very important, human genome projects in news. Okay, hope you are retaining the complete focus at any cost. Don't get distracted. Okay, now first one is human genome project. This is the very first such project in the world, right? For this, they have taken the samples from all over the world. They have taken the samples from all over the world. It means you may get one doubt. Okay, do we all have the same genome sequence or different genome sequence? You and me have the same DNA sequence or different genome sequence? Different, right? Then whose genome did they sequence in this project work? Whose genome did they sequence in that project work? No, not the sequence of one genome. They have taken, okay, lakhs of people from all over the world. Is it clear? Okay, lakhs of people from all over the world, all their genomes sequenced. All their genomes sequenced. With this you can understand the diversity, isn't it? How Africans are different from Americans, how Americans are different from Asians, like that. I hope it is clear. Fine. Next one. Genome Asia 100K. Simple. Okay. 1 lakh individual from only Asia. First one is lakhs of people from all over the world. Second one is 1 lakh people's genomes sequenced from Asia. 50,000 Indians only, other 50,000 from the other Asian countries. I hope it is clear. Okay. Next one, Indigen project. Okay, Indigen project. It is done by IGBI Delhi, CCMB Hyderabad. As the name itself is suggesting, okay, this is only Indians, not Asians also, not world also, only Indians. Next one, Genome India project, launched in 2020. Okay, recently 10,000 individuals' genomes are sequenced. I mean, they understood the order of nucleotides in 10,000 Indians. Is it clear? Okay, so with this we can understand more about the unique mutations in Indian populations. Is it clear? Okay, only 10,000, that is called sample pragna. Okay, these 10,000 people we take randomly. Is it clear? Randomly from Northeast India, North, South. Okay, random sample we take. Okay, you cannot sequence the genome of every Indian, right? So we take sample. Okay, next one, telomere to telomere consortium. What is telomere? Look at the chromosome. In the chromosome, this part is called centromere. The tips of the chromosomes are called as telomeres. The tips of the chromosomes are called as telomeres. Okay, then this is another telomere. Telomere to telomere means, sometimes they say you have to read this book from cover to cover, they say. It is like that. You have to completely read the nucleotide sequence from one end of the chromosome to the other end of the chromosome. Okay, So like that we have to sequence all the chromosomes. That project work is called as T to T consortium. Is it clear? Telomere to telomere consortium. Is it clear? Next one, human genome project, right? Is it clear? It means what? In all these cases, you are only reading. You are reading the sequence of an existing genome. Understood? You are reading the sequence of an existing genome. Okay, but here you are creating a new genome. You are creating a genome artificially. I hope it is clear. You are creating a genome artificially. Clear till now? Okay. All these human genome project, Genome Asia 100K, Indigen project, T to T consortium, you are not creating a genome. Genome is there. You take the genome from a person and you read it. But here you are creating a genome. So therefore it is called HGP, right? This is called as synthetic biology. This topic is called synthetic biology. Okay. Then what are the applications of human genome project? One is pharmacogenomics. Use the word once. Pharmacogenomics. Did you notice sometimes when two people take the same medicine, the medicine works better on one person but does not work better on the other person. Okay. We take the same medicines but the impact is not the same. Why? Where is the difference? The difference is in the genome sequence. So what best you can do? Okay. Read, for example, I am a doctor. A patient comes to me. I will read his genome sequence and then give suitable medicines. Is it clear? Okay. When I go to a doctor, the doctor will read my genome and then prescribe me suitable medicines. I hope it is clear for all of you. Of course, I am not a doctor. Okay. Next, coming back to this pharmacogenomics. Done? Okay. Next one. Earth Biogenome project, quite interesting. Not human genome project. Sequence the genomes of every organism that is living on the earth. Bacteria, ant, cockroach, okay, mango tree, human being, dog, cat, wolf, okay. Every organism which is living on the earth, we have to do the genome sequencing. Of course, it is a never-ending project and we completed only 0.2% of this because how many such organisms are there on the earth? Okay, only 0.2% of the project is completed. Is it clear? Okay, it will never complete, but it's okay. But with every day, our knowledge about the living organisms will go on increasing. That is what is the greatness of science. It will not expect to know everything in a single day. Patiently we work, we keep on working, our understanding about the life increases. But it will never complete. Even if it is complete, a new species will be formed. Okay, like that. Okay. Next application, we can also use genome sequencing in the diagnostics, disease diagnostics. Okay, a simple logic. Okay, all of you answer this. For example, okay, we are human beings. You have taken the throat swab like COVID-19 test. Okay, diagnostic test. You have taken the throat swab, nose swab. Okay, in that if you find coronavirus DNA, what does it mean? Human sample, but in that coronavirus DNA, what is the meaning? It means I am infected by coronavirus. Isn't it? Otherwise, why do I have coronaviral DNA? I will get it only if I am infected by the virus. Or take a human sample, if you find tuberculosis bacterium DNA in that, what do you mean? Okay, it means you are infected by TB. Is it clear? So like that, by finding the genome sequence of a particular pathogen in the sample, you can use genome sequencing for, okay, disease diagnosis. Is it clear? Okay, then next one. Okay, Global Initiative on Sharing Influenza Data, GSAT, okay, GISAT, okay, what is this? You know, viruses mutate very quickly. We get different variants of the viruses. every variant will have a new genome sequence. Is it clear? So this GSED, what it will do? It will sequence the genome of every variant of the influenza virus. It will sequence the genome of every variant of the influenza virus, maintain the database, and in the world they keep sharing this database. For example, a new variant is detected in Africa. They will share this information with the other countries. Another new variant is detected in America. They will share the information with other countries. So such a platform is called as GSED. Same way, we have SARS-CoV-2 genomic consortium called INSACOG. Okay, it is a consortium of several laboratories. Okay, what is INSACOG? It is a group of laboratories. It is a group of several laboratories. What they will do? They will sequence the genomes of, okay, SARS-CoV-2. They will sequence the genomes of SARS-CoV-2. Is it clear? And then maintain the database. Maintain the database. Okay, so that it will have a record of all the new variants. Maintain a record of all variants, okay, of SARS-CoV-2. All variants of SARS-CoV-2. Is it clear? Okay, this will help in the discovery of new vaccines, discovery of new medicines, like that. That is INSACOG. Okay, next, biological dark matter is not the same as junk DNA. Junk DNA means used like non-functional DNA, whereas biological dark matter can be functional or non-functional, but we don't know about that. Like dark matter, is it clear? The part of the genome whose function is not clear is called as biological dark matter. Is it clear? That part of the genome whose function is not clear. It does not mean that there is no function. There can be function, but we don't know. Okay, it can be genes or junk DNA, we don't know. Such part of the genome is called as biological dark matter. Okay, so these are all the points related to the genome sequencing. Is it clear? Then the next topic we are discussing is genetic engineering or recombinant DNA technology or gene cloning. Is it clear? Gene cloning. Can we continue? Okay. Look at this diagram. I'll explain with this diagram. Complete focus, please. Okay. So, what is this one? What is this? It is a human cell, right? It's a human cell. In that, imagine this is the insulin gene. Insulin gene. Okay? Done. Then, next one. What is this? This is a bacterial cell. Complete focus, please. It is a bacterial cell. Okay? What is this one? This thread-like structure? Bacterial DNA. Okay? Bacterial DNA. Right? Then, what is this? This is a plasmid. Okay, what is a plasmid? That is also, okay, DNA only. Plasmid is also DNA. Okay, like a small DNA. Okay, smaller than the bacterial chromosome, but that is also a DNA. Clear till now? Okay. Now what you do? Take out this plasmid. Okay, take out the plasmid from the bacteria. Add the human insulin gene to it. The resulting one is called as recombinant DNA. Why that is called recombinant DNA? That is a new combination of DNA, isn't it? Bacterial DNA in that human gene is a new combination of DNA we created. So that is called recombinant DNA. And then next step, place that recombinant DNA into the bacterial cell. Place that recombinant DNA into the bacterial cell. Okay. And then you got recombinant bacteria with human insulin gene. The bacteria is called recombinant bacteria. Why is that called recombinant bacteria? Because now it is having recombinant DNA in it. Therefore, it is called recombinant bacteria. and then let the bacteria perform cell division. Let the bacteria perform cell division. Then you got a group of bacterial cells. If you observe, it just looks like a miniature version of this. Is it clear? Bacterial cell, bacterial DNA, plasmid, insulin gene. Is it clear? Okay. You got a clone of bacteria with human insulin gene. Is it clear? Okay. Clone means you have a group of bacteria with human insulin gene. Is it clear? Okay. Now you will get few questions. Okay. Why did we keep the human insulin gene in the bacterial cell? Why did we keep the human insulin gene in the bacterial cell? Because you are using bacterial cell as a Xerox machine to copy our gene. The innocent bacteria will think that it is a part of its own DNA and will replicate our gene also. Is it clear? So that is the reason why you are, okay, therefore this is called gene cloning, because I hope you remember clone means copying. Okay, you have one copy of the human insulin gene here, but here you have millions of copies. Okay, then comes the next question. But why do you want millions of copies of insulin gene? Because, okay, the bacteria not only copy the human insulin gene. Look at here, okay, the bacteria will do two things. Bacteria will copy the human insulin gene, plus it will also produce insulin hormone. Then you may get one doubt. Can the bacteria produce human insulin? Yes. Naturally, no. But remember, we kept insulin gene into that now. We kept human insulin gene in that. Now this recombinant bacteria can produce human insulin hormone. Okay. Then what we can do? Okay. What we can do is, okay, extract, purify and use as, okay, drug for diabetes. In the medical shops, people purchase, diabetic patients will take insulin injections. They purchase insulin from the medical shops. How do they get that? By this procedure only. Okay. I'll just repeat the process once. Okay. Human cell, human DNA, insulin gene on that. Bacterial cell, bacterial DNA, plasmid. Plasmid is also DNA. It is a small piece of DNA. Take this plasmid out, add the human insulin gene. We call this as recombinant DNA. Keep it in the bacteria. We call this bacteria as a recombinant bacteria. Then let the bacteria perform cell division. You get a group or a clone of bacterial cells. All of them are containing the human insulin gene. Okay. The bacteria will do two things for us. One, it will copy the human insulin gene. Two, it will also produce human insulin hormone. And you extract the human insulin hormone, purify it and use it as a drug for the diabetic patients. I hope it is clear. Okay. Next. Okay. Can we produce only insulin like this? Can we produce only insulin like this? No, we can also produce blood clotting factors. Did you hear about a disease called hemophilia? Okay. In hemophilia, what happens is the blood cannot clot. In hemophilia, the blood cannot clot. Is it clear? Why? Because clotting factors are missing. The blood clotting factors are missing. Is it clear? Then what is the treatment? Okay. What is the treatment here? Clotting factor injection. For example, diabetes. What I don't have? Insulin I don't have. So give insulin injection. Hemophilia. What I am missing? Clotting factor. So give clotting factor injection. Is it clear? Okay. How to produce clotting factor? Same procedure. Look at here. Instead of insulin, clotting factor gene. Okay. And it is a recombinant bacteria with clotting factor gene inside. then the bacteria will copy the clotting factor gene and also it will produce the clotting factor, okay, and use it for hemophilia. I hope it is clear. Same procedure, okay, whatever protein you want, add that gene to the bacterial cell. Got it? Okay? Yes, yes, perfect. Okay. Same way, growth hormone also, you know, dwarfism, some people will be very short, just 1 feet, 2 feet tall, like that. Okay. So growth hormone, same thing, growth hormone injection. Is it clear? Okay. Instead of insulin, growth hormone gene in that. Then you will get a recombinant bacteria with growth hormone gene in it. A clone of bacteria with human growth hormone gene, okay, growth factor or growth hormone, and okay, it will copy the growth hormone gene and it will produce the growth hormone. It can be used to cure, okay, dwarfism. It can be used to cure the dwarfism. I hope it is clear. Okay. Now, same way, recombinant vaccines also, recombinant vaccines as well. Okay. But before we understand recombinant vaccines, we are also going to discuss different types of vaccines. Is it clear? Different types of vaccines. Done? Okay. First, okay, what is like the basic principle of vaccine? Quickly answer this. For example, if I take polio vaccine, will it contain a medicine for polio or the polio virus itself? Or if I take coronavirus vaccine, will it contain the coronavirus itself or a medicine for the coronavirus or the medicine for coronavirus? Is it clear? Vaccine contain, okay, the pathogen itself. I mean, it will contain the, we call this as antigen, okay, and when we inject this, okay, our immune system produce antibody. Is it clear? And this is our body's defense mechanism. Is it clear? And the basic principle of vaccines is, okay, digging the well before you feel thirsty, making the weapons, like for example, we are ready with S-400s. So when the attack happened, we could quickly retaliate, like that. They are like S-400s in our body. Antibodies are like, we first make the antibodies ready. Is it clear? So that what happens, okay, if really the virus infects, the antibodies will destroy. Is it clear? If you don't take a vaccine, if directly the coronavirus enters into my body, it will take 72 hours for me to produce antibodies. Meanwhile, the virus will destroy my organs. So therefore, if the antibodies are ready, then if the virus comes readily, these antibodies can attack on that. That is the basic principle of vaccine. Clear for all of you? Yes. Now let us see. Obviously, when the vaccine is the pathogen itself, you may get one doubt. Because basically, when I inject that virus, what if the vaccine itself caused the disease? What if the vaccine itself caused the disease? Because you are injecting the virus only, right? Okay, I hope the question is clear. So, okay, we have different procedures. First procedure is take the whole virus or the bacterium. Either kill it or inactivated or attenuated. Is it clear? Take the complete virus, kill it or inactivate it or attenuate it. Okay, number 1, number 2. Are the two mechanisms clear? I am going to give you some 4 or 5 mechanisms of making the vaccines, right? Around 4 to 5 mechanisms of making the vaccines I am going to give you. First mechanism, killed vaccine. Second mechanism, attenuated vaccine. But both are whole virus or whole bacterium vaccines. Done? Next. Okay. Next type is called as subunit vaccines. Is it clear? Subunit vaccines are also called as recombinant vaccines. Is it clear? Use the word subunit vaccine or recombinant vaccine. I'll tell you how to make it. Okay. Look at this. This is corona virus and this is spike protein. Is it clear? Spike protein. You know that? Coronavirus and spike protein. Now look at here. What we are doing. Okay. Now imagine this is, okay, SARS-CoV-2. Not human cell. Imagine that is SARS-CoV-2. From that which gene you have taken? Not insulin gene. Spike gene. I hope it is clear. Spike protein gene. Clear till now? Then, okay, take the plasmid out as it is. Okay. What? Spike protein gene. Okay. And then the next story is the same. Take the plasmid out. Add the spike protein gene into this. Okay. And then you will get a recombinant bacteria. Recombinant bacteria with spike protein gene. Recombinant bacteria with spike protein gene. Is it clear? Next, you will get a clone of bacteria with spike protein gene. I hope you are getting this clearly. A clone of bacteria with spike protein gene. Right. Then the bacteria will do two things. Okay. It will copy the spike protein gene. It will copy the spike protein gene and also produce, okay, the SARS-CoV-2 spike protein. This is, okay, this is injected as vaccine. Injected as vaccine. I hope you understood the process. Quick response please. Spike protein gene in the bacteria. The bacteria will copy the gene, also will produce a spike protein. Use that spike protein as a vaccination. Okay. Then this is called as, this is called subunit vaccine. Okay. Because you are using only one part, that is spike protein in the vaccine. Did you understand? Okay. You are not using, like for example, I am not taking, if this is a virus, I am not taking the complete virus, I am taking only one part of it. So subunit vaccine. Okay. This is also called as recombinant vaccine. This can also be called recombinant vaccine. Okay. Why? Because it is made using, okay, recombinant DNA technology, isn't it? What is this process called as, this entire process? What is this called as recombinant DNA technology? This vaccine is made using recombinant DNA technology. Okay. Therefore, it is called as recombinant vaccine. I hope it is clear. Okay. Two procedures. First one, okay, subunit vaccine. Okay. See, first one, weakened or inactivated vaccines, first category. Example, Covaxin for coronavirus. I hope this box is clear for everybody. This is the first box. What is this called as? Okay, weakened vaccine, okay, or inactivated vaccine. Okay, like I explained here. Take the complete virus, not subunit, complete virus. Inactivate it and use it. Okay. Example, Covaxin. Same way, Sinopharm, Sinovac. All these are coronavirus vaccines. I hope it is clear. Coronavirus vaccines. Right? Okay. Second one. Second box. I hope the first box is clear for everybody. Second box is also clear. Subunit vaccine. Okay. The same procedure which I explained here. Take the spike protein gene, keep it in the bacteria, let the bacteria multiply, spike protein gene copied, the bacteria will produce spike protein, use that protein as vaccination. It is called subunit vaccine or recombinant vaccine. Box number 2. Example, Corbevax, Covovax, okay, and Novavax for SARS-CoV-2. And cervical cancer vaccine, HPV, human papilloma virus. Is it clear? Right? And in this box, if the disease name is not mentioned, they are coronavirus vaccines. COVID, these are, most of these vaccines are made during the COVID-19 pandemic. Okay. Here I mentioned it is not coronavirus vaccine. So I mentioned the name, cervical cancer, HPV. This is Ebola. Like that, if it is not mentioned, remaining all are SARS-CoV-2 vaccines only. Don't get confused. Clear? Okay? Right. HPV is human papilloma virus. It will cause cervical cancer. I hope the two boxes are clear. Third box, what we do is we directly take the viral RNA or viral DNA and use it as vaccine, not protein. Here we use protein, isn't it? Here we use protein. Here we use, okay, genetic code. Is it clear? I mean RNA or DNA. RNA or DNA as vaccine. I hope it is clear. Examples are, okay, Zycov-D, that is a DNA vaccine. Pfizer, BioNTech, Moderna, these are RNA vaccines. For which disease? SARS-CoV-2. As I already told you, if nothing is mentioned, it is about SARS-CoV-2. Clear till this point? Okay. What is used here? Protein. What is used here? DNA or RNA. What is used here? Complete virus. Okay, then this one, okay, here we used bacterial plasmid as vector, isn't it? Here we are using bacterial plasmid as vector. Vector means what? Carrier. Vector means carrier. Like see, this is vector. It is carrying the gene, isn't it? Look at here, this is plasmid is vector and it is carrying the gene. Is it clear? But fourth mechanism is we use, okay, viral vector. Simple viral vector. Yeah. So we use viral vector instead of bacterial plasmid as vector. I hope it is clear. We use viral vector instead of bacterial plasmid as vector. That's all. Okay. Examples, Covishield, Sputnik 5, Incovac, that is nasal vaccine for coronavirus, plus also vaccine for Ebola. Is it clear? Okay. Which vector we use here? The vector we use here is, okay, ChAdOx, CH for chimpanzee, AD for adenovirus, and that is some series number. Is it clear? That is some series number. So chimpanzee adenovirus vector means what? It is an adenovirus that will infect chimpanzee. It will not infect humans. It will infect chimpanzee. That viral DNA you are taking and using it as a vector. So that is called viral vector DNA. Is it clear? Okay. So here we use one protein. Here we use, okay, I just repeat. Here we use the complete virus inactivated, example Covaxin, Sinopharm, Sinovac. Here we use only one protein. That protein is made using recombinant DNA technology, therefore it is called recombinant vaccine or subunit vaccine. Okay, example Novavax, Corbevax, Covovax plus cervical cancer or human papilloma virus vaccine. Next one is not protein, DNA or RNA is directly given as vaccine. Is it clear? Okay. So examples are Pfizer or BioNTech, Moderna, these are the examples. Also Zycov-D, it is a DNA vaccine. Okay. Lastly, viral vector vaccine is very similar to subunit vaccine, but in the subunit vaccine we are using bacterial plasmid as a vector, but here we are using chimpanzee adenovirus DNA as vector. Instead of bacterial plasmid, we are using chimpanzee adenovirus DNA as vector. So that is viral vector vaccines. These are different vaccines, and knowing the names is very important because you can get questions like this. Read this question out, everybody. Okay. In the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. Serum Institute of India produced COVID-19 vaccines Covishield using mRNA platform. Is Covishield an mRNA vaccine? No, it is a viral vector vaccine. So this is wrong. Okay, Sputnik 5 is vector based. Okay, Sputnik 5 viral vector. Yes, this is correct. Covaxin is inactivated. Yes, Covaxin is inactivated. That is correct. So remembering the names is important here. Okay, that is the story of vaccines. Okay, and then moving on to the next topic, that is gene therapy. Gene therapy means, okay, treatment using genes. Is it clear? By editing the mutations. By editing the mutations. Is it clear? Devyani, first break will be at 4:10 p.m. Okay, 4:10 to 4:30 you will get the first break. Okay, right. It is difficult for me also. Okay, but great targets need great efforts. That is all I can say. Okay, and also the only way I can motivate you is by saying that I am also with you in your effort. Okay. So I can also work with you. That is the only way, okay, I can motivate you in this crucial moment just before the examination. Is it clear? We are also working with you. Let us work together for your success, right? Okay. Now, so now let us try to understand. Yes, hum saath saath hai. Okay. So now coming back to this treatment using the genes by editing the mutations. If you remember, we corrected the mutations, we understood that the mutations are like spelling mistakes, okay, and then gene therapy is like editing, okay, you have to correct the mistake, okay, that's all. That is gene therapy. How does the gene therapy work? Okay, by CRISPR-Cas9 mechanism. Is it clear? Okay, CRISPR-Cas9 mechanism. Let us try to understand the CRISPR-Cas9 mechanism. Complete focus. You have to understand it in the first go. Okay, I can't repeat it. Okay, right. Right. Now, what is CRISPR-Cas9? In the CRISPR-Cas9, Cas9 is a nuclease. Means it is like scissors. It can cut the DNA. Okay, ase means cut. Nuclease means cuts the nucleic acid. Is it clear? Okay, nuclease means cuts the nucleic acid. It can cut the DNA. What is CRISPR? CRISPR is a guider RNA. CRISPR is a guider RNA. Right? Okay, means this is basically an RNA that guides the Cas9 where to cut. Okay, Cas9 can cut the DNA, but it does not know where to cut. So CRISPR will tell the Cas9 where to cut. I hope it is clear. Okay, right. Then CRISPR and Cas9 are linked to each other. Cas9 goes wherever the CRISPR goes. I hope it is clear. Cas9 goes wherever the CRISPR goes because CRISPR and Cas9 are linked to each other. Okay. And where is CRISPR? Look at here. This is CRISPR. This is CRISPR. Okay. And what is this one? This is the mutated gene, okay, where you have to do the editing. Where is the mutation? Look at here. T with A, T with A with T, G with C. But G with T, okay, this is a mistake. I hope you understood that. That is a mistake. So, what is this one? This one is a mutated gene. I hope it is clear. What is this? It is a mutated gene. Done? Mutated gene. Now what I want to do? Okay, delete mutated gene. Add correct gene. Okay. Delete mutated gene and add the correct gene. That is what we have to do. Okay. Now, all of you look at the CRISPR sequence. CRISPR sequence is complementary to the mutated gene. Did you notice? CRISPR A for U, A, because I told you that, okay, A will always pair with T, G will always pair with C. If it is RNA, then T will not be there. So, A will pair with U. If you remember, RNA do not contain thymine. So, in the RNA, adenine will pair with uracil. Is it clear? Okay. Right. ATGC, same way AUGC also. Okay. Now, look at this. A, this is correct, this is correct, this is correct, this is correct. Okay. What is the sequence of CRISPR? CRISPR is complementary to the mutated gene which is to be deleted. Clear? CRISPR is complementary to the mutated gene which is to be deleted. Done. Next. Okay. This is one gene, gene number 1. Okay. Now, answer this question. Gene number 1, gene number 2, gene number 3. Can the CRISPR bind to the second gene? Can this CRISPR, can UUAGA, can this CRISPR, same thing, can it bind to the second gene? All of you? No. Okay. Very good. Okay. Then tell me, can this CRISPR, okay, bind to this third gene? No. Is it clear? Means it is complementary. Is it clear? Okay. 2 points you have to remember about CRISPR. First thing, CRISPR is artificial. I mean, we made this. Second one, CRISPR is gene specific. CRISPR is gene specific. I mean, the CRISPR made for one gene cannot work for the other gene. The CRISPR which I made for the first gene do not work for the second or the third gene. Remember these 2 points about the CRISPR. Is it clear? Okay. Right. So, now the CRISPR bound to which gene? Everybody please. The CRISPR bound to which gene? 1 or 2 or 3? All of you. The CRISPR bound to which gene? 1 or 2 or 3? Fast everybody, 1. Okay, and the Cas9 will go wherever the CRISPR go, right? So now the Cas9 will cut the first gene. Means CRISPR is like marker. For example, a tailor, okay, a tailor is stitching a shirt for you. What he will do? With a chalk piece, first he will mark where to cut. Then he will cut. That marker is, okay, CRISPR, and that scissor is Cas9. Is it clear? CRISPR bind to the first gene. Cas9 will cut wherever the CRISPR bound. So the first gene is deleted. And in that place we have to add the correct copy of that gene. I mean, okay, here, okay, what is deleted? Okay, look at here. AATCT cut, AATCC add. Remove AATCT, add AATCC. That's all. That is CRISPR-Cas9 mechanism. I hope it is clear. I repeat, Cas9 is a nuclease enzyme. It is like a scissor. It can cut the DNA, but it do not know where to cut. CRISPR is basically an RNA which will guide the Cas9 where to cut. How does it guide the Cas9? Because basically it is an artificial one and gene specific one. It is complementary to the mutated gene which you want to delete. So the CRISPR will bind to the mutated gene and the Cas9 will cut the mutated gene. Once the mutated gene is cut, in that place we add the correct copy of the gene. The mutation is corrected, gene editing, gene therapy. When the mutation is corrected, you got a permanent treatment for that disease. You don't have to give insulin injection every day. You directly corrected the insulin mutation. You don't have to take the clotting factor injections regularly. You directly corrected the mutation in the clotting factor gene. That is called gene therapy. And if you do the gene therapy in, okay, somatic cells, it is called somatic gene therapy. And if you do it in embryonic cells, we call it as embryonic gene therapy. Okay. In the somatic gene therapy, the advantage is it is legally permitted. But drawback is, how many somatic cells we have in our body? Infinite. Is it clear? So can you do this correction millions of, billions of, trillions of times? No, we cannot. So that is the reason why it is difficult. A large number of cells to be edited. Is it clear? Embryonic gene therapy, okay, the problem is, okay, in the embryonic gene therapy, the problem is it is legally not permitted. But the advantage is it is easy because embryo will have a few cells. So you will have to do this procedure only few times. You don't have to repeat this procedure unlimited number of times. That is about the embryonic gene therapy and somatic gene therapy. Can we continue? Okay. Right. Next one is designer baby. What is designer baby? Same thing, same procedure. Okay. But here, imagine that, okay, if you cut, see, if you delete mutated gene and treat a disease, that is called gene therapy. Okay, but if you delete dark skin gene, if you delete dark skin gene and add fair skin gene, we call it as designer baby. I hope it is clear. Okay. So that is the only difference. For example, imagine this is the dark skin, this is dark skin gene. Dark skin gene deleted, fair skin gene added. Okay, means you are designing the baby with fair skin, sharp nose, like that. Okay, so that is about the designer baby. Okay, for CRISPR-Cas9 mechanism also there was Nobel Prize. Okay, CRISPR-Cas9 mechanism also received Nobel Prize. Okay, next. What is Casgevy? Casgevy is a CRISPR-Cas9 based treatment for, okay, thalassemia and sickle cell anemia. Is it clear? Okay, right. So what is Casgevy? There are some diseases, Okay, like sickle cell anemia, thalassemia. What are they? Here, okay, the adult hemoglobin is defective. Adult hemoglobin is defective. Okay. Due to defective hemoglobin, we get diseases called sickle cell anemia, thalassemia. Okay, first point to be remembered. Casgevy is a CRISPR based treatment for sickle cell anemia, thalassemia. That's all. For prelims, these are the only 2 points you have to remember, because you don't have to write the mechanism, you don't have to explain what it is. Okay, the only 2 things that you have to remember here is sickle cell anemia, thalassemia. Okay, what are they? They are the diseases. Why do we get those diseases? Because of the mutation in hemoglobin gene. My hemoglobin gene is mutated. I got a defective hemoglobin. So I got sickle cell anemia, thalassemia. What is the gene therapy for it? CRISPR-Cas9 based. What is the name of that gene therapy for these particular diseases? Casgevy. But remember, okay, Casgevy is only for these 2 diseases, okay, only hemoglobin related. Diabetes, if I cure, that cannot be called Casgevy. Casgevy is the name of the CRISPR based treatment particularly for defective hemoglobin diseases. Is it clear? Casgevy is CRISPR-Cas9 based gene therapy for hemoglobin abnormalities. CRISPR-Cas9 based gene therapy for hemoglobin related abnormalities like sickle cell anemia, thalassemia. For prelims, this is enough. Okay. Then look at this question. Consider the following statements. Genetic changes can be introduced into the cells that produce eggs or sperms. Okay. Actually quite simple. You don't even have to check this. Actually genetic changes can be made. Genetic changes can be introduced into any cell. Is it any cell will have DNA? The DNA you can edit. So yes, into the sperms and prospective parent also. So that is correct. Next, a person's genome can be edited before birth at early embryonic stage. Yes, embryonic gene therapy we call it as. Also we call it as designer embryo. By the way, I already told you it is illegal. It is not permitted to edit the human embryos. Is it clear? Okay. Next, human-induced pluripotent cells can be injected into the embryo of a pig. Yes, here it is. That pig one. Long ago. This one. Human iPSCs can be injected into the pig. Yes, we can do that. Okay. So, this is also correct. Which of the statements given above are correct? Okay, 1, 2 and 3, all the 3 are correct, right? Next one. Okay, what is Cas9 protein that is often mentioned in the news? Molecular scissor. Done? Clear till now? Can we move on? Next one. GM crops, genetically modified crops. Genetically modified crops. First one is BT cotton. In BT cotton, B stands for Bacillus. Okay. T stands for thuringiensis. Bacillus thuringiensis. Use the word once. What is Bacillus thuringiensis? It is a soil bacterium. Look at here. Bacillus thuringiensis. Okay. So, what is this? This is a soil bacterium. Okay, and imagine this is its DNA. Okay, its DNA. On that there is one gene called as Cry1AC. Cry1AC will produce BT toxin. The word toxin means what? The word toxin means poison. It is a poison producing gene. It's a poison producing gene. Okay. This gene is transferred to cotton seed cells. Cotton seed cells. You may ask me, why seed cell? Why not plant cell? Same reason as why embryonic gene therapy is easy, not somatics gene therapy is easy. Is it clear? Because seed will have few cells, plant will have more cells. Adding a gene to those many cells is difficult. So adding a gene to these few cells is easier comparatively. I am not saying that it is a very easy thing that anybody can do like this, not like that. But comparatively adding the gene to the seed cells is easy. So transfer that CRIBN gene to the seed cells. Then grow the crop. Okay, you will get BT cotton. Why this is called BT cotton? You got a cotton plant with Bacillus thuringiensis gene in that. Cotton plant with BT gene in that. So it is called BT cotton. Is it clear? And you remember CRIBN, this gene is a poisonous gene. It produces a toxin. So if the bollworms try to eat that cotton plant, then what happens? The bollworms will die. So you don't have to use any additional pesticides. The plant itself is pest resistant. What is the advantage here of making this? Is it clear? The advantage is high yield pest resistant. Is it clear? High yield pest resistant. Is it clear? Why the yield increases? Because if it is pest resistant, look at here, high yield pest resistant. Okay. Why it is high yield? Because if the pest cannot damage the crop, obviously the crop will be healthy and the yield increases. Okay. No damage by the pest, like that. Next. Researchers at Monsanto USA developed this BT cotton. It was introduced in India in 2003, okay, by Monsanto and Mahiko Seed Company. They both together are making this BT cotton. Okay. Advantages: resistance to bollworm, reduce the use of pesticides, okay, improved yield, yield increases, okay, and most importantly, you may get this doubt, okay, what if these cotton seeds are consumed by some animals, okay, or some worms or insects that we need for agriculture, isn't it? Okay, we need some insects for agriculture, for pollination. What if those insects also die? Okay, so it is a very narrow spectrum toxin. It's not a wide spectrum toxin. It will not kill any organism that comes on the way. Especially it cannot kill the mammals, human beings, because this toxin will get activated only in basic environment. Okay, but okay, higher animals have acidic stomach. My stomach pH is highly acidic. In that conditions, this BT toxin will be disintegrated. So nothing happens to me if I consume BT crops. Is it clear? It will affect only the worms, not to all the worms, only the bollworms. Not only bollworms, but narrow spectrum, only few types of worms. Okay. It promotes multiplication of beneficial insects that are predators on the bollworms. Bollworms will die. Okay. Some beneficial worms will eat these, and insects, birds and all, they will eat these worms and they will help in the agriculture. These are the advantages of it. Okay. Disadvantage: seeds are more expensive and bollworms are developing resistance, and Monsanto company is working with commercial interests. Is it clear? What is the commercial interest of this company? Okay, these seeds can be used to raise only a single crop. The seeds from this crop cannot be used to get the next crop. So that is called terminator seeds. Terminator seeds. These seeds can be used only to get a single crop. From that crop, whatever seeds we get, we cannot use them to get the next crop. So every year the farmer need to purchase the seeds. Commercial benefit for the company, but loss for the farmer. So these are the disadvantages. Next one, BT brinjal. Is it clear? BT brinjal. Okay. BT brinjal, same thing. Okay. Just replace the word, okay, cotton with brinjal. I hope you understood. Okay. and replace the word bollworms with Lepidopteran insects. We call as fruit and shoot borers. FSB we call. You might have noticed that brinjals always will have holes in that, and if you cut open the brinjal, worms will be. That is the reason why several people are afraid of cutting the brinjals, including me. Okay, so Lepidopteran insects, they usually attack the brinjal crop. Okay, and if you add this particular gene, such a brinjal crop will be naturally resistant to Lepidopteran insects. You will not have worms inside such brinjals. Is it clear? Okay. In India, we are cultivating BT cotton, but we are not cultivating the BT brinjal. Is it clear? We are not cultivating the BT brinjal. Is it clear? Why? Because brinjal is a food crop. Brinjal is a food crop. Okay, directly we eat it. So though we say that the toxin is not very poisonous to the human beings, in the acidic gut it will get digested, true, but still, okay, why to eat that toxic brinjal only, as if we don't have anything else in the world to eat? Okay, human beings have so many things to eat. Okay, so you don't have to dare to eat that. But in some countries, I think in Bangladesh and all, they are actually consuming the BT brinjal. It is not actually harmful. But to be on safe side, we are not consuming it. In India, the only GM crop which is commercially cultivating is BT cotton. Is it clear? What is the only GM crop that is approved for commercial cultivation in India, not for imports? Import, you can import any GM crop. What is the only GM crop which is approved for the commercial cultivation within the country? Okay, BT cotton. I hope it is clear. BT mustard is not approved for commercial cultivation. Is it clear? Approval for trials is different. Approval for commercial cultivation to sale in the market is different. Okay. The only GM crop approved for commercial cultivation is BT cotton. I hope it is clear. Then the next one is GM mustard. Okay, Dhara mustard hybrid. After this, you are going to get the break. So in the enthusiasm of having the first break after 10 minutes, make this 10 minutes most productive. Okay. Okay, the details are given here. Okay, who will approve it? I have given some details there. Okay, Ministry of Environment, Forest and Climate Change. Right? Ministry of Environment, Forest and Climate Change. Done? Okay. Next, Dhara mustard hybrid. So here, okay, try to understand. So we are going to make this 10 minutes very productive, and we are going to complete this topic within this 10 minutes, right? Okay, otherwise your break time will be consumed. So more focus. Earlier, the bacterium which we have used is Bacillus thuringiensis. Now the bacterium which we are using is Bacillus amyloliquefaciens. Okay, Bacillus amyloliquefaciens. Amyloliquefaciens. Earlier we have taken only one gene, Crivinaceae. Now you are taking 3 genes, bar star, bar nace, bar. Okay, now you are taking 3 genes, bar star, bar nace, bar. Okay, now some background information. Okay, see, for example, okay, if you take a flower like this, you will have stamen, pistil. Okay, this is male part, this is female part, then the pollen grain will fall on this. This is called self-pollination. Is that clear? Means this plant is pollinating its own self, not pollinated by the other plant. Clear till now? self-pollination. Then what do you mean by cross-pollination? Is it clear? Look at here. Okay, this is stamen, that is male plant. Then this is pistil, this is female plant. Okay, if you observe in the self-pollination, male, female both are in the same plant. We don't have a separate male plant, female plant. But here you have male and female in two separate plants. Is it clear? Then the pollen grain is transferred like this. This is called as cross-pollination. Is it clear? This is called cross-pollination. Clear till this? Okay. And now, complete focus please. Natural mustard. Natural mustard is self-pollinating. Is it clear? Natural mustard is self-pollinating. Okay? But, okay, we want to make it cross-pollinating. Understood? Natural mustard is self-pollinating, but we want to make it cross-pollinating. Why do you want to make it cross-pollinating? So that, okay, you can make hybrids. You can make hybrids. Okay. I hope you know what is hybrids. Okay. Hybrid means what? For example, you have red rose. You have white rose. A hybrid rose is red and white rose. Okay. I mean mixing two varieties. Mixing two varieties. I hope it is clear. Okay. So, is self-pollinating plant suitable for hybridization? Is a self-pollinating plant suitable for hybridization? No, because its pollen grain will fall on its own self. If it is cross-pollinating, then one variety pollen grain falls on the other variety, so that the two varieties are crossing and you will get a mixture of the two. Is it clear? Okay, very good. Clear till this point? But now you want to make it cross-pollinating. So what it will do is, okay, look at here. Okay, why cross-pollination is not possible here? Why cross-pollination is not possible here? Let us try to understand. Complete focus here please. This stamen is like a possessive boyfriend. Is it clear? I am comparing this stamen as a possessive boyfriend. Okay. Not letting the pollen grain of the other plant fall on his girlfriend. Is it clear? Not letting it mate with anybody else. But now you want to cross-pollinate. I hope it is clear. You want to add the pollen grain of another variety. What you have to do? Is it clear? Inactivate the stamen. I mean, okay. So give some sleeping pill so that that possessive boyfriend will sleep for some time. Is it clear? Boyfriend ko sula do. Is it clear? Okay. So this stamen is now inactivated. Okay. Now which gene will do that? Which gene will do this? I hope it is clear. Okay. Barnase gene, okay, will cause male sterility. I mean it will inactivate the stamen. I hope it is clear. Okay, inactivate the stamen. Now, the possessive boyfriend is sleeping happily for some time. Now, meanwhile, what you can do? You can add the pollen grain of this plant on that. Secretly add the pollen grain of this plant on that. Did you understand? Now your work is completed. Cross-pollination is over. Now remember, we only made the boyfriend sleep for some time. We did not kill him. Is it clear? So another gene bar star will restore the male fertility. It means it will reactivate the stamen so that, okay, in the next generation we will get fertile seeds. Is it clear? I don't want sterile seeds. I just want to inactivate the stamen only till I add whatever pollen grain I wanted to add. I hope it is clear. Okay? Right. Restore the male fertility. That's it. Okay. Inactivate the Barney's gene will inactivate the stamen. You add whatever pollen grain you want, and then Barstar will reactivate the stamen. Now the mustard is cross-pollinating, means you can cross it with any other variety you want. Okay. And what is the role of the third gene? Third gene is Bar gene. It will give herbicide tolerance. A herbicide tolerance. Why do we use herbicides? Herbicide means what? You know, herb means plant. Sidal means killing. Plant killing. Why do we use herbicides? Okay, to kill the weeds. To kill the weeds. Weeds. Is it clear? So that is how we use herbicides. Okay, but Bar gene will give herbicide resistance to this particular plant. So what happens? Okay, even if you spray the herbicides, only the weeds will die. Your mustard plant will not die. Is it clear? So that is the role of the third gene. I hope it is clear. The three genes are clear. Okay, Barnase will inactivate the stamen. Then you add pollen grain from whatever variety you want. Create hybrid. Barstar gene will restore the male fertility, reactivate the stamen, so that you will again get fertile seeds. Bar gene will give herbicide tolerance, so that even if you spray the herbicides, only the weeds will die, nothing will happen to your mustard plant. Is it clear? So these are the three advantages. But why it is not approved? What are the problems with this? Is it clear? Long-term ecological, social economic problems are possible. How come social economic? The seeds will be costly. Okay, next. There is a possibility of this herbicide tolerance gene spreading. If the plants will become herbicide tolerant, what is the problem? The problem is people will use more herbicides. For example, if I spray the herbicide, if my crop also will get damaged, then I will hesitate to use herbicides. But if my crop is herbicide tolerant, freely I will use herbicides without hesitation. It will lead to soil pollution and all those problems. Moreover, it needs more water, more fertilizers. So it is not a very sustainable crop. So therefore, it is not approved. Okay. Look at this question. With reference to GM mustard, consider the following. GM mustard has the genes of a soil bacterium that give the plant the property of pest resistance to? No. Okay. It is not pest. Pest resistant is BT cotton. This is herbicide. Herbicide resistant. Is it clear? Okay. Pest means worms. Herbicide means some chemical. Is it clear? It is resistant to that chemicals. Okay. GM mustard, the genes allow the plant to cross-pollinate and hybridize? Yes, correct. Okay. That possessive boyfriend putting him to sleep. Okay. GM mustard has been developed jointly by IARI and Punjab Agricultural University. No, those details are actually given here, which, okay, made by this Delhi University and Bayer Corporation. Okay. So this is also, okay, wrong. 2 only is the right answer. So sharply 4:10. Now we are starting the break, first break. Let us again join the session sharply by 4:30 p.m. Thank you so much. Have a break. Okay. Bye-bye. Welcome back all of you. You're able to hear me? Is my audio video clear? Yes. Okay. So let us continue. Till now we have seen genetically modified crops. Now the next topic is transgenic animals. Okay. Let us see the examples for transgenic animals. Okay. First example is cows. Sorry for the spelling mistake. Dairy cows. Is it clear? Containing extra copies of casein gene. What is casein? Casein is a milk protein. Casein is a milk protein. Okay. And do the cows naturally have the casein gene or not? Yes, all of you. Do the cows naturally have the casein gene or not? Yes, they have. But what we are doing here? We are adding extra copies of the casein gene. So what happens? You don't have to mix a protein powder in the milk. The milk itself will be like a protein shake. 13% more milk protein will be there in that. Is it clear? Okay. I repeat, add extra copies of casein protein gene into the cow embryo. Then the resulting cow will give the milk which is more proteinaceous. Just the milk itself is like a protein shake. That is one application. Okay. Second example, cows that produce human milk. Simple. Human casein gene transferred into cow embryo. Then the cow will give the human milk. Sometimes, okay, in the case of premature babies and all, the mothers will not get milk or few women do not get enough milk. Then usually we give powder milk to the babies. We call it as formula milk. But that is having its own disadvantages. More sugar contents, more fat content. Okay. So therefore it is better to give natural milk. Is it clear? So transfer human casein gene into the cow embryo so that the cow will give you the human milk. That is the second example. I hope both the examples are clear. Third one, transgenic fish that grows faster. You know, we all have some growth hormone. Growth hormone is responsible for growth. Now tell me, does a natural fish have a growth hormone? Yes. But we are adding extra copies of growth hormone gene to the fish so that the fish grows faster, larger, weighs more. So commercially beneficial for the fish farmer. I hope it is clear. Third example. Fourth example. Okay. What we have done is look at this image. Quite interesting. This is called glowing monkey Andy. Andy glowing monkey. You know, we have jellyfish. Have you seen the jellyfish? And they live in the deep sea water and they are fluorescent. Is it clear? From them, in them they have a protein called green fluorescent protein. Green fluorescent protein. Who will have this protein? Jellyfish. Who will have the green fluorescent protein? Jellyfish. Okay. Cut that particular gene from jellyfish. Transfer to monkey embryo. Transfer to monkey embryo. You get a glowing monkey, fluorescent monkey. I hope it is clear. Okay. Deep sea water, jellyfish. It has a protein called green fluorescent protein. You are cutting the gene for the green fluorescent protein from the jellyfish, adding to monkeys, rat, cats, so that you will get fluorescent animals. Okay. So this is not having much significance. Okay. This is just for, just imagine having such a fancy pet. That's it. No specific advantage because of that. Okay. Then moving on to the next topic, that is cancer therapy. First of all, what is cancer? Okay. To understand what is cancer, we have to know what is tumor. tumor is defined as unregulated cell growth, that is excess growth of the cells. Excess growth of cells is called as tumor. Okay, and tumors are two types: benign tumors, malignant tumors. Benign tumors cannot spread, malignant tumors can spread. So only malignant tumors are called cancers. So tumor is an extra growth of the cells. We have two types of tumors, benign, malignant, and only malignant tumors are called cancers. That is a cancer. Okay, and in our body, okay, a constant number of cells is being maintained by two processes. One is cell division. Second process is called as apoptosis. Apoptosis is also called as programmed cell death. Apoptosis is also called as programmed cell death. So this leads to increase in the cell number, isn't it? This leads to decrease in the cell number. Is it clear? Then when do we get cancer? When cell division, okay, increases, when cell division increases or apoptosis decreases. It means cells grow more than the requirement or cells do not die at the expected rate. In both the cases, what happens? Number of cells increases. That is nothing but a tumor. Tumor is an extra growth of the cells. I hope it is clear. Okay. So it means a coordination between these two is very, very important. Coordination between cell division and apoptosis will prevent cancer. Do you agree? What causes cancer? Increase in the cell division or decrease in the cell death. Okay, increase in the cell division or decrease in the cell death will lead to cancer. So what will prevent cancer? A coordination between these two. I mean, if the cell number increases, if the cell division increases. Okay, simultaneously if apoptosis also increases, it means what? Okay, more cells are formed but more cells are dying also, so no problem. Cell division decreases, apoptosis also decreases, no problem. The rate of new cell formation is less, so the rate of cell death is also less, balanced, no problem. So we do not get cancers as long as these two processes are balanced and coordinated. But if the cell division increase and apoptosis decrease, cells are not dying but more cells are forming, that leads to cancer. Is it clear? Okay. And we have some genes called proto-oncogenes. What is their function? They regulate and coordinate the cell division and apoptosis. So tell me, do they prevent cancer or cause cancer? Read this. What is the function of proto-oncogenes? They regulate and coordinate the cell division and apoptosis, balance these two. So do proto-oncogenes cause cancer or prevent cancer? Very good. They prevent cancer. Okay. Then what happens if the proto-oncogenes gets mutated? If the proto-oncogenes gets mutated, then we get oncogenes. If the proto-oncogenes gets mutated, we get oncogenes. So what are oncogenes? They are the mutated or defective proto-oncogenes. So do they cause cancers or prevent cancers? Oncogenes are the mutated or the defective proto-oncogenes. Okay, if the proto-oncogenes fail to balance those two mechanisms, then we form oncogenes. Okay, so oncogenes cause cancers. Oncogenes cause cancers. So very good. Then what is the cancer treatment? As you all know, the regular cancer treatment procedures are chemotherapy, that is treating the cancers using some chemicals, radiotherapy, killing the cancer cells using some gamma rays, high intensity radiation, okay, or surgery, surgically removing that cancer. These are the traditional treatment procedures, isn't it? Traditional treatment procedures are chemotherapy, radiotherapy, or surgery. But our topic is not about chemotherapy, radiotherapy, surgery. We are going to discuss about new developments in the cancer treatment. Okay. First one is gene silencing using CRISPR-Cas9. All of you answer this question. Which gene you have to silence to cure the cancer? Which gene you have to silence to cure the cancer? Everybody. Very good, perfect. Oncogene you have to silence. Is it clear? How to silence the oncogene? Simple. Do you remember this CRISPR-Cas9 mechanism? Okay, look at this CRISPR-Cas9 mechanism. In the CRISPR-Cas9 mechanism, now imagine that this is mutated. Look at this, all of you. That is mutated proto-oncogene. Mutated proto-oncogene means what? Oncogene. Oncogene. Okay. So, what is this one? You removed the, you removed the oncogene and add the proto-oncogene. That's it. That is cancer treatment. Using CRISPR-Cas9, remove that oncogene, that is a mutated one, add the correct one, that is the proto-oncogene. The oncogene is silenced, cancer cured. Okay, this is one new method, gene therapy in cancer treatment. Clear till now? Yes, perfect. Same thing. Now let us see a second procedure. Okay, RNA interference. RNA interference. Okay, complete focus here, please. Let us suppose this is oncogene, right? Then this is oncogene mRNA, UACGGGAUAG. This is mRNA of oncogene. Is it clear? Then what is this procedure called as? Transcription. We call this procedure as DNA to RNA is called as transcription, not translation, transcription. Then if that becomes protein, if that becomes a protein, that process is called as translation. Is it clear? DNA to RNA transcription, RNA to protein translation. Okay. But now what we are doing is, look at here. You are adding what is this? This is called as small interference RNA. Is it clear? Small interference RNA. Okay. Or RNA I. Interfering RNA. Interfering RNA. Okay. Why this is called interfering RNA? Because it will interfere with the translation of oncogene mRNA. I hope it is clear. It will interfere with the translation of oncogene mRNA. Okay, look at here, I will explain this. Imagine this is oncogene mRNA, right? What is this? siRNA, small interference RNA. Okay, it will bind to the oncogene mRNA. Now this is blocked. It is not expressing. Okay, so like earlier this pen was writing, but then you covered it with the cap. Now the pen cannot write. Just like that, earlier the oncogene mRNA can undergo translation to produce a protein, but now the siRNA is bound to it just like a cap. Okay, it will prevent the translation, so that protein is not produced. Okay, so that protein is not produced. So cancer, okay, not occurring. I hope it is clear. That is called as small interference RNA. Gene silencing using small interference RNA or RNAi, we also call this as antisense RNA. We also call this as antisense RNA. Difference between the two. In the first case, we have cut the oncogene and added the proto-oncogene. Is it clear? We are doing it at the DNA level. In the first case, you are correcting at the DNA level. In the second case, you are not touching the DNA. You are dealing at the RNA level, blocking the translation of the oncogene RNA. Whereas in the first case, okay, you are directly cutting the oncogene and adding the correct copy of the oncogene. Is it clear? Okay. I hope the difference is clear. Very good. Okay. Then the third mechanism. Complete focus, please. Okay. The third mechanism, immunotherapy for cancer. Immunotherapy for cancer. Actually, this is also a Nobel Prize topic. Okay. James P. Allison, Tasuku Honjo won the Nobel Prize. Okay, for this. Okay. Right. So here, let us see a little terminology to understand this topic. Okay, we have two important cells in our immune system. One is B cells. They produce antibodies which will destroy foreign antigens. It is like border security force. I mean, if any pathogens are coming from outside, the B cells will kill them. I hope it is clear. Next, T cells will destroy altered self cells. I mean cancer cells. For example, cancer cells. For example, for a cancer patient, cancer cell is not coming from outside. It is developing from inside. Okay. For a cancer patient, cancer cell is a self cell, not a foreign cell, but modified. This is something like, okay, the Kurkure advertisement, Mera hai par teda hai, something like that. Is it clear? Mera hai, okay, it is my cell only, par teda hai, I mean modified. Now I should not protect it, I should not save it, I have to kill it. What is an example for such Mera hai par teda hai? That is cancer cell. Is it clear? Our own cell, but dangerous, became dangerous. Okay, it means internal security. T cells can be compared to internal security. I hope it is clear. B cells are like border security force. If somebody is coming from outside, they will take care. T cells are like if there are some enemies from within the body, they will take care. Is it clear? Okay. Now, what is CTLA4 and PD1? Look at here. This is a killer T cell. That kill cancer cells. We call this as CTL. CTL means cytotoxic T lymphocytes. Okay, cytotoxic T lymphocytes. Okay. Yes, Tejaswini, it is like microRNA. I mean, I can also call this as microRNA. Okay, fine. Coming back to this. Immunotherapy for cancer. We understood what are the B cells, what are the T cells. Now imagine that this is a T cell. Such killer T cells are also called as cytotoxic T lymphocytes. Okay. On them you have some structures. Complete focus, please. On them you have some structures called as CTLA4 and PD1. CTLA4 and PD1. Right? What are they? They are brakes on the T cell. Okay. Why? To slow them down or to stop. I am comparing it like the leash of a dog. Okay. For example, you have a pet dog. You are taking it for walking and then you will hold it with a leash. Is it clear? So like that, it is like a leash, like a brake. It will keep the T cells under the control. Now, okay, you got cancer. Okay, now complete focus here, please. So what they have done is, okay, they made James P. Allison produced an antibody for CTLA4. It will destroy CTLA4. I mean like a brake fail. I hope you understood this. Now it is like a car whose brake failed. Okay, like just imagine you have a pet dog, you are taking it for a walk with a leash. Is it clear? And then all of a sudden some enemy is attacking on you. Is it clear? Okay, you have some enemies all of a sudden. Then what you will do? You will just unleash the dog. Now the dog will go and attack on your enemies. Like that, the CTLA4 and PD1 are like brakes. Normally we need the car to be under the control, we need the pet dog to be under the control. But when enemies are there, here the enemies are cancer cells, what you have to do? Unleash it, remove the brakes. Okay, James P. Allison produced antibody for CTLA4. Okay, it will destroy the CTLA4. Now it will behave like a dog which is unleashed, or it will behave like a vehicle whose brakes are failed. Same way Tasuku Honjo made an antibody for, same thing, this is an antibody for, okay, PD1. So this is another brake fail. Both brakes failed, both leashes removed. Now just like the dog will attack on the enemies, this T cell will attack on the cancer cells and kill them all. Okay, they will attack on cancer cells and kill them. Got it? Okay. Right. Okay. Next one. CAR T cell therapy. Okay. CAR T cell therapy. So what is like a CAR T cell therapy? Complete focus here, please. So here also same cytotoxic T lymphocyte. Okay. Cytotoxic T lymphocyte. Then on this there is one structure called as, okay, T cell receptor. Okay, on the cytotoxic T lymphocytes there will be something called as T cell receptor. Okay, and then now imagine this is a cancer cell. Which cancer cell? Okay, particularly it is, okay, some anemia. Some anemia. On this there is one antigen called as CD19. Okay, this is a unique antigen. Okay, on this cancer cell. I hope it is clear. Okay, this is a unique antigen present on this cancer cell. I mean, do healthy cells have CD19? No. Do other cancer cells have CD19 cell like liver cancer, pancreatic cell cancer, or lung cancer or cervical cancer? Do they have CD19? No. Only anemia cells will have it, or only leukemia, sorry, not anemia, leukemia, only leukemia cells. That is blood cancer. Okay, have this antigen. I repeat, normal cells do not have this antigen. Other cancer cells also do not have this antigen. Only leukemia cells have this antigen. Okay. And till now, this is all natural. But now what we are doing is, we are adding, okay, CD19 antibody to this. Okay. And this is, okay, I'll just write it here. What we are adding here is CD19 antibody. This is artificial. Remaining all naturally, I mean we naturally have cytotoxic T lymphocytes. They naturally have a T cell receptor. And the patient also got cancer naturally only, we did not artificially create it. And leukemia cells naturally possess this unique antigen called CD19. What is the only thing that we have added here? An antibody for CD19. Is it clear? Okay. And always remember, antigen and antibody are highly specific. Antigen and antibody are highly specific. Okay. So, now what happens? Now it is something like, again I will explain with a dog example. Military and police departments will train the dogs. Is it clear? So what they can do, for example, let us suppose you show the smell of a criminal's shirt or something to the dog. Okay. Then the dog will remember that particular smell and will particularly attack on that criminal only. Will not kill anybody on the way. Especially target that criminal. Like that only. It is like, now the CTL is like our dog. Is it clear? Now I am comparing the CTL with the dog. And adding the CD19 antibody is like showing the smell of the criminal's shirt to this dog. Now this particular CTL will particularly attack only on the leukemia cells. Okay, you tell me, can this particular CTL, I have added the CD19 antibody to this CTL, can this cytotoxic T lymphocyte now attack on the normal healthy cells or not? Everybody please. We have added the CD19 antibody to the cytotoxic T lymphocyte. Can such a cytotoxic T lymphocyte attack and kill the normal healthy cells or no? No, very good. Can they attack on any other cancer cells like cervical cancer, uterine cancer or ovarian cancer or something like that? No. Now you have, like, it is something like you have shown the smell of the criminal's shirt to this dog. It will now hunt that particular criminal. Like that, now this CTL will kill only leukemia cells. In general, what is the problem in cancer treatment? The problem in cancer treatment is not killing cancer cells, saving the healthy body cells. But by this mechanism, healthy body cells are not dying, only specific cancer cells are dying. This is called as CAR T cell therapy. But why the name CAR T? Okay, now look at here. This complete focus please. Okay, this T cell receptor plus CD19 antibody. I mean, okay, this complex. I hope it is clear. Is called as chimeric. I hope you remember chimeric means combination, mixture I said. You are combining the two. Which two? The T cell receptor plus CD19 antibody. It is a combination of the two. Chimeric antigen receptor. Antigen receptor means antibody. Okay, what will receive the antigen? Antibody, right? The receptor for antigen is antibody. So, CAR. Okay, why this is called CAR? Chimeric antigen receptor. CAR. What is CAR? Okay, T cell receptor, the blue color T cell receptor, look at here. This T cell receptor plus this CD19 antigen, both of them together is called as CAR. And the T cell with CAR is CAR T cell. I hope you got this. The T cell with CAR. T cell with CAR is, okay, CAR T cell. This is called CAR T cell therapy. Okay, so look at here what they are doing. Step 1, take the T cells out. Okay, step 2, make the CAR T cells in the laboratory. Okay, step 3, inject the CAR T cells into the patient's body. Step 4, the CAR T cells will attack on the cancer cells. Take the T cells out of my body, add the CD19 antibody to it, and then re-inject them into my body. Now they are like, take my dog out, okay, show the smell of the shirt to it and send that dog again into my home. something like that. I hope it is clear. That is CAR T cell therapy, right? Okay, next. There is one question here. RNA interference technology gained popularity in the last few years. Why? It is used in developing gene silencing therapies. RNA interference. I hope you remember this. RNA interference, small interference RNA, RNAi, interfering RNA, microRNA, okay, antisense RNA, okay. Such a therapy is gaining popularity. Why? Okay, it is used in gene silencing therapies, correct. Developing therapies for cancer, correct. It can be used to develop hormone replacement, no. Okay, hormones are some chemicals, they are not genes, so it is something related to gene therapy. Okay, then it can be used to produce crop plants that are resistant to viral pathogens, yes. Is it clear? Okay, how? I'll tell you one example. So this is a plant cell, okay. Then it has a receptor for virus, right? Okay. Then this is the virus, plant virus. This is the plant virus, okay. It means this will work like a gate for the virus. The virus enters into the plant cell through this gate. Okay. I will tell you another famous example. Okay. That is, did you hear about this ACE2 receptor? ACE2 receptor or ACE receptor. Okay, and this is coronavirus, spike proteins. Okay, ACE2 receptor is like a gate for it. Did you notice during the COVID pandemic, some people who got COVID, they got very severe symptoms. Okay, even it led to the death. But for some people, there are no symptoms at all. What is the difference? There are many differences, age, okay, like that, other comorbidities, like that there were several factors, but one important factor is this ACE2 receptor. What this ACE receptor will do? It is like the gate for the virus. For example, my lung cells have more ACE receptors, then more gates. So through more gates, the viruses can flood into my lung cells. So I'll get more severe symptoms. If my lung cells do not have ACE2 receptors, okay, then what happens? The virus cannot enter into my cell, then I will not get the disease. Even if the virus enters into my body, I will not get symptoms. Or I have ACE2 receptors but few of them, then I will get mild symptoms like some mild cold or something like that. So like that, even plants have such gates, okay, even plants have such gates for, okay, even plants have such gate for the plant viruses. How to make, okay, this particular plant cell resistant to this virus? Okay, just gene silencing of the receptor producing gene. I hope it is clear. Okay, the gene which is producing this gate, suppress it using RNAi. Using RNAi, suppress the gene which produce this gate. So virus cannot enter. Now the plant is resistant to viral infection. I hope it is clear. Okay, so 1, 2 and 4 is the right answer. Very good. Then moving on to the next topic. Okay, DNA fingerprinting, DNA testing, DNA profiling, DNA barcoding, all of them mean the same. Is it clear? Okay. Now, I will not enter into the procedure of this topic because it is a lengthy procedure, and in the prelims examination they cannot ask you the questions about the procedure of it. I mean, they cannot ask you How do you do it? Tell me. Is it clear? Okay. So basically I will tell you the principle and then I will proceed to the next topic. Okay. First of all, why this is called DNA fingerprinting? I mean, do we do this? Okay. Do we use fingerprints, thumb impressions here? No. Okay. Then why the word DNA fingerprinting? Because you all know that our fingerprints are unique to us. I mean, no other human being will have the same fingerprint as I have. Same way, DNA is also unique to me. I mean, no other human being will have the same DNA as I have. So, indicating that uniqueness, it is called DNA fingerprinting. It is also called DNA testing. Also called DNA profiling or DNA barcoding. Why this is called DNA barcoding? A barcode is also unique, isn't it? For example, if you go to a supermarket, if you scan the barcode of one item, you will get the cost of that item only, right? You will not get the cost of the other item. Every object in that supermarket will have a unique barcode. So like that only every human being will have a unique barcode, unique DNA sequence. So the DNA testing can also be called as DNA barcoding. And yes, identical twins are an exception for it. Okay, what is one limitation of this technique? Okay, this cannot differentiate. This cannot differentiate between identical twins. I hope it is clear, because identical twins have the same DNA sequence, or even clones also, but anyway we do not have human cloning, so that is not an important thing. Okay, but actually between all the human beings, most of the DNA is similar. For example, like around 95-96% of our DNA is actually similar. But do you remember we studied about the junk DNA? That part of the DNA is highly variable. Okay, so most of the differences between you and me, okay, exist in the junk DNA. We also call it as highly variable region. Also call as, we can also call them as short tandem repeats. Like for example, a sequence called ATG repeating several times, like this ATG, 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 ATG. We call them as short tandem. Tandem means continuous. Tandem means continuous. Okay, repeats. Short tandem repeats. Okay. For example, if I have 4 repeats, then you have 7 repeats, okay, then we both will differ. So that technique is called as short tandem repeats. Is it clear? I have only 4 repeats there, but you have 7 repeats. It means my DNA sequence is different from your DNA sequence at that particular place. Okay, so based on that we can identify that we both are unrelated. Okay, so like that. Such DNA is also called as mini satellite DNA. Such repetitive DNA is also called as mini satellite or microsatellite DNA. Is it clear? Okay, so we compare different people at such sequences. Okay, if 100% similar, if the DNA sequences are 100% similar, one possibility is they are identical twins, or they are the two samples from same individual. They are the 2 samples from same individual. Okay, if there is 50% similarity between 2 human beings, if the DNA is around 50% similar, it means they both are parent and child. Is it clear? Around 75% similarity, they both are siblings. If there is less than 50% similarity, okay, we cannot predict. You cannot predict what is the relation. Is it clear? Okay. And 100% similarity, 2 samples from the same individual means what? You have taken one sample from the crime scene and the second sample from the suspect's body. For example, let us suppose a criminal is having, in the forensic cases, a criminal is having hair fall problem. Okay, and he committed a murder. In that place, one of his hair is left. So that is the DNA. From that I will extract the DNA, and that is the DNA which I got in the crime scene. And then I am doubting a person. I will take DNA from his body. I will compare the two. If they both are 100% matching, it means that is his hair which is left in the crime scene. So he did that particular murder. Or rape, for example, a rape case. Okay, victim's body, you found some semen. Is it clear? From the semen we have extracted the DNA. And then you have a suspect, somebody caught in the CCTV camera footage and all. You match the DNA with that person. If it is 100% matching, then he did that particular rape. Okay, 50% similarity we use in the disputed paternity cases. Okay. Disputed paternity cases means, like say for example, okay, a surrogate mother. Okay, let us suppose in future, if the surrogate mother claims that the Priyanka Chopra and Nick Jonas son or daughter, I don't remember, the child is her child only, can we solve that case using DNA testing? Yes. The DNA of the baby will be similar to Priyanka Chopra and Nick Jonas, not similar to the surrogate mother, because she is only the biological mother. DNA came from them. So like the disputed paternity cases, ART cases, ART means assisted reproductive technologies, that is like surrogacy cases. In future, if the surrogate mother claim that this is my baby, we can solve it using, okay, DNA testing. Is it clear? Same way, wars, plane crashes, mass calamities, to identify the remains of the body. In such cases, we use the DNA fingerprinting. So that is about the DNA fingerprinting. What kind of questions you can expect in the prelims? Okay. Consider the following statements. DNA barcoding can be a tool to assess the age of the plant? No. Our DNA sequence will not change with age. Whatever DNA, I got my DNA from my mother, father. I will retain the same DNA throughout my life. Yes, mutations can occur, but mutations are very, very rare. They do not alter my DNA sequence considerably. Is it clear? Okay. So you cannot use DNA testing to assess the age of the person. No. Distinguish between the species? Yes. Okay. When you can distinguish between two human beings, that is two members of the same species, why can't you distinguish between two different species? For example, if you and I differ in the DNA, why can't I and a tiger differ in the DNA? So there will be more differences, right? Yes, we can. Identify the undesirable animal plant material in the processed food. For example, okay, I am eating some burger, okay. In that, okay, I got some, or for example, I am eating some brinjal curry, okay, and in that some goat DNA is observed. Means what? It is contaminated. Or some bacterial DNA is observed. Means what? It is contaminated. Yes. Okay. So we can use it for that also. 2 and 3 is the right answer. I hope you understood. Okay. Done. Next one. Another question. With reference to the recent developments in science, which of the following statements is not correct? Okay. Pieces of artificial functional DNA can be created in the laboratory? Yes. Okay. One example, we created the CRISPR. Okay. Okay, CRISPR is an RNA, but do you remember HGP, right? There I told you that you can create a complete genome also. Then why can't you create a small piece of DNA? Pieces of artificial functional DNA can be created in the laboratories. Yes, you can also create a whole genome as well. I hope it is clear. Synthetic biology. A piece of DNA taken out from an animal can be made to replicate outside a living cell in the laboratory. Yes, we call this as gene cloning, if you remember that recombinant DNA technology bacteria diagram. Cells taken out from the plants and animals can be made to undergo cell division in the laboratory. Yes, this is called tissue culture. Okay. Which among the statements is not possible? This is not possible. We cannot join the fragments of chromosomes to form a new chromosome. Okay. Functional chromosomes cannot be created by joining segments of DNA taken from different species. Okay. Human chromosome, one piece. Okay. Goat chromosome, one piece. Together I can make a functional chromosome out of it. No, not possible. 1 is not possible. So the correct answer is 1. Okay. A is the right answer. Okay. Cow milk we have done. Okay. But that is not like a new chromosome. We just added one gene. But here it is like 50-50. Okay. 50% chimpanzee, 50% human, chimeric chromosome as such. That we cannot do. Okay. Okay, Nagendra, I hope the difference is clear. Okay, next one. Some infections in the news. Okay, let us see about the infections. Okay, first, okay, first one, prions. What are prions? This is just a free living protein. Okay. Free living protein. In fact, this is like a misfolded protein which is infectious. Okay. Prion examples are Kuru, Scrapie. Okay. They are above 90% fatality. Okay, fatality. It means almost sure shot death. Very dangerous diseases. Okay. Wrongly folded protein or a free living protein. Okay, they are called prions. They cause diseases. 2 examples are Kuru, Scrapie are the examples of the diseases. They are very dangerous diseases. Okay, and they are almost 100% fatality. Is it clear? Okay. Scrapie, first one. Second one, okay, viruses. Viruses. Virus is, it will have 2 components. Okay, I'll explain about the viruses here. Yes. Okay, viral infections. A virus will be having 2 components. Okay, one is a protein coat plus genetic material. Genetic material. Okay, this genetic material can be either DNA or RNA. I hope it is clear. The genetic material can be either DNA or RNA. Right. So, viruses are acellular, I mean no cell. They have only a protein coat and genetic material inside. They are much smaller than the bacterial cells. They are basically just capsules that contain a protein capsule that contain genetic material. They have either DNA or RNA, never both of them. Is it clear? They are never both of them, right? Okay, then now if the virus contain RNA, I mean protein coat plus RNA inside, they are called RNA viruses. What are the examples for RNA viruses? Okay, most of the human viruses are RNA viruses. Is it clear? Zika, Nipah, Ebola, SARS-CoV-2, flus. Is it clear? Hepatitis. Most of the human viruses are RNA viruses. So it is not possible to give examples for that. But what I will do, I will give examples for other viruses. Except them, remaining all are RNA viruses. I hope it is clear. So second type are DNA viruses. DNA virus will be having a protein coat and DNA inside. I hope the difference is clear. Protein coat and DNA inside. For this we have examples. Most of the plant viruses are DNA viruses. Most of the animal viruses are RNA viruses. Is it clear? And most of the plant viruses, but some of the human viruses are also DNA viruses. What are they? Herpes virus, smallpox virus, adenovirus, hepatitis B. Interestingly, hepatitis A, E, C, they are RNA viruses. Only hepatitis B is DNA virus. Okay, remember this point. Human papilloma virus that cause cervical cancer, they are DNA viruses. 2 types over. Now third type is retrovirus. Okay, complete focus here, retrovirus. Retrovirus means they have RNA genetic material. Okay, they have RNA genetic material. But, when they infect the host cell, they send this RNA, there the RNA will get converted into DNA. I hope it is clear. Okay, they convert their RNA into DNA. This is viral RNA. They will convert this into DNA. Where? In the host cell. Okay. And insert this into host cell DNA. I hope it is clear. This is host cell DNA. In that, this will get inserted. I hope you got this. It will insert its DNA into our DNA. For example, HIV virus. It will bind to my cell, send its RNA into my cell, convert its RNA into DNA, and that DNA will get inserted into my DNA. Such viruses are called retroviruses. Why the word retro? Because I hope you all remember the actual order is DNA will become RNA. So if RNA becomes DNA, that is a kind of retro. I mean, it is a kind of a backward step. Is it clear? DNA to RNA, RNA to protein is the natural order. So such viruses are called as retroviruses. Okay. Yes, this is done by an enzyme called as reverse transcriptase. Who will do this? By reverse transcriptase. So total 3 types of viruses. First, viruses that contain RNA. Second, viruses that contain DNA. Third, viruses that contain RNA but will convert their RNA into DNA and then insert their DNA into the host cell DNA. Those are called retroviruses. These are the 3 types of the viruses and the examples. Okay. After virus, the next level, third one is bacteria. Okay, viruses are acellular, isn't it? Bacteria are unicellular. One cell. And also they are prokaryotic. Prokaryotic means what? Cell without true nucleus. Cell without true nucleus. They are bacteria. Okay. Next level we have, fourth level we have protozoa. Protozoa are also unicellular, that is one cell only, but they are eukaryotic. Examples, look at here, I have given the examples. Protozoal infections, malaria, Plasmodium that cause spreads through Anopheles, which will cause malaria. Leishmania that cause Kala Azar. Trypanosoma that cause trypanosomiasis. Entamoeba histolytica that cause amoebic dysentery. Okay, loose motions, blood motions, etc. That is protozoa. Basically, what is protozoa? Okay, they are unicellular but eukaryotic. I hope it is clear. Okay. Next one, common viral infections list I have given. Common bacterial infections list I have given. Okay. So like that I have given the list of, there is nothing to explain in this list of protozoal infections, bacterial and viral infections. Few important things I already mentioned here. Okay. Then now moving on to the next topic, that is immunity. Okay. In the immunity, we have two types of immunity. One is humoral immunity. Okay. As I told you, this is like the border security force, B cells. They produce antibodies for, okay, microbes, I mean foreign antigens. Foreign antigens, that is pathogens. Okay. Second one, cell mediated immunity. This is something like internal security, T cells. They are protected against intracellular pathogens and cancers. I mean, if there is an enemy coming from inside, either intracellular pathogen. What is an intracellular pathogen? Virus. Virus. Is it clear? Okay. And vaccines I already explained. Okay. And then look at this question. Okay. Which one of the following statements best describe the role of B cells and T cells? They protect the body from environmental allergens? No, actually correct, but in the multiple choice you have to give the most appropriate answer. So we are not choosing this. They alleviate body's pain and inflammation? No, they actually cause inflammation because immune response is a type of inflammation. So this is clearly wrong. They act as immunosuppressants? No, they actually cause immunity. So this is actually wrong. This is correct but not best, not most suitable. Is it clear? This is most suitable answer. They protect the body from diseases caused by the pathogens. I hope it is clear. Okay. Next. So that's all. With this we completed the, don't be super excited. With this we completed only the first segment, that is biotechnology. Okay. Now moving on to the second segment, that is nanotechnology. Right? Nanotechnology, but it will be little similar to biotech. Okay. So what we will do is first we will complete the next one, then we will come back. Okay. Nuclear technology. Okay. Now we are discussing nuclear technology. Right? Okay. Same way, complete focus please. Okay. First let us try to understand what is nuclear fission. What is nuclear fission? Okay. Based on your okay, let us first complete nuclear because it is a small topic. Okay. First we are discussing about nuclear fission. Fission means what? One heavy atomic nucleus breaks down into two light atomic nuclei. That is called nuclear fission. Is it clear? Complete focus please. Okay. Right. For example, uranium-235. In this, what is 92? This is the number of protons. Okay. What is 235? Number of protons plus neutrons. Is that clear? We started nuclear technology. Okay. And in the nuclear technology, first we are discussing about nuclear fission. Nuclear fission means one heavy atomic nucleus will split into two light atomic nuclei. Okay, breakdown is called fission. Example is here. Okay, uranium 23592. In this, what is this number 92? That is number of protons in uranium. What is 235? That is number of protons plus neutrons together in the uranium. Is it clear? Okay. It means this is having an odd number of neutrons. Agreed? This is having an odd number of neutrons. Okay. And okay, like neutrons, okay, will always love to be in pairs. Is it clear? They don't want like odd number. Okay, they prefer even number, pairs. Is it clear? So what happens is, when you give one neutron to it, did you notice you are adding one neutron? More focus please. When you add one neutron to it, no, okay, how come neutron is equal to electron? Okay, no. So like you speaking about the numbers? Fine. Now, so when you add one neutron to it, what happens? Uranium 235 is just like, okay, like some like a famous quotation, I am single ready to mingle, like that. Uranium is like that, it is single ready to mingle. So it means it is waiting for that neutron. The moment it comes, it will take it. Is it clear? When it takes that particular neutron, what happens is, it will lead to rearrangement of the neutrons. That will lead to breakdown of that uranium. Once you add a neutron to it, that uranium will split into barium and krypton. Barium and krypton. Is it clear? Plus 3 neutrons will be released out. Okay. This is a fundamental nuclear fission reaction. Uranium 235 plus 1 neutron. Okay. Then the uranium 235 will split into barium and krypton, releasing 3 neutrons. Clear till now? Okay. This is called neutron induced fission. Is it spontaneously breaking down? No. You are stimulating the fission by adding a neutron. Is it clear? Neutron induced fission of uranium 235 release about 200 mega electron volts of energy per atom, and this is the principle in the atom bomb. Okay, what is used in the atom bomb? Recently, last 1 week we have been discussing about nuclear threat in the context of the war between India and Pakistan. Okay, so so this is very much in the context. What is the principle in a nuclear weapon, atom bomb? This nuclear fission. Okay, but that is weapon usage, but we can also use this principle for peaceful usage. That is, if you do this nuclear fission in a controlled manner, okay, then what happens? It will generate heat. Heat can be converted into electricity. So we can also use the same principle in nuclear reactors for power generation. I hope it is clear. The weapon usage is atom bomb. The peaceful usage is nuclear reactors to generate electricity. That is the application of nuclear fission. Okay. Then moving ahead. We have 2 types of material. Is it clear? Fissile material, fertile material. It will be little confusing, so complete focus on the class please. Complete focus. Fissile material. Always remember, okay, fissile material will have odd number of, okay, neutrons. I mean, if this number is odd, uranium 235, plutonium 239, plutonium 241, okay, odd number of neutrons, then such material is called fissile material. Is it clear? Okay, right. Next, fertile material. Fertile material means they will have even number of neutrons. Okay, like uranium 238, plutonium 240, thorium 232. Okay, these are called as like fertile material. Is it clear? Okay, means this is like, as I told you, single ready to mingle. Is it clear? I mean, this is kind of, okay, even a, see, let us suppose, okay, say this is already single, there is no pair. Okay, now what happens? A girl need not be very attractive. Even a very dull and average girl can, okay, cause this particular reaction. Same way, you don't need a fast neutron. Just add one slow neutron to it. Look at here. Fissile material, uranium 235. You are just adding one slow neutron, enough. Okay, so for this reaction to occur, because it is waiting for the neutron, so any neutron is okay. It does not need a fast neutron. Any neutron is okay, just come inside like that. Is it clear? Okay, so when you add a slow neutron, directly it will undergo fission. Is it clear? So all of you tell me, what happens if you add a slow neutron to a fissile material? Fast, everybody. What happens if you add a slow neutron to a fissile material? What happens to a fissile material when you add slow neutron? It will directly undergo fission. Fission reaction starts, right? Okay. But, okay, now let us take fertile material. Example, thorium 232. Is it clear? Okay. Now, you add a slow neutron. See, fissile plus slow neutron. What happens? Fission. Okay. Now, fertile plus slow neutron. What happens? Nothing. Okay, because they are all paired. Is it clear? So if a super fast girl comes, I can't say, I can't give a guarantee, but if a slow girl comes, there won't be any reaction. I hope you got my point. Okay, so like that, here the neutrons are all paired. They are not waiting for a neutron. So if a slow neutron comes, they don't react. Nothing happens. They'll just keep, acha, okay, you came, it's okay, fine, like that. I hope it is clear for all of you. Okay. Then, now, okay, to fertile material plus fast neutron. Okay. Then what happens? Tell me fast. Yes, they need extra push. Okay. So like what happens here? Fertile material to that fast neutron is added. No, it will not become fission. It will not undergo fission. It will first convert to, okay, it will first convert to fissile material. I hope it is clear. Look at here. This is fertile material, thorium 232. You have added one fast neutron. Then it will take up the fast neutron and will become uranium 233. 232 became 33. Did you notice the number? Thorium 232 became uranium 233. I hope it is clear. This process is called as transmutation. This process is called as transmutation, right? So simple, I have given the summary here. Fissile material plus slow neutron, okay, it will undergo fission because ready for the neutron. Fertile material plus slow neutron, no reaction, okay, because it is not waiting for a neutron. Its neutrons are already in pairs, okay. But fertile material with fast neutron, okay. So first of all, the neutron enters into the atom. It will become fissile material, okay. And then, okay, then if you add one slow neutron to it, Okay, then it will undergo fission. I hope it is clear. Two-step process. Fertile material plus fast neutron, it will become fissile material. To this, if a slow neutron, step 1, step 2, then it will undergo fission. I hope it is clear. Fertile material, to that fast neutron is added. Then the fast neutron will penetrate into the cell. Okay, and then it will first become the fissile material, that is transmutation. Now it is fissile material. So if a slow neutron is added to it, then it will undergo fission. Okay, I hope the three processes are clear, the three alternatives. Very good, super intelligent. Thank you so much. Okay, next, what do you mean by isotopes? Isotopes means, okay, simple, this number must be same, this number must be different. Is it clear? Okay, two or more types of an atom, they are the different types of same atom. Okay, their atomic number is the same. Atomic number same. Atomic number, it means what? Number of protons. It also means position in periodic table. Position in periodic table, same. Okay. So, number of protons or atomic number or its position in the periodic table. For example, position of helium in the periodic table is, okay, second element. So, it means its protons are 2. For example, okay, uranium proton number is 92. It means it is 92nd element in the periodic table. Is it clear? Like that. Is it clear? Okay, they are same, but okay, different mass number. What is mass number? Protons plus neutrons number. The above number is different. This same, this different. Such things are called isotopes. Is it clear? Can I call uranium 235 and uranium 238 as isotopes? Uranium 92, 235. Uranium 92, 238. They are isotopes. I hope you all understood the concept of isotopes. Yes. Very good. Okay. So, till now, again I am giving you the same instruction. The topics will have connectivity. If you understand the first point, you will understand the second point better. So, complete focus. Nuclear fission we have seen. Fissile, fertile material and isotopes we understood. We have seen three alternatives: fissile material plus slow neutron fission, fertile material plus slow neutron nothing, fertile material plus fast neutron, then it will become fissile material. To that, if you add a slow neutron, then it will undergo fission. Two steps process. Okay. So, these are the things. Next one, uranium enrichment. What do you mean by uranium enrichment? Complete focus, please. Okay. Right. Uranium enrichment means, see, natural uranium is 99.3% uranium 238, 0.7% uranium 235. I hope it is clear. That is natural uranium. What do you mean by enriched uranium? Okay, if uranium 235 is more than 0.7%, that is called enriched uranium. Is it clear? For example, okay, 97% uranium 238 plus 3% uranium 230, okay, 5. Can I call this as enriched uranium? Yes. Okay, and remember, enriching up to 20%, okay, can be used in the nuclear reactors. Okay, and 20% is not minimum, 20% is maximum for reactors. Is it clear? Around 3 to 6% is enough for reactors. Is it clear? Okay. Low enriched uranium. This is low enriched uranium. Okay, enriched but low enriched. I mean, uranium-235 is more but not very high. That is called as low enriched uranium. 3 to 4%, 3 to 6% is enough. 20% is upper limit. You don't, at any cost, reactor cannot use more than 20% uranium-235. I hope it is clear. Okay. Unnati, nothing, simple. Okay. When you extract uranium for the ore, the natural composition is 99.3% uranium-238. Only 0.7% uranium-235. But actually, Unnati, which one is the needed for us? Okay, in like as a fuel, which is fissile material? This is fissile material. So we need this one, we need uranium-235. So if its concentration is increased, that is called uranium enrichment, that is rich uranium. Uranium which is rich in the fissile material. Uranium which is rich in the fissile material. I hope it is clear. Okay. If it is having some 3% to 5% like that, it is enriched but low enriched. Is it clear? It is more. We have more fissile material but not very high. Is it clear? 20% is maximum for the reactor. If any company, if any country is enriching the uranium beyond 20% or even up to 10% also, we have to doubt that probably it is trying to develop the nuclear weapons, because we don't need 10%, 20% for the reactors. So for which purpose they will make about 10% uranium enrichment? Okay, so we have to doubt that probably they are trying to develop the weapons. But actually, okay, 90% is weapon grade. I mean minimum 90% for weapons. This is high enriched uranium. Is it clear? And also we call it as weapon grade uranium. Weapon grade uranium means, okay, the uranium in which, okay, you have more than 90%, okay, we have more than 90% fissile material. Is it clear? But this is not maximum, please. This is minimum. 100% is also okay. For weapons, 100% is also okay. Minimum 90% is needed to make an atom bomb. Maximum 20% is for reactor, but we don't even go up to 20%. 3 to 5% is enough for a reactor. Is it clear? But maximum for a reactor is 20%. If a country is enriching beyond some 10%, then you have to doubt the country. Okay. You might have studied in international relations or other subjects the equations between Iran, USA and the P5+1 or E3+3, 5 powerful nations, okay, 3 European nations and 3 other nations. Is it clear? Their bargain with Iran, okay. Iran is a country having rich uranium resources. Is it clear? Also, they have good enrichment technology. So, technologically plus resources wise, by both the concepts, Iran is capable of making weapon grade uranium. I hope it is clear. It's not mean that, okay, it is making the weapon grade uranium, okay, but it is capable of making the weapon grade uranium by 2 reasons. One is technologically, uranium enrichment technology they have. Plus resource wise, they have good resources of uranium. That is the reason why Iran will be in the discussion in this particular uranium enrichment topic. The moment uranium enrichment comes into the context, the first country we remember is Iran. Is it clear? Okay. So JCPOA, okay, all these things that do not come under science and technology, you might have studied in the international relations. If not, you can just go through that once. Is it clear? We will touch the technical aspects of it more. Is it clear? Okay. But I roughly told you what is the concept. Okay. So Iran is enriching the uranium And P5+1, the powerful countries, they do not want Iran to enrich the uranium, okay. So they are imposing some restrictions, okay, and sanctions on commerce, okay, like that some, okay, discussions will be happening between those countries. I hope it is clear, okay. Yes, HRX, okay, uranium-235 must be 90% for weapons, okay. Then can we continue? That is about the uranium enrichment. And how do we do the uranium enrichment? Okay. Uranium enrichment happens by a process called centrifugation. A centrifuge is almost like a mixer grinder. So like if you put these vials in a centrifuge and it will rotate very fast. When it rotates like that, okay, in that the two components 235 and 238, okay, will form two separate, okay, uranium-238 here, uranium-235 here. Based on their density, okay, they get separated into two layers. Then we can separate the two and mix them in the concentration you want. Separate the two components, mix them in the composition you want, okay. Like mix 3% of this and 97% of this, or mix 20% of this and 80% of this, or mix, okay, 90% of this and 10% of it, okay. That is how we do the centrifugation for uranium enrichment. Clear till this point? Okay. Next, okay, after we understand that, the next point is uranium fission chain reaction, okay, nuclear fission chain reaction. Can we continue? Can you see this slide? Okay, so we are now discussing nuclear fission chain reaction, okay. Look at here, this is first uranium-235. To that what is added here? What is this one? This is a slow neutron. Then it is broken down into barium and krypton. Plus remember that it will also release 3 neutrons. Is it clear? I hope you remember the first reaction. Uranium-235, slow neutron, barium, krypton, 3 neutrons. I hope it is clear. This is uranium-235. Slow neutron is added to it. It is broken down into barium, krypton, and 3 neutrons are released. Then what happens? Now you don't have to add the neutrons from external sources. These neutrons which are coming from the first reaction, they will hit the other uranium molecules. Is it clear? Uranium atoms, sorry. Okay. They will hit the other uranium atoms. This is again uranium-235. So, the 3 neutrons, if they hit 3 uranium-235s, next you will get 9 neutrons, isn't it? 1, 3, 9, 27 and so on, like that. I hope it is clear. Okay. So, it means you don't have, for example, how much ever uranium you take, you don't have to take so many neutrons, just add 1 neutron. When you add just 1 neutron, then the uranium-235 breaks into barium-krypton and it will release the 3 neutrons. These 3 neutrons, okay, will now hit 3 uranium-235s. Then they will release 9 neutrons. I hope it is clear. 9 neutrons. The 9 neutrons will hit 9 uranium-235s. Then they will release 27 neutrons. The 27 neutrons will hit 27 uranium-235s. And the reaction goes on like that. That is called as nuclear fission chain reaction. I hope it is clear. Right. Okay. Now let us try to understand how a nuclear reactor works. Okay, let us try to understand how a nuclear reactor works. Okay, different components of a reactor. First component is fuel. What is the fuel? Fissile material like uranium-235 or plutonium-239. What do we use it? Okay, what do we use it? Use as a fuel? Fissile material. Is it clear? What is used as fuel? Fissile material. Next. Okay. What is a coolant? What is a coolant? Obvious point. Okay. To absorb the heat from the reaction, we use water for it. We use water for it. Okay. I hope the tick marked components are clear. What we use it as a coolant? Water. Why? To cool, because you know the fission releases enormous amount of heat. So we definitely need a coolant. Next one. Okay. Containment. Containment means like this. This is called contain. See, look at here. This is called containment box. Okay. So that the nuclear material will not be leaked outside, or the heat generated will not impact the people outside. Is it clear? The nuclear material will not be leaked or the heat will not be released outside. For that reason you need a containment, a concrete, okay, or some construction around that. I hope it is clear. Safety barrier made up of concrete or steel to prevent the heat and the radioactivity exposure. Okay. Right, yes, to prevent radioactivity leakage, also for the heat not to get exposed outside. Containment. I hope the three are very clear. Next one. Okay. Next component is moderator. It will slow down the neutron. It will slow down the neutron. Tell me, what does it slow down? Will it slow down the reaction or it will slow down the neutron? Will it slow down the reaction or will it slow down the neutron? No, Saurabh, it will not slow down the process. It will not slow down the process. It will slow down the neutron. And by slowing down the neutron, it will actually facilitate. It will actually facilitate the fission. Is it clear? It is not slowing down the fission. It will in fact help the fission by providing the slow neutron. Remember, you need a slow neutron for the fissile material to undergo fission. So the moderator will provide that slow neutron so that the fission can occur. Okay. So will the moderator speed up the reaction or slow down the reaction? Yes, everybody. Will the moderator speed up the fission or slow down the fission? Very good, wonderful, super intelligent. Okay. So the moderator will actually speed up. It may not speed up, but actually it will facilitate the fission. Okay. Or facilitate fission. Okay. Next one. Okay. Controller. Okay. Controller. What it will do? Okay. It will absorb the neutrons. It will absorb the neutrons. Okay. Tell me what the controller will do. Controller will absorb the neutrons. Right? Then tell me, for example, if these three neutrons are absorbed, Okay, I mean, okay, 1 neutron I have added, 3 neutrons are released. If they are absorbed, okay, can they trigger the fission of the next 3 uraniums? Will the chain reaction continue? Will the chain reaction continue? No. Is it clear? So it will absorb the neutrons so that, okay, it will either control or even stop the fission. Is it clear? If you remove all the neutrons, okay, complete my sentences, okay, if the controller absorbs all the nutrients here, if the controller absorbs all the nutrients, then the fission will very good. Everybody please. Stop. If we absorb only few neutrons, then the reaction will slow down or stop or speed up or no change. If only some neutrons are absorbed, the reaction will slow down. The reaction will slow down. Okay. So, actually I first completed fuel, coolant, containment because there is no confusion in them. Coolant means obviously water to cool down. Containment is obviously to prevent the leakage of the nuclear material. Fuel is obviously the fissile material. There is no chance of confusion, but most of the students will get confused between this moderator and controller because they sound similar. Be little careful in remembering them. Is it clear? Moderator will actually facilitate the fission, whereas controller will actually stop or slow down the fission. I hope it is clear. Okay. Right. Now let us see the working of this. Okay, look at the working here. Okay, first of all, this is containment. I'll just keep on putting a tick mark to the completed things. Make sure that you understood them. Okay, first one. Okay, containment. Is it clear for everybody? Containment. Then, okay, this is reactor vessel. Is it clear? Actual reactor is there inside the containment. Okay, reactor vessel means where the reaction is occurring. Which reaction? Fission reaction. Reactor vessel is a vessel in which the reaction is occurring. Okay, then in this what you have? Fuel rods you have, that is uranium-235. Okay, fuel rods plus control rods. Why control rods? So that the fission occurs slowly. The fission occurs slowly. Because if the same reaction occurs fast, that is bomb. Did you understand? Atom bomb, it will blast. You want the reaction to occur, but you want it to occur in a controlled manner. That is the nuclear reactor. I hope it is clear. Okay? Yes. Fuel rod will provide the fuel. Control rods will see to it that the fuel will undergo fission in a slow manner. Okay. And then here it is condenser. Condenser will condense the steam. That is the reason why this blue color thing is water. The blue color thing is water. Okay. Or water means H2O. Or even we can also use heavy water also. Heavy water means D2O, deuterium 2O. Okay. What are these? Like hydrogen, deuterium, tritium. Okay. These are 3 isotopes of hydrogen. Is it clear? They all have just one proton, but the number of neutrons is different. Is it clear? They are heavy isotope. Deuterium is a heavy isotope. Of hydrogen. Is it clear? Okay. So, either water or heavy water. Okay. Now, complete focus here. So, water comes into the reaction vessel. Step number 1, water or heavy water comes into the reaction vessel. Here the reaction is occurring, fission is occurring in a controlled manner. Fission releases enormous amount of heat. So the water gets heated up and the water gets converted into steam. This light blue color thing is steam. I hope it is clear, steam. Now the steam will rotate, the fifth step is the steam will rotate the turbine. Is it clear? Okay. So the steam will rotate the turbine. So it means this will get converted into a kind of kinetic energy. So generator will convert that kinetic energy into electrical energy. That electricity will be supplied to different villages, cities, factories, wherever you want to electrify. Okay, I repeat the steps once again. Step 1, water enters into the reaction vessel. Step 2, in the reactor vessel, the fission reaction is occurring in a controlled manner because we have controllers. This fission reaction will heat up the water, third step, and convert it into steam, because of this heat it will get converted into the steam. Fourth step, it will rotate the turbine, fourth step. That kinetic energy will be converted into electrical energy, generator will convert like that, that is fifth step. Sixth step, the generated electricity will be supplied to different factories or villages, towns, cities, etc. Okay. So this is how a reactor works. I hope it is clear for all of you. Okay. And then, we have types of reactors here. We have types of reactors here. Okay. Types of reactors, first one, boiling water reactor. Okay. And here we have to use enriched uranium as the fuel. Coolant is water, moderator is also water. Next one, pressurized water reactor. Okay. Now listen carefully. What is the advantage of a pressurizer? What is the advantage of a pressurizer? Can you all tell me what is the relation between pressure and boiling point? As the pressure increases, what happens to boiling point of water? Everybody? Increases. I mean, they both are directly proportional. Okay. So, in a pressurized heavy water reactor, the components are as it is. Just an additional component will be there. That is pressurizer. Okay. What is the difference between boiling water reactor and pressurized water reactor? Okay. The difference is just one component, that is, it will have a pressurizer. That will increase the pressure. I hope it is clear. That's all. That is the only component, as it is, same mechanism. Just it will have another instrument called as pressurizer. Where? Somewhere here it will have an additional instrument called as pressurizer. Okay. Then what happens? Boiling point increases. Okay. What do you mean by boiling point increase? So here what happens is water boils at some 1000 degrees Celsius, something like that. Normally water boils at what temperature? 100 degrees Celsius. But due to in the pressure cooker, water boils at some 121 degrees Celsius. But in this reactor, the water will start boiling at a very high temperature. Okay, 400 degrees centigrade water, not boiling. Is it clear? It will not start boiling because pressurizer is used. So because of this, what happens? We get very high temperature steam. Is that clear? We get very high temperature steam. High temperature steam will rotate the, fastly rotate the turbine. Because of that, what happens? You can generate more electricity. That's it. That is the advantage of pressurized water reactor. Okay. Here also we have to use enriched uranium. Coolant, moderator, both are water. Okay. Next one. Pressurized heavy water reactor. Okay. Pressurizer as it is. But instead of water, you are using heavy water in this. Is it clear? That is called pressurized heavy water reactor. Pressurized heavy water reactors are more efficient reactors. Pressurized heavy water reactors are more efficient reactors. So that is the reason why they do not even need enriched uranium. Even the natural uranium is enough for those reactors. Is it clear? Their efficiency is more, so they can use even the natural uranium. For example, you cannot enrich, you don't have a uranium enrichment facility, you cannot enrich, it's okay, even the natural uranium works in these particular reactors because we use heavy water. How can we define a pressurized heavy water reactor? A reactor in which both coolant and moderator is heavy water is called pressurized heavy water reactor. Is it clear? A reactor in which coolant is also heavy water. Moderator is also heavy water. What is moderator? It is used to slow down the, yes, complete my sentence. Moderator is used to slow down the neutron or slow down the fission. Moderator is used to slow down the neutron or slow down the fission. Yes, slow down the neutron. Very good. Thank you so much. Okay, next. So we have seen three types of the reactors. Now we have to see, okay, the fourth type of the reactor, that is fast breeder reactor. Is it clear? We understood three types of the reactors: boiling water reactor, normal reactor, the one that we have seen here. To this, if I add a pressurizer, that is pressurized water reactor. In this, if I add D2O, that is pressurized heavy water reactor. But the reactor mechanism is roughly the same. I hope it is clear. But fast breeder reactor will be totally different. Okay, fast breeder reactor is totally different. Now let us see what is fast breeder reactor. Can we continue? But I'll just summarize this. The basic reactor, this reactor plus pressurizer, pressurized water reactor, this reactor, okay, plus heavy water here instead of water, that is pressurized heavy water reactor. Three types over. Now the fourth type, okay, fast breeder reactor. Let us try to understand what is this. Okay. In the fast breeder reactor, we use what is the meaning of this word fast? Fast refers to fast neutron. Is it clear? We do not add a slow neutron here. We add a fast neutron here. Is it clear? Okay. Next. In the fast breeder reactor, the word breeder means, okay, this reactor breeds. Breeds means what? Produces. Okay. Produces what? Fissile material from fertile material. Understood? Okay. Breeder. Why are you calling this as breeder? Breeding means producing something. What is it producing? Fissile material. From where it is producing fissile material? Fertile material. So that is the meaning of the word breeder. But how the fertile material produces fissile material? By adding a fast neutron. So fast breeder reactor. A reactor in which we use fast neutron to breed the fissile material from the fertile material. A reactor in which we breed the fissile material from the fertile material by adding a fast neutron. That is called as fast breeder reactor. I hope it is clear for all of you. Okay. Then reactors, those produce more fissile fuel than they consume. You add little fissile fuel, but they will produce more fissile material from the fertile material. Done? Clear? Okay. While generating energy using the fast neutrons. Now, okay, I hope the first point is clear. Now focus on the second point. Complete focus here, please. Complete focus here. We use, okay, in this we cannot use water or heavy water. Cannot be used. Why? Because they are strong moderators. They will slow down the neutrons. But we need fast neutrons. Is it clear? They will actually slow down the neutrons. But we should not slow down the neutrons. So it means, can I use water or heavy water in a fast breeder reactor? Everybody? Just keep a rule that you have to answer each and every question of mine so that your focus remains in the class. Okay? Can we use water or heavy water? No. Why? They will slow down the neutron. But we should not slow down the neutron. We want fast neutron here. Okay. So, we cannot use water, heavy water. But we need a coolant. Do you agree? We cannot use water or heavy water. But we need coolant. But as a coolant also I should not use water. Is it clear? Because if I take water, how does the material know whether it is to cool down or whether it is to slow down the reaction? It will do both of them, right? So even as a coolant also I should not use water. No water in this reactor. Understood? Right? As a moderator also we should not use water, Gaurav. Because we don't need mod. What is moderator's role? Slow down the neutron. Okay. But we need fast neutron here. So no moderator here. I mean no moderator. We should not use moderator. Is it clear? Gaurav, I hope it is clear. Because we need fast neutrons. We need fast neutron. Okay. Moderator is not needed. But coolant is needed, right? So as a coolant we have to use, we have to use a very weak moderator. Is it clear? The material we use as a coolant should not slow down the neutron. I hope the concept is clear. The material we use as a coolant should not slow down the neutron. So, we use liquid sodium instead of water to absorb the heat. We use liquid sodium instead of water to absorb the heat. Because liquid sodium will cool down the reaction mixture but will not slow down the neutron. Is it clear? Because it is a very weak moderator. Next. Now, here I will explain through this image. Look at here. I will put a tick mark to each component, confirm whether the tick marked point is clear. Okay. First one, this is biological shielding, I mean containment. I hope that is clear. Okay, outermost containment. Next one. Okay, the red color ones you can see here, 1, 2, 3. Can you see this red color rods? They are fissile core. Fissile core, that is clear. Tick marked points must be clear. Carefully look at the diagram. Fissile core. Then you have this black color rods here, these ones, black colored rods. They are control rods. Is it clear? Control rods means control rods will slow down the reaction or slow down the neutrons. Control rods will slow down the reaction or slow down the neutrons. Very good. Okay, control rods will slow down the fission. Why to slow down the fission? In the fast breeder reactor, reaction is not fast, neutron is fast, that's it. If the reaction occur fast, that is atom bomb, that is not fast breeder reactor. I hope you understood my point. Okay, you should not slow down the neutron because the neutron should, okay, hit the fertile material, but the reaction should occur in a controlled manner. So control rods, I hope it is clear. Okay. Now, next, blue color, what you can see here in this blue color, what is this one? This is breeder blanket, breeder blanket. In the breeder blanket, what we keep is, okay, something like, for example, you keep uranium-238. Is it clear? We keep our thorium-232. That is some fertile material. Okay. We keep. Okay. Because fertile material is a breeder, isn't it? Because it will breed the fissile material. It is a breeder. Fertile material is a breeder. It will breed the fissile material. I hope it is clear. Done? Okay. So I hope this is also clear. Okay. Then now what happens? Complete focus here, please. Okay. Complete focus here. Now, tell me what is there in the red color rods? Very fast. Red color rods, fissile material. Black color rods, they will help the fission reaction to occur in a slow manner. Now, okay, I mean fissile core, you have uranium-230, okay, 5 for example. Red color, uranium-235. Blue color, uranium-238. Now the fission will create neutrons, these ones, the 3 neutrons which will come out. The fission creates 3 neutrons. Yes, everybody? Okay. Those neutrons, okay, fast neutrons from fission will hit the breeder blanket. I hope it is clear. Right? The fission releases 3 neutrons. The fission releases 3 neutrons. They are fast neutrons. Why are they fast neutrons? Because there is no moderator used here. Plus they are coming out of a fission reaction. So they come super fast outside with high energy because fission releases enormous amount of energy. So with high energy, fast neutrons are coming out. I hope it is clear. Okay. What will come out of it? Fast neutrons. From the fission, they will hit the breeder blanket. I hope it is clear. Okay. Once the fast neutron hit the breeder blanket, what is there in the breeder blanket? Uranium-238. Then it will form plutonium-239, fissile material. So it breeded the fissile material, which we can further use as a fuel. This is a fast breeder reactor. I hope it is clear. Fast breeder reactor completed. Okay. The word fast is for fast neutron. The word breeder is for the fertile material which will breed the fissile material. Here first we used the containment. Okay, I am just summarizing it. First we use the containment. And then I have given you the fissile core, that is uranium-235. And then control rods. Okay. The fissile material will undergo fission. This fissile material will undergo, okay, fission. The fission releases fast neutrons. The fission releases fast neutrons. These fast neutrons will hit the breeder blanket. The breeder blanket will contain uranium-238 that will convert into plutonium-239. Now you got more fissile material so that the reaction will continue. This is called as a fast breeder reactor. I hope it is clear. Right. Now we understood all the 4 types of reactors. Now we have to understand India's 3-stage nuclear power program. India's 3-stage nuclear power program. You tell me, in geography classes you might have studied, okay, in the natural resources and all. Do India have rich resource of uranium? Do we have rich resource of uranium? Yes, everybody? No, we do not have much uranium. Very good. What about thorium? Do we have good amount of thorium in India? Comparatively, yes. Comparatively, yes. Okay. So, in India, we have uranium less, but thorium-232 is more. We have more thorium, but the problem is, okay, yes, very good. Okay, we have more thorium, but the problem is thorium is fertile. Is it clear? It is not a very active fuel, not an active fuel. So, we must convert thorium-232 to uranium-233. So, we need a fast breeder reactor. Is it clear? Why India needs a fast breeder reactor? Because we cannot depend on uranium. We do not have rich resources of uranium. Okay, we can import, but it depends on so many international relations. Is it clear? Always there will be some sanctions, there will be different equations between the countries. There is no guarantee that we can always import the uranium. Moreover, when we use imported uranium, there will be more restrictions on its usage. Is it clear? When we use uranium, imported uranium, there will be more restrictions on the usage. I hope it is clear. Okay. So, therefore, okay, for Atmanirbhar Bharat, okay, if we have to be self-sufficient, self-reliant, we cannot depend on uranium. We have to target using the thorium. But thorium problem is it is fertile. So, we have to breed fissile material from that. For that we need a fast breeder reactor. I hope it is clear. Okay. Right. Okay. So, what is in the stage 1? Okay. Stage 1 is pressurized heavy water reactor. I hope you all remember what is pressurized heavy water reactor, isn't it? Stage 1 is pressurized heavy water reactor. And they are operational since 1973. They are in the commercial stage. They are in the commercial stage. and they produce 12 gigawatts of electricity. And once you establish this reactors, they will continuously work for 30 years. Is it clear? Once you establish this reactor, they will continuously work for 30 years. Done? Next. I already told you that pressurized heavy water reactors are very efficient. So they work even with a natural uranium also. I hope you remember this point. You don't have to use enriched uranium. You don't have enrichment facilities. Still no problem. Okay. Natural uranium is also enough for this. Okay. Pressurized heavy water reactor. Natural uranium. Okay. Then uranium-235 undergo fission, produce heat, and uranium-238 will produce plutonium. Okay, means here little carefully please, complete focus on the class. What is natural uranium? Uranium-235, okay, and uranium-238. What will happen to 235? It will undergo fission. What will happen to uranium-238? It will take up the neutron and will become plutonium-239. Plutonium-239. Right? Okay. I hope the first box is completely clear. First box, stage 1 pressurized heavy water reactor, commercial stage, this much electricity it will produce. We are taking natural uranium, and natural uranium will have two components. 235 will undergo fission, but 238 will take up the neutron and will become plutonium-239. Is it clear? Okay. Now focus on the second box please, complete focus. Okay. Into the second box, what is going from the first box? Plutonium-239, isn't it? What is the source? If you remember uranium-238. Uranium-238 formed this plutonium-239. Is that clear? Okay. Next. Next what you have look at here? Depleted uranium. Okay, what do you mean by depleted uranium? Okay, I'll tell you this. Okay. Earlier I said that this is natural uranium, 99.3%, 0.7. Above, okay, uranium-235 more than 0.7% is, okay, enriched uranium, isn't it? That is enriched uranium. Okay. And opposite, if uranium-235 is less than 0.7%, that is called depleted uranium. I hope it is clear. If uranium-235 is more than 0.7%, enriched uranium. Okay, less than the natural uranium, then it is called as depleted uranium. Is it clear? Its quality will be less than the natural uranium. Is it clear? Not low enriched, Antara. Okay, not low enriched. Okay, fine. Answer these questions. Okay. Okay, answer these questions all of you, particularly Antara, answer this. Uranium-235, uranium-238. Okay, 0.7, 99.3. What is this uranium? Antara and everybody. This is natural uranium. Okay, 3%. Okay, and 97%. This is low enriched uranium. Low enriched uranium. Okay, and some okay, 30%, 70%. This is high enriched uranium. 90%, 10%. This is weapon grade uranium. But, okay, 0.1%. Is it clear? Okay, 99.9%. That is not low enriched, dear. That is depleted uranium. I hope it is clear. That is not low enriched means more than natural uranium. Okay, enriched but less enriched. This is not enriched, not even normal, less than normal, depleted uranium. I hope it is clear. Very good. Okay. So, I hope depleted uranium is clear. So, here depleted uranium is also coming. From where the depleted uranium is coming? If you observe, this uranium-235 is already undergoing fission here. Is it clear? Because, okay, because of that, the uranium-235 content decreases. Is it clear? Because this already used in stage 1 for fission. Therefore, depleted uranium is coming into stage 2. Right? There are two components going from stage 1 to stage 2. First one, okay, plutonium. What is the source of plutonium? All of you tell me. From stage 1 into the stage 2, plutonium is going. What is the source? Everybody, what is the source for it? From where it is coming? Very first, okay, Tanno. In the first one only, from where it is coming? Abhishek, very creative, uranium, but I want specific answer. 235 or 238? Plutonium is coming from uranium-238. Is it clear? Okay. Mukesh, it is coming from stage 1. But from where it is coming? From the uranium-238. Okay. From the stage 1 to the stage 2, depleted uranium is also coming. From where that is coming? That is coming from this uranium-235. Because in the stage 1, uranium-235, some amount of it. Originally you have 0.7%, isn't it? Originally you have 0.7%. But some amount is used in the stage 1. So, less than 0.7% is going into the second stage. So, depleted uranium. These two are going from the stage 1 to stage 2. Number 1, number 2. Is it clear? And thorium, it is not from stage 1, it is coming from outside. We are giving from outside. Is it clear? Thorium we are giving from outside. Right. Okay. Then what is stage 2? It is a fast breeder reactor. Okay. Operational since 1985 but not in the commercial stage. This is only in the technology demonstration stage. I mean to show off the technology. Is it clear? We show it to the for demonstration, technology demonstration to prove that it is working. Is it clear? To prove that it is working. Okay? And it will generate 300 gigawatts of energy for 30 years. Is that clear? Next one. From stage 2, I hope the second box is completely clear. Second box is completely clear. Into the second box, 3 things are going. Plutonium-239, that is coming from this uranium-238. Depleted uranium, that is coming from this uranium-235, which is only 0.7%. In that also some amount is used in the stage 1. And thorium, which is coming from outside, we are adding into this reactor. And this thorium is nothing but we are using this thorium as the breeder. What is the breeder here? Thorium is the breeder. Is it clear? Okay. Not only, there are two breeders here. One is thorium-232 plus uranium-238 also. We use both of them as breeders. Understood? Why do we not use it commercially? Abhishek, simple. We still did not go to that level. We just developed the technique. We are just testing whether it will work or not. After first stage, developmental stage. Second stage, technology demonstration stage. That means development is completed. We have the reactor ready. We are just checking whether it will work or not. Testing phase. Next phase, commercial operation. I hope it is clear, Abhishek. Okay. So, in the second stage, what is used in the breeder blanket? Everybody, in the second stage, what is used in the breeder blanket? Thorium-232 from outside, uranium-238 from stage 1. This uranium-238 from stage 1. I hope it is clear. Right. Because uranium-238 is also breeder material. Then what is the third stage? Third stage is advanced fast breeder. This is also fast breeder only. Third stage is also fast breeder, but more advanced. It will produce more energy, and once you set up, it will continuously work for 500 years. Is it clear? Once you establish, it will continuously work for 500 years. Okay? Right. So, this is still in the technology development stage, not even in the technology demonstration stage. Okay. This primarily depends on thorium. This primarily depends on thorium, which is our final target. That is the reason why our nuclear power program is like this, with the final target of depending on thorium, not uranium. Clear? Right. Now, so is India's 3-stage nuclear program clear for all of you? Yes. Now, okay, the types of reactors completed, nuclear program completed. Okay. Now, next one is small modular reactors. This is a recent topic. Small modular reactors. Okay. The word small, because they are physically only a fraction of size of a conventional reactor. I mean, they are much smaller than a regular reactor. Is it clear? They are much smaller than a regular reactor. Next one, they are modular. Modular means what? Okay, they are factory assembled and directly transported as one unit and installed in your location. Is it clear? They are assembled in the factory, and directly they will come and install it in your location. I mean, they will not manufacture at the location. Okay, manufactured in the factory, directly assembled, installed in your location. That is called modular. Is it clear? Okay. Right. Saurabh, by chance if you get more doubts like this, what you can do is, this is for all of you, just look at this particular diagram more carefully and you think you will get it. For example, you will just think like this: plutonium, from where it is coming? From the uranium-238. Okay, depleted uranium, from where it must come? From this. Thorium, it's not coming from stage 1, so must be coming from outside. Is there anything else used? Check the arrow marks. Anything else used? No, that's all. Like that, if you just carefully observe this diagram, all your doubts will be clarified. Okay? Right. I tried to be as clear as possible, but still. Next. Reactor, because it is harnessing the, harnessing the nuclear fission heat to produce electricity. I mean, heat energy converted into electrical energy. Therefore, it is a reactor. Okay, the 3 words justified: small, small in size; modular, directly installed in your place, pre-assembled unit directly installed. Okay, reactor, because it will generate electricity. So, small modular reactor. What are the advantages of it? Is it clear? Okay. Right. Advantages of small modular reactor. It is prefabricated, as I told you, factory made and directly installed in your place. They can be deployed incrementally to match the increasing demand. For example, I got one small modular reactor, but now my usage increased. So I can add one more unit to it. Next, my usage further increased. Then I will add one more unit to it, like that. Is it clear? Then you have micro reactors. Okay, micro reactors are a subset of the small modular reactors. I mean, within the small modular reactors also, okay, if it is very small, okay, only 10 megawatts of electricity, that is called as a micro reactor. I hope it is clear. Micro reactor. Okay, see, large conventional reactor 700 plus megawatts of electricity. Small modular reactor 300 megawatts of electricity. Micro reactor only 10 megawatts of electricity. That is the difference between the three. Done? Next. What are the other advantages? Auto turn off option. For example, any earthquake comes or any like unwanted incident happens, you don't have to go to the reactor and switch it off. There is auto turn off option. Okay, so that the heat or the radioactivity will not be leaked outside. Is it clear? The power plants based on SMRs may require less frequent refueling. Okay, the refueling we have to do less frequently. We don't have to frequently refuel them. Is it clear? For India, which aims to achieve net zero emissions by 2070, SMRs are very important. Which net zero emissions we are speaking about? We have a target of reaching net zero carbon emissions. Is it clear? We have to reduce the carbon emissions. Means we have to reduce the fossil fuel consumption. How to reduce fossil fuel consumption? We have to increase the alternative fuels. Okay, so one such thing is nuclear reactor. So SMRs are very important. And what are the limitations of SMRs? They are still experimental stage. Many SMR designs require the use of high assay low enriched uranium. Okay, what do you mean this? It is defined as uranium enriched to greater than 5%, less than 20. 5 to 20% is called as HALEU. High assay low enriched uranium. Definition is, okay, above 5% but less than 20% uranium-235 you have. Okay, so that is called as HALEU, HALEU. Okay, the low grade uranium as nuclear fuel will need to be refueled more often, which is not desirable. Is it clear? Okay, these are the limitations. And nuclear energy is the 5th largest source of electricity in India. Okay, and 3% of our total electricity is coming from the nuclear reactors. Is it clear? Nuclear energy is 5th largest source of electricity in India. Okay, hydroelectricity we have, okay, like that, okay, thermal electricity, like that. In that 5th position is nuclear electricity. Is it clear? And 3% of the total electricity is coming in the country. Is it less or more? It is very less. We are only 3% of our total electricity is coming from the nuclear reactors. Same if you take Israel, it is like up to 50% electricity is coming from the reactors. Is that good? No, that is dangerous. Okay, because if we keep so many reactors, in the situations of any war, nuclear war is not needed. A conventional explosive which falls on the nuclear facility also will turn out to be as dangerous as nuclear weapon. I hope it is clear. So, up to 50% nuclear electricity is risky, but 3% total electricity is from nuclear is too less. So, we have to increase but not up to 50%. That is the situation of India. I hope it is clear. Okay. So India has over 25 operating nuclear reactors in 8 power plants producing total this much amount of electricity. Is it clear? Currently we have 25 operating reactors in 8 power plants. Okay. Most of them are pressurized heavy water reactors and few of them are light water reactors. Remember, we do not have a fast breeder reactor in India. We do not have an operating, we have fast breeder reactors, but in the testing condition. We do not have an operating fast breeder reactors in India. Most of our reactors are pressurized heavy water reactors. Some of them are light water reactors. I hope it is clear. Okay. Next, expansion of nuclear installation. This is just some factual content. I think you can go through that. Okay. India's indigenous move. Can we continue? The world's first thorium, India's indigenous move means thorium. I hope it is, because we cannot depend on uranium. Done? Okay. World's first thorium based prototype. Prototype means what? Model. Okay. Not in the working condition. Is it clear? Okay. You may get confused. You just said that we do not have a fast breeder reactor. Thorium based means fast breeder reactor only we have now. Yes, we have. But it is prototype. Fast breeder reactor. It is called Bhavni. Okay. Using uranium-233. Then you may get confused. You said thorium and again you are saying uranium-233. Yes. Thorium-232 is fertile material. To this, if I add a fast neutron, then it will undergo transmutation into uranium-233. So, uranium-233 means thorium based only. I hope it is clear. Uranium-233 means thorium based. Uranium will have only 235 and 238. We don't have uranium-233 in the uranium ore. I hope it is clear. Do we have uranium-233 in the uranium ore? Do we have uranium-233 in the uranium ore? No, uranium-233 comes from thorium-232 by transmutation. It is being set up at Kalpakam in Tamil Nadu. This plant will be entirely indigenous and will be the first of its kind. The experimental thorium based reactor Kamini already exists in Kalpakam. Okay, they both are fast breeder reactors. Is it clear? Use of large thorium reserves to produce nuclear fuel is a key strategy of India's nuclear energy program. What is the key strategy of India's nuclear energy program? Okay, using thorium. I hope it is clear. Next, with this, we completed everything about nuclear fission. So now we are discussing about nuclear fusion. Nuclear fusion is opposite to fission. Two light atomic nuclei will fuse with each other to form a heavy atomic nucleus. That is called as nuclear fusion. I hope it is clear. Okay. Two light atomic nuclei fuse to form one heavy atomic nucleus. Right? Okay. What are they? Deuterium. Is it clear? With one neutron, okay, and one proton. Tritium with one proton, two neutrons. Is it clear? Okay. Then these two will form helium. Helium will have two protons plus two neutrons. Okay, but here you have one extra neutron which is released. One neutron is released. This is fusion reaction. I hope everybody understood fusion reaction. Deuterium plus tritium will combine to form helium plus 1 neutron released. Okay. Then, what are the advantages of it? First advantage, 4 times more energy compared to fission. It gives 4 times more energy compared to fission. Second advantage, unlimited fuel, because in the atmosphere you have enough hydrogen. You don't have to import hydrogen from some country. You don't have to mine it. I hope it is clear. Okay. So, that is second advantage. Third advantage is it is 100% clean. No radioactivity, isn't it? Only helium is the product. Is it clear? Only helium is the byproduct. 100% clean. Okay. So 100% clean, unlimited energy. If you can use this particular reaction in the reactor, okay, then that will give you 100% clean, unlimited, no pollution, no completion of fossil fuels. Okay. So that is the advantage of nuclear fusion. I hope it is clear. Okay, then actually this fusion reaction, one, it occurs in the stars. Second, it occurs in the hydrogen bomb. I told you in atom bomb, okay, the principle is fission. But in the hydrogen bomb, the principle is fusion. I hope it is clear. In atom bomb, the reaction is, the point is fission, and in the hydrogen bomb the principle is fusion. But actually, okay, this is very difficult because you all know that like charges repel each other, isn't it? You all know that the like charges repel each other, and the nucleus are positively charged, isn't this is positively charged, this is positively charged. Okay. So therefore, okay, therefore it is very difficult to fuse the two neutrons. They will not fuse spontaneously. Do you agree? The two nuclei are positively charged and like charges repel each other. So the two nuclei do not spontaneously fuse. Even if you try to fuse them, it is very difficult. Is it clear? Okay. Then, if the fusion reaction is so difficult, how it is occurring in the stars? It occurs due to the gravitational force. I hope it is clear. Star will have enormous amount of gravitational force. That gravitational force will push the nuclei together so that they will fuse. Is that clear? Stars will have enormous amount of mass. So, okay, enormous amount of gravitational force. The gravitational force will push the two nuclei together so that they can fuse. Okay. Whereas in the hydrogen bomb, you know, first we blast an atom bomb. That will release heat. This heat energy is used, okay, to trigger fusion. Then the hydrogen bomb will blast. Do you know this? We need an atom bomb to blast the hydrogen bomb. Is it clear? First, fission. The fission releases enormous amount of energy. That energy is used to overcome this repulsion force. So, in the hydrogen bomb, it's okay, we can use atom bomb. In the star, it's okay, gravitation is there. But how to achieve fusion in the reactor is the real challenge. But if you can achieve it, 100% clean energy. Is it clear? If you can achieve it, 100% clean energy. I hope the problem is clear. In the star gravitational force, in the hydrogen bomb, atom bombs. But how to achieve this fusion in the reactors? From where do you get that much energy to force the two nuclei to fuse? So that is the problem in the fusion reactors. Okay. Then now let us see what is a fusion reactor. ITER, International Thermonuclear Experimental Reactor. Can we proceed? Okay. ITER is an experimental fusion reactor facility at Cadarache in South France. Okay. ITER is an experimental fusion reactor. And partner countries are European Union, China, India, Japan, South Korea, Russia, US, America. But European Union is the host party. So 45% contribution is by European Union. Remaining countries have 9% share each. Is that clear? Remaining countries have 9% share each. What is the role of European Union? Providing the site and operating the reactor. Is it assembling the reactor, operating the reactor? Everything will be taken care by European Union. Okay. What is the role of the other countries? The role of other countries is to provide the components. For example, India delivered cryostat. What is cryostat? Coolant. It is a fusion reactor. It generates more heat when compared to a fission reactor. So water is not sufficient as a coolant. So we have a special coolant called cryostat. Okay. India will supply cryostat. Like that only other countries also will supply other components. That is the role of the other countries. I hope it is clear. And assembling, operations, providing the site, everything will be taken care by European Union. India signed the ITER agreement in 2006. ITER has been designed to produce 500 megawatts. Okay. Now look at this. It is designed to produce 500 megawatts of output power, 50 megawatts of input power. Is it clear? Why do we use, can you tell me why do we use this 50 megawatts of input power? To force the nuclei to fuse. Did you understand? And this is called as activation energy. This is called activation energy. Yes. Okay. 50 megawatts of input energy. Why are you giving 50 megawatts of energy? To overcome the repulsion between the two nuclei and to force them to fuse. We need this input energy. We call it as activation energy. But once if they fuse, they will release 500 megawatts of energy. Means it should be exergonic. Exergonic means it release more energy, okay, than the energy consumed. Such reactions are called as exergonic reactions. I hope it is clear. Next. But this is an experimental one. If this is successful, later a 2000 megawatt reactor is proposed. I mean, if this ITER becomes successful, okay, somewhere in the year 2035 or so, then they will start working for a 2000 megawatt actual reactor. This 500 megawatt is just an international thermonuclear, okay, experimental reactor. Is it clear? The 500 megawatt one is an experimental reactor. Okay. See, That's it. This is ITER, looks like this. It looks like a donut or South Indian vada donut, like that it looks. I hope it is clear. Okay. And what is this? This is deuterium plus tritium in the form of, okay, in the form of plasma. That is, yes, we also call this as a tokamak reactor. Okay, that is given here. We also call this as tokamak reactor. I think the word I did not use, but yes, that is called tokamak reactor. Right? Okay, we call this as tokamak reactor. Okay, ITER, it is in the donut shape. This bright yellow color circle is deuterium plus tritium in the form of plasma. Okay, what is plasma? It is the fourth state of matter. It is the fourth state of matter, and it is super hot, very hot. Okay, that heat energy will, this heat energy is used to overcome repulsion. I hope it is clear. This is deuterium plus tritium plasma. This is where the deuterium and tritium will fuse with each other. But why should we maintain deuterium, tritium in plasma form only? Why don't you maintain deuterium and tritium in solid form or liquid form or gaseous form? Okay, we want enormous amount of heat energy to overcome this repulsion. So we are taking the deuterium and tritium in the plasma form. Is it clear? Not in solid state, not in liquid, not in gaseous state, but we use it in plasma state. I hope it is clear. Yes. And okay, this particular plasma is always kept in motion. It is always kept in motion so that it will never touch the containment. If it touch the containment, blast. Is it clear? Okay, it is always kept in motion in a ring-like structure so that it will never, if you observe, it is not touching the walls of the containment. Is it clear? Right. Okay, next. Check this question. India is an important member. Important or not, I don't know, but a member of ITER. Is it clear? If this experiment becomes successful, it is still in the assembly stage itself. Okay. If the experiment succeeds, what is the immediate advantage for India? It can build fusion reactors for power generation. Is it clear? Okay. It will build, very obvious answer. Thankfully, in science and technology, what happens is, I will advise all of you to take this subject very, very seriously. Okay, because actually around 15 questions are coming from science and technology, which is equal to some geography, history or environment or any other subject. When you spend one complete year or more than that on those subjects, that much importance you have to give to science and technology also. Because many times the prelims score, we will get marks 1 or 2 marks less than the cutoff, isn't it? Several times it happened with my students. Okay, very good students, they can write beautiful answers in the mains, but lost prelims by 1 or 2 marks. If that should not happen, you have to excel each subject. So you have to take science and technology as serious as every other subject. And also the questions are quite simple in science and technology. Is it clear? Right? Okay. And so now, okay, ITER, okay, that is in France. Now, other fusion research in the world. Okay, US, okay, National Ignition Facility, Joint European Torus, China's artificial sun. You might have heard about China's artificial sun and you might have thought that it is like some sun globe shape, not like that. That is also fusion reactor. Why the fusion reactor is called artificial sun? Because what is the reaction occurring in the sun? Fusion. What is the reaction occurring in tokamak reactor? Okay, fusion. So tokamak reactor is nothing but artificial sun. I hope it is clear. Artificial sun is nothing but a tokamak or a fusion reactor. South Korea's K-star, all these are the research on nuclear fusion, similar to ITER, but by the other countries. Is it clear? United States National Ignition Facility, Joint European Torus, China's artificial sun, South Korea's K-star reactor, all these are the research going on in the fusion. The benefits of nuclear energy. Now, not fusion. Fusion benefits I told you, unlimited amount of hydrogen, 100% clean energy, more energy per unit amount of the fuel. Those are the advantages. These are the benefits of nuclear energy. I mean fission, fusion, both together. Is it clear? Fission, fusion, both together. Reliability, okay, low cost, long term usage. You have seen that once a reactor is established, it will work for 30 years, 50 years, 500 years, like that. Long term usage. No greenhouse gas emissions. Then you can argue that, okay madam, there are no greenhouse gases, but radioactivity leakage problem will be there. Yes, will be there. But why do we speak about the problems here? For problems, I have given the next slide. Issues with nuclear energy. Next slide, issues. Here advantages, no greenhouse gases. Is it clear? Next, huge potential compared to other forms like wind energy, solar energy, or biofuels. When compared to them, here you can generate much more amount of electricity. I hope it is clear. Issues, cost. Then you may say, you said less costly here, and you are saying in the problems you are saying more costly here. Which is correct? Both are correct. It is very costly to set up a reactor. But once you set up a reactor, it will work for 500 years now. Then the per generation of the cost per generation of 1 unit electricity will be less. Is it clear? Costly to set up the reactor, but once the reactor is ready, per unit cost of electricity will be less. So both are correct. Safety issues, accidents, earthquakes, wars. Okay, see presently there are kind of news regarding the leakage of radioactivity in Pakistan. Our BrahMos hit the air base there, okay, near the nuclear facility, and a flight from US went there. This is all we are following in the news. Such accidents are possible. Is it clear? Same way Chernobyl incident, Fukushima incident and all those things. Is it clear? Next, nuclear waste management. That is, how do you dispose this nuclear waste? Health, okay, nuclear leakage, okay. They say that Madame Curie used notebook. That notebook is still releasing radioactivity. Is it clear? Okay. And Hiroshima, Nagasaki, still the people are suffering from cancers and the consequences of it. So that is the reason why like we should always try to solve the things without war, because a nuclear war is not a joke. Sitting in our room comfortably under air conditioning, we can say yes, you have to fight and all, but decades, generations should suffer if a nuclear attack occurs. Is it clear? So nuclear is not a joke. Is it clear? Okay, right. Nuclear material, and I think all the countries should definitely go for non-discriminatory disarmament. I mean, you don't use the wars, nuclear weapons, I will not use the nuclear weapons. All the countries should jointly agree not to use the nuclear weapons at all, because that is unimaginable loss. Is it clear? Okay. Right, nuclear material like plutonium can be used to produce highly destructive nuclear weapons. Terror threats are possible. These are the problems in this. Lastly, nuclear policy of India. Okay, the primary objective is to achieve economic, political, social, scientific and technological development within the democratic framework. Building and maintaining credible minimum deterrence. I mean, we will cause minimum harm through our nuclear facilities or nuclear weapons. Minimum deterrence. This is first policy. Is it clear? Yes, coming to that. No first use posture. That is the reason why we are facing the nuclear blackmails also. Is it clear? Okay, because we have a policy of not using the nuclear weapons first. Okay. It means nuclear weapons to be used only in retaliation against a nuclear attack on Indian territory or Indian forces anywhere. Is it clear? Okay, but Pakistan do not have no first use posture. So we should remember that. Next, nuclear retaliation to the first effect will be massive. But what is the advantage in massive retaliation? Okay, when generations will suffer from cancers, tumors and decades, like it will take few decades for a small plant to come out after the nuclear war. Is it clear? Okay, so nuclear attack should not be there. Massive retaliation should not be there between any two countries. Non-discriminatory disarmament is very, very important. Okay, there is no use of massive retaliation. Is it clear? Okay, then so okay, and designed to inflict unacceptable damage. Okay, the nuclear retaliatory attacks to be authorized only by the civilian, I mean they are not battlefield weapons. We have two types of weapons: tactical weapons, strategic weapons. These are strategic weapons. Civilian political leaders should take a decision of using it. Okay, and non-use of nuclear weapons on non-nuclear weapon states. I mean, okay, we don't use nuclear weapons on the states that do not have nuclear weapons. I mean, okay, we will be very much justified. I mean, when they cannot, okay, with only an equivalent we will use the nuclear weapons. Then when they don't have nuclear weapons, we will not use. Is it clear? Okay, then continuance of strict controls on export of nuclear material, because we don't use nuclear weapons, okay, but if we export the nuclear material to other country, if they use nuclear weapons, then we are indirectly responsible. So we have strict controls on even exports also. Is it clear? So directly or indirectly, we don't want to cause any harm to the world. That is the greatness of India, Mera Bharat Mahan, right? Okay, now participation in FMCT, Fissile Material Cut-off Treaty negotiations. We want to cut off the usage of the fissile material. We participate in these discussions. And India retain option of retaliating with nuclear weapons in the case of biological war or chemical war. Is it clear? For example, if any other country uses biological weapons on India or chemical weapons, examples, for example, okay, we can never know whether it is true or not. Okay, but one thing is, coronavirus is actually like blamed as it is a bio war by China. Is it clear? Okay, it is a conspiracy theory. We don't know and we can never know whether it is a fact or not. Is it clear? Even if the virus is released from a Chinese laboratory, how do we say whether they willingly released it or accidentally leaked? Is it clear? Okay, but such conspiracy theories are in circulation. Same way, like arsenic, such chemicals can be used in chemical weapons. If the other country uses chemical or biological weapons also, we can use nuclear weapons on them. So that is nuclear policy. Okay, next one, nuclear tests. Okay, we conducted 2 nuclear tests. Pokhran 1, which is called Smiling Buddha. Okay, which is tested on 18th May 1974. This is a fission bomb. Is it clear? Fission bomb. It means atom bomb. Pokhran 2, this is called Operation Shakti. Okay, it took place on 11th and 13th May 1998. This is a fission bomb and a fusion bomb. I mean hydrogen bomb plus atom bomb. Obviously, I told you to blast a hydrogen bomb, you need an atom bomb first. Is it clear? For activation energy, is it clear? Okay. Clear till now? Next. Okay. Now, International Atomic Energy Agency, and please remember the dates of our tests, 1974, 1998, is it clear? Remember the dates. Our first test date is 1974. There is some significance to it, okay, in the NSG, National, okay, and NPT, which I will discuss now, okay. So, remember this date, okay, right. International Atomic Energy Agency, what is this? This is an international body which will supervise different countries and keep checking if anybody is making nuclear weapons or doing any tests or enriching the uranium beyond the required concentrations. That is the work of IAEA. Is it clear? International Atomic Energy Agency. It is an international body which will supervise different countries if they are making nuclear weapons or enriching the uranium beyond certain point, something like that. Clear till now? Next. Okay. Next one, Nuclear Suppliers Group, NSG. Nuclear Suppliers Group, NSG. It is a body of total 48 countries. It's a body of total 48 countries. You might have studied about this also in international relations, but we will also briefly cover it. Okay. So, 48 countries. Okay. Brought in 1974 in response to our Smiling Buddha test. Is it clear? It is a multilateral export control regime. I mean, these 48 countries will keep exporting among themselves. Is it clear? They share technology, they export resources among only themselves. Right? Next. Okay. Now, admission of a new member is done through consensus. India has been attempting to formally join this particular nuclear suppliers group. Okay. What is the advantage if you join this group? You will get access to technology and resources. Is it nuclear technology, nuclear resources? Because these countries will keep exchanging the technology and resources among themselves. Is it clear? Okay. Right? So, India wants to join. But if a new member should join in this, we need consensus. Consensus is not majority. Every member should agree. And China is not agreeing for the joining of India. Is it clear? China is not agreeing for India to join the nuclear suppliers group. Clear till now? Okay. When China is not agreeing, majority of the other countries are in favor to India. They are okay for the India to join the group. So, now China should show a reason why it is not agreeing for the membership to India, right? What is the reason? That India is not a signatory of NPT, Non-Proliferation Treaty. Okay. India is not a signatory of NPT. Is it clear? Okay. Then carefully. If you do not sign NPT, then China will not accept our membership in the NSG. But if you sign the NPT, it means it is agreeing that we will never make the nuclear weapons. I hope it is clear. It is a discriminatory disarmament. I mean, China, you make weapons, but we don't make weapons. It is something like that. I hope it is clear. If you sign the NPT, It means it is agreeing that we will never make the nuclear weapons. If you don't sign the NPT, you cannot join the NSG. That is the twist. I hope it is clear. What are the benefits if you join the NSG? I already told you, you will get access to resources and technology. Okay, China is not agreeing, but still, we are not a member of NSG, but still. Okay, NSG granted a waiver to India in 2008. India is the only country which did not sign the NPT, but got this waiver at least. Is it clear? Okay, no nuclear weapons which is not a party of NPT but is still allowed to carry out nuclear commerce. Is it clear? India is the only country which is having nuclear weapons, did not sign NPT, but got a waiver for nuclear commerce. I mean, we can do trading with these countries. We got a waiver. For example, Pakistan is having nuclear weapons, did not sign the NPT, okay, but it did not get the waiver. So India is the only country which is having weapons, okay, did not sign NPT, but still we got the waiver. It means that is the confidence that those countries have on the have on India. Is it clear? Right? Next, look at this question. What are the consequences of a country becoming a member of NSG? It will have access to latest and most efficient nuclear technologies, correct. It will automatically become a member of NPT? No, it is opposite. Actually, if you sign NPT, then you will get membership in NSG. But if you become a member in NSG, you will not automatically become a signatory of NPT. So, opposite is correct. This is wrong. One only is the right answer. I hope it is clear. Okay. Then what is non-proliferation treaty? Okay. See, the treaty defines nuclear weapon states as those which conducted the test before 1967. NPT defines nuclear weapon state as a state which completed the test before 1967. Did we complete the test before 1967? Did we complete the test before 1967? No. So, according to NPT, is India a nuclear weapon state? According to NPT, please observe the sentence. According to NPT, is India a nuclear weapon state? No. According to NPT, only 5 states are nuclear weapon states. USA, P5 we call it as. USA, Russia, UK, China, France. Is it clear? According to NPT, only 5 states completed their nuclear test before 1967. So they are nuclear weapon states. India completed the test, India did the first test in 1974. So we are not a nuclear weapon state. And NPT delegitimizes proliferation of nuclear weapons fissile material by non-nuclear weapon states. It means see how discriminatory it is. According to NPT, these 5 countries can proliferate nuclear weapons and proliferate fissile material. But other countries should not proliferate the nuclear weapons or fissile material. So signing this means agreeing that we will not proliferate our nuclear weapons or fissile material. I hope it is clear for all of you. That's it. Okay. So with that we completed nuclear technology. Okay. Next let us proceed with information and communication technology. Okay. Or you want space or information, you can give me a choice if you want. Okay, I'll proceed with information and communication technology. This is related to computers, right? This is related to computers. Information and communication technology. Break at 8 p.m. 8 to 8:30 you will get the break. Okay. Information and communication technology. Okay. I also need a break, but we don't have time, so kindly understand. Okay, break as planned will be at 8 to 8:30 p.m. Information and communication technology. Okay. Tarak, we got a waiver for that reason only. Is it clear? Consequence of not signing NPT. We cannot join NSG. Prince Kumar, if you do not sign NPT, you cannot join NSG. If you cannot join NSG, then we don't have complete access, okay, to the nuclear technology or nuclear resource material. That is the problem. Okay? Clear till now? 8:30 is the planned breakup. It's okay, then we will do like that only. Okay? Now let us see the next topic, that is information and communication technology, right? In this, the first topic we are discussing is wireless technologies. Okay. In wireless technologies, okay, in wireless technologies, yes, Delhi's vibes, it is complete revision course only. Okay. Right. First one, short range devices. Short range devices. Okay. In the short range devices, they are radio devices which are low transmitting power, okay, in less distance. Is it clear? Like for example, one example here is CCTV. Let us suppose, okay, let us suppose there is one CCTV in my corridor outside my flat. Let us assume there is a CCTV. And if I can see the CCTV footage in my cell phone, is it clear? Then the communication between CCTV and my mobile, in which I can see my CCTV camera footage, that communication is a short range communication. That communication is a short range communication. Is it clear? Then what is the medium for this? The medium for this communication is, okay, radio waves. Radio waves are the medium for the communication. How the signals are traveling from my CCTV camera till my mobile, this 3-4 meters distance, how the signals are traveling through a medium called radio waves. Is it clear? Another example is wireless local area network. Okay, I mean, for example, there is an office. In that office, you have some 100 computers. These 100 computers are interconnected without wires. Okay, that is also short range communication, because same office, same building, 100 computers, they are connected. How are they connected? What is the medium? Medium wave for these computers communication is radio waves. The medium is radio waves. Is it clear? Next one, alarms and movement detectors, motion sensor. For example, I am in this room. In my corridor, if I keep a motion sensor, then what happens? If somebody walks in my corridor, then it will ring an alarm. Okay, and that will give an alarm in my mobile phone. I mean, that motion detector, movement detector is giving communication to my mobile phone, communicating over 2-3 meters of distance. That is called as short range communication. Is it clear? Okay. Again, medium is radio waves. Same way, Bluetooth, for example, we use Bluetooth AirPods headset or a Bluetooth pen like this. Okay. So, a Bluetooth like keypad to a computer. Okay. The communication is again short range communication. Okay. Your headset and your mobile phone, the medium for communication is radio waves. Medium for the communication is radio waves. So again example for short range communication. Next one, RFID. That is also short range communication. What is RFID? You know, FASTag, okay. In the FASTag, what happens is your vehicle will have tag. Tell me what will be there on your vehicle? Your vehicle will have tag, okay. And the toll plaza will have antenna. What will be there in the toll plaza? Antenna, right? The step 1, antenna will emit the radio waves. Is it clear? Antenna emits the radio waves. Is it clear? Step 2, okay. The radio waves will hit the tag on the vehicle. Is it clear? The radio waves will hit the tag on the vehicle. Step 3, okay. Tag will reflect the radio waves. Tag will reflect the radio waves. Is it clear? Okay. And then the tag will reflect the radio waves. Then step 4, okay. The antenna will send that signals to the reader. Is it clear? Then the reader will send the signals to the computer, okay. Computer will analyze the data in the tag and also will deduct what data will be there about how much distance the vehicle travelled. Is it clear? The data in the tag about the distance travelled also will deduct the amount from your FASTag account. This is how it will work. Is it clear? Okay. So I repeat, step 1, antenna will emit the radio waves. They will hit the tag on the vehicle. Then the tag will reflect the radio waves. It will be received again by the antenna. Then the antenna will send that signals coming from the tag into the reader. From the reader it will go to computer. Computer will do the processing. Which processing? It will analyze the data in the tag, that is how much distance it travelled and all those things. And then it will deduct the correct amount from your FASTag account. And then your vehicle will be permitted to cross the toll plaza. This is how the FASTag works. And that is also short range communication device. Is it clear? Okay. Okay. RFID is, okay, the actual radio frequency identification, whereas FASTag is one application of it. Okay, it means basically what is this? Basically this is RFID and FASTag is one application for it. Radio frequency identification. You are identifying a vehicle based on the radio wave frequencies. Is it clear? Radio frequency identification. One application is FASTag. What are the other applications? Asset tracking, personnel tracking. I mean, for example, let us suppose military personnel. You can keep RFID tags on them so that you can track them whether they are safe or not. I hope it is clear. Military personnel in the battlefield, okay, can be given the RFID tags. Is it clear? So that you can track them to know whether they are safe or not. Is it clear? Okay. Next, same way asset tracking, vehicles, ATM machine, cash carrying vehicles, vehicle which is carrying the cash for an ATM. We can use FASTags to track them. Okay. Controlling access, I mean employee or student ID card. Okay. Presently student ID card, you have to wear it in the neck and you have to show the ID card. Okay. But what if you have a tag? Is it clear? Automatically it will check whether you are the employee of that office or not, whether you are the student of that college or not, and you will be permitted. Is it clear? Indian Railways is using to track the bogies. Is it clear? Okay, wagons and coaches. Vadodara Municipal Corporation is using the RFID tags for the cows. You know, in India, cow is a very sacred animal and we want to protect the cows. So, we want to track the cows. Okay, as a part of the process of protecting them, we can attach RFID tags to those ears. Is it clear? RFID tags on their ears. These are various applications. Another example is Bluetooth. Bluetooth is also short range communication device. Then solve this question. Consider the following communication technologies. Which one is a short range device? All the above, all of them. CCTV, RFID, wireless LAN. Okay, all these are short range communication devices. Is it clear? Okay. That can be a signal issue. Okay. You are able to hear me? Next. Okay. After that, Wi-Fi. Wi-Fi is also, medium is radio waves. Medium is radio waves, but long distance. Long distance. Is it clear? For example, if you have Wi-Fi router in your home, okay, why do you keep a password for it? Because, okay, the other flats in your apartment also can use your Wi-Fi, isn't it? So, the Wi-Fi signals travel a little more distance. Is it clear? Not just like Bluetooth. For example, if I go to the next room, my Bluetooth will not work. If I go to the next flat also, okay, my Wi-Fi router will work. So, this is also wireless only. This is also radio waves as a medium, but not short range. I hope it is clear, but not the short range. Okay, uses radio waves to transmit the information at specific frequencies. Initially developed in 1990s, can offer high speed internet, range up to 100 meters, whereas short range is just only 1 or 2 meters, but its range is up to 100 meters. Speed, average speed of 100 Mbps, megabits per second, that is the average speed. Is it clear? Peak speed can be much more than that, but average speed is 100 Mbps. Clear for all of you? Next one. Okay, optical wireless communication. To explain this, I will have to show you the spectrum. Did I keep the image? No. So, let us see like this. Optical wireless communication, okay, and light fidelity, okay. Okay, optical wireless communication, okay, we will discuss that, Abhishek. Okay, first of all, in the electromagnetic spectrum, first you will have, okay, can you tell me the order? First you will have radio waves. Next comes, okay, yes, tell me, next comes microwaves. Okay. Then after that, after microwaves, next IR, next visible, next UV, next X-rays, next gamma rays, isn't it? Next gamma rays, okay, and every wave will have wavelength. Wavelength means the distance between two successive crests or troughs, like this, the distance between these two or those two. Okay, and we also have frequency. This is number of crests or troughs that pass a given point, in unit time. Okay, the number of crests and troughs that pass a given point in a unit time. Okay, like okay, for example, you are able to hear me, right? So right, so let us suppose this is that point. And this is that point. Okay, A, B. Which wave's frequency is more? This is high frequency. Okay, because distance between two crests is less. So more crests or troughs can pass through this particular point in a given time. Okay, more crests or troughs can pass through this point in a given time, that is frequency. And always wavelength and frequency are inversely proportional. Wavelength and frequency are inversely proportional. Okay, and one small announcement. In today's class, we cannot complete all the subjects. We still have space technology, we still have defense technology, and we also have nanotechnology. Okay, so unavoidably I am dividing this into 2 sessions. Okay, 2 sessions. So roughly I will take this class till around 10:30 or 11 p.m. I will stop this class. Let us assume 11 p.m. I will stop this class. Okay, and then I will take one more session. Is it clear? I will take one more session to complete the other 3 areas: space, defense, nano. Okay, so in the first session we have completed biotechnology, nuclear technology, and we will complete ICT. In the second session I will complete nano, okay, defense and space technology. Okay, right. So that is how I am trying to plan this in 2 sessions. So just remember this point. Okay, I will have to complete, I will try to complete ICT. Let us see when we can complete this. Okay, now, okay. So now coming back to this, what is optical wireless communication? Optical wireless communication. Okay, in optical wireless communication, tell me, in the normal Wi-Fi, which waves do we use? Everybody please. In the normal Wi-Fi we use radio waves, right? But if we use IR and visible, that is called optical wireless communication. Did you understand? Instead of radio waves, if we use light waves as medium for communication, if we use light waves as a medium for communication, that is called as optical wireless communication. Okay, and one more small announcement. Okay, I'll give you the break 8 to 8:30, small change in the program. Break 8 to 8:30 I'll give you, and then again 8:30 to 11 I will continue. Okay, let us try to close at 10:30. If it is not possible, the target is we have to complete ICT. Whenever we complete ICT, I will stop the class. It can be 10, 10:30, 11, maximum 11. Okay. Then after that, in one more session, the date will be announced in our Telegram channel. I will give you the date for the new session. Okay. So in the next session, I will complete nano, defense and space. I hope it is clear. The break at 8 to 8:30. Right? Okay. Completing is impossible because almost, okay, we are left with half. We will be completing only half. Okay. So completing it is almost impossible, or if I complete it will be meaningless. I mean, I will just have to read out some slides and all, which I don't want to do. Is it clear? If we do it, it should be meaningful and helpful to you. Right? Okay. Right. Yes, we will cover AI, ICT, AI. So what is the difference between Wi-Fi and like optical wireless communication? Tell me, in the Wi-Fi we use which waves as a medium? In Wi-Fi, which waves we use as a medium? First, radio waves. But optical, the word optical itself means what? Light waves. Light waves. As medium, then, okay, that is called optical wireless communication. Light waves means what all will come? Visible, IR, UV. Is it? Any light waves we use, it is called optical wireless communication. Can you tell me what is the advantage of optical wireless communication? Wi-Fi will be faster or OWC will be faster? Which one will be faster? Wi-Fi faster or OWC faster? OWC faster, because light waves are, okay, as we have seen, light waves are high frequency waves. Frequency in gamma rays are highest frequency. Highest frequency. Radio waves are lowest frequency. Lowest frequency means they are slow waves. So they carry the information slowly. So the internet will be slow. If we use IR, visible, UV, then the internet will be faster. I hope it is clear. That is the main advantage of this. Done. Next. What is LIFI? LIFI means light fidelity. LIFI means light fidelity. Wi-Fi means wireless fidelity. What is the difference between OWC and LIFI? OWC is, okay, IR, visible, UV, all the three. But LIFI is particularly only the visible light, okay, as medium. Is it clear? Only the visible light as medium. Clear? Okay. It can transmit data up to 100 Gbps, which is 100 times faster than Wi-Fi, because light waves, visible light is much high frequency when compared to radio waves, then micro, then IR, then visible, much high frequency. So 100 times faster than Wi-Fi and much higher when compared to Bluetooth. Okay. Very high speed when compared to Bluetooth, and even when compared to Wi-Fi it is 100 times faster. Then, what is used as a router here? An LED lamp is used as a router. LED lamp is used as a router. Clear? LED lamp is used as a router. Okay. Why? See, for example, okay, Wi-Fi router emits radio waves, but LiFi router, what is a LiFi router? It is nothing but an LED lamp. Is it clear? Okay, will emit the light waves. I hope you understood the similarity between the two. Okay, it will emit the light waves. Okay, But yes, can any LED lamp, let us suppose I have an LED lamp here, but is that a LiFi router? No, that is not a LiFi router. To work as a LiFi router, that LED lamp should be connected to a server. Look at this image. Okay, keep responding. What is this? This is LED lamp. Means we are using this one as LiFi router. Is it clear? This is LED lamp. That is LiFi router. Okay. But now it is connected to a server. What is this? It is a server from where you are getting the information, and it is the internet. This lamp, if this LED lamp is connected to internet and a server, that can work like a LiFi router. I hope it is clear. If that LED lamp is connected to internet and server, then it will work like a LiFi router. Is it clear? Okay. Next, from the internet and from the server, the streaming content is coming. For example, right now I am streaming this class. So this content is coming from server and internet. Is it clear? That will go to lamp driver. Use the word once. That will go to lamp driver. What is lamp driver? Okay, it will it will divert or channelize the signals to the LED lamp. I hope it is clear. Okay. It will channelize that particular signals towards the LED lamp. Look at here like this. It will divert, it will drive the signals, okay, to the LED lamp. Okay. I repeat, server and the internet. From there the streaming content is going. Step number 1, server and internet. From that the streaming content, whatever content you have to stream is coming. That will next go to second step, lamp driver. This is number 1, lamp driver. Lamp driver will receive the signals and channelize them towards the LED lamp. Then step number 3, LED lamp, okay, will send the light waves. See, light. Is it clear? Light is the medium. This light medium is carrying the signals, information, isn't it? The streaming content. Is it clear? Okay. From the LED lamp, the light waves are coming. Now the light waves are bringing the content to me, But, okay, my laptop should have a photo detector, otherwise it is useless. It means, okay, there is LED lamp. If it is not connected to server and internet, that is useless. That cannot work like a LiFi router. Okay, that is connected to server and internet. But I don't have a photo detector. My laptop do not have a light detector. It cannot detect the signals coming from the light waves. Then also it is useless. Is it clear? Then if my laptop is having a photo detector, from the photo detector it will go to amplifier. Amplifier will amplify the signal. Is it clear? Then the signal, okay, will go to my computer. My computer can receive the signal. My computer will have a photo detector and an amplifier. Photo detector will detect the signal. Amplifier will amplify the signal. And finally, I will get the signal. I will get the streaming content. The content which is streamed here, I will get it here in my laptop. Clear for all of you? Okay. So, step 1, server and internet will give the streaming content to the lamp driver. Step 2, lamp driver will take that content towards the lamp. Okay, step 3, lamp will emit the light. Okay, so that light will contain the signals. Light will contain the signals. Step 4, the photo detector in my laptop will detect that light signals. Step 5, amplifier will amplify the signals. And finally, I will get that signal. Whatever content you have streamed there, I received that content in my laptop or my system. Is it clear? This is how the LIFI works. Light fidelity works like this. I hope it is clear. Okay. Then what are the applications? Okay. Answer this question. If I switch on the lights here, close that room, will the light go to the next room? Can the light penetrate the walls and the doors? Can the light penetrate the walls and the doors? No. So, the main limitation is needs line of sight. I mean, I should be capable of seeing that LiFi router. I should be capable of seeing that LiFi. Next room, I cannot see this lamp, so I cannot get a signal from that. In the next room, I cannot see this lamp, so I cannot get signals from this. So, line of sight is compulsory. Therefore, limited range. It cannot go to the next room or next flat. My light, the light of this bulb is not going into the next flat. So, limited range. Installation cost is huge because my laptop will not have photo detectors. So, we have to again purchase new laptops, new devices. Is it clear? Just like some 1970s computers, they are not Wi-Fi compatible. Same way our present computers are not Wi-Fi compatible. Is it clear? Just like our 1970s or 1980s computers are not Wi-Fi compatible. They don't have radio wave detectors. Our present computers are not Wi-Fi compatible. They cannot get the signals in the form of light waves. Is it clear? So, therefore, high installation cost. These are the limitations. Is it clear? Wi-Fi can penetrate the obstructions, Antara. You only tell me. Right now, actually my Wi-Fi router is also in the next room, but I am getting the signals. Not only next room, you are keeping a password so that your neighbors will also use your Wi-Fi. Yes or no, Antara? Okay, means, okay, Wi-Fi signals can pass through the walls and doors, but light signals cannot pass. Radio waves can pass through the walls and doors. But, okay, light waves cannot pass through the doors and walls. I hope it is clear for everybody. Right. Then what are the applications? Big offices, aeroplanes. Big office, there are no doors and walls. Huge hall, then we can use Li-Fi. Street lights. You know, in 1960s movies, they used to say that I studied under the street light and I became a district collector. Is it clear? Now you can also say like that because the street light is a Li-Fi router. For example, if you don't have Wi-Fi, you can sit under the street light, but not for the light, for the internet. They sat under the street light for light. But we can sit under the street light for internet, free internet. Street lights can be used as Li-Fi routers. Is it clear? Is it clear? Next. Okay. Accident prevention. I mean, there are two cars which are going. Then the backside light of one car and the front headlights of the other car. If they both are Li-Fi routers, they will communicate with each other, and the first car will say to the last car, please stop, otherwise you will hit me, you will meet with an accident. Is it clear? In the winter season on Yamuna highway in Delhi, there will be several collisions. Is it clear? Because completely fog, you cannot see the vehicles. If two cars collide with each other, then all the next cars which are coming will collide with them. But if they have this Li-Fi capacity, then they will communicate with each other and then can avoid the collision. Is it clear? the back side light of the car, front side light of the car, they will communicate with each other. The car which stopped can ask the car which is coming from behind from a few meters distance to stop or slow down. Like that you can prevent the accidents. I hope it is clear. Okay, next, solve this question. With reference to LIFI recently in the news, which of the following statements are correct? It uses light as a medium for high speed data transmission, correct. It is a wireless technology which is several times faster than Wi-Fi, correct. Both 1 and 2. Did you notice that the questions are quite simple? If you roughly understand the concept also, you can solve most of the questions in science and technology. Okay. So, that is about the LIFI router. Now, let us take a small break. I will come back by 8:35 and the next class will be roughly till 11 p.m. Okay. Thank you so much. Take a break. So, you are able to hear me? Thank you so much, Fahim Raja. Thank you so much. Yes, I came 2 minutes early because I have to again set the mic and all those things. I'll start at 8:35 itself. Right. Hope I'm audible, visible. Okay, right. So, before the break, we completed about the LIFI, and I'll give you a small recap. First, we have radio waves. Next, we have microwaves. Next, we have IR. Next, visible. Next, UV. Next, X-rays. Next, gamma rays. These are the highest frequency. I mean fastest. These are the lowest frequency. It means slowest. So you will get slow internet. Slow internet. If you use IR, visible, UV, this is called optical wireless communication. Okay. And if we particularly use visible light, that is called visible light communication. Is it clear? VLC. Okay. Visible light communication, also called as light fidelity. Okay. It will give you faster internet. Okay. Light fidelity will give you faster internet, but if I want much more faster internet, can we use UV rays, X-rays, gamma rays? LiFi will give you high speed internet when compared to Wi-Fi, but if I want much more faster internet, can I use UV rays, X-rays, gamma rays? Yes, everybody? No, 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 because okay, UV, X-rays, gamma rays, they cause cancers. They are harmful. So, for faster internet, we cannot go beyond visible light waves. I hope it is clear. For faster internet, we cannot go beyond visible light. So, what is the disadvantage of the Wi-Fi? What is the disadvantage of the Wi-Fi? Minus point, slow internet. Plus point, okay, is it is not sensitive to obstacles. Because radio waves can pass through the walls, doors, etc. Okay, here quite opposite. VLC, light fidelity, advantage, plus point is fast internet. Minus point is it is sensitive to obstacles. Is it clear? You cannot use the internet in the next room or the next flat. Whereas Wi-Fi, if you have Wi-Fi router in any room of your flat or your apartment, you can use Wi-Fi in the other rooms also. Okay, in one office or one home, we will have one Wi-Fi router, but we can use it in all the rooms. In fact, the other flats, neighboring flats people also will get the signal. So we protect it with password. I hope you understood. So this is the comparison between Wi-Fi, LIFI. LIFI is 100 times faster than Wi-Fi and several times faster than Bluetooth. Okay, right. And the other applications are, we can convert street light into a free internet facility. Is it clear? You can just sit under the street light and study, but not for light, for the internet. Is it clear? That is the applications. Aeroplanes, big offices, to prevent accidents between two vehicles, so that the two vehicles can communicate with each other through their lights. Is it clear? And as I told you, like every LED lamp is not a LiFi router, it must be connected to the server and internet. And every laptop cannot use LiFi. Even if the LiFi technology comes, I cannot use it in my existing laptop. My laptop should have a photo detector to detect the light signals which are coming. I hope you got complete clarity on this. Is it clear? Okay. After that, we are moving on to the next topic, that is supercomputers. So what are the supercomputers? Okay. Supercomputers are just like your regular computer, but it is something like, in your CSAT you will do this work and time problems. If one person can do this task and construct a building in 10 days, 356 people can construct this building in how many days? So this kind of problems you will get. It is also same thing. It is based on, okay, a principle called as parallel computing. Is it clear? It is based on a principle called parallel computing. For example, your PC, your personal computer will have one processor, isn't it? How many processors you have in your personal computer? One processor. Okay, but a supercomputer will have lakhs of processors. Is that clear? Lakhs of processors, not just one processor, lakhs of processor. And all these processors will work together. If one processor can solve this problem in 1 second, 1 lakh processors can solve this problem in a microsecond or even less than that. So that is how the supercomputers are much faster. They don't work based on a different principle. It is just like your regular computer, but it is just like more computers working together. I hope the concept is clear. Is it clear? Okay. Next, we have two types of supercomputers, right? First one is called cluster computing, second one is called grid computing. Use both the words. We have two types of supercomputers, cluster computing and grid computing. First, let us see what is cluster computing. In cluster computing, nodes. What do you mean by nodes? Nodes means processors. One node is one processor. Is it clear? Nodes means processors. Nodes must be homogeneous. It means they should have the same type of hardware and same operating system. Okay, like Windows, Linux, like that. Is it clear? Okay. For example, if you are connecting 1 lakh processors, all the 1 lakh processors should have a similar hardware and a similar operating system. Tell me a few examples for operating systems you know. Few examples for operating systems you know. Okay, Windows, Mac OS, iOS. Is it clear? So here you can connect Windows plus iOS, you can connect. Possible. But that is not possible here. Okay, all the nodes must be using Windows only. All the nodes must be using iOS only. I hope it is clear. Okay, like that. Whereas grid computing, nodes have different operating systems and hardware. Machines can be homogeneous or heterogeneous. Is it clear? Okay, then one more thing. Here, okay, Grid computing means what? Okay, like even connect, they can connect the processors using different operating systems. So just tell me, in a grid computing, can we also connect the nodes which are using the same operating system? In the grid computing, can we also connect the nodes which are using the same operating system? Obviously. For example, okay, if you can make friendship with a different country person, why can't you make friendship with the same country person? I hope it is clear. You are so friendly that you can even make friend with another country person. So obviously you can be friend with your own country person. So they can even connect the nodes which are using different operating systems. So if they are using same operating systems also, it is okay. But whereas cluster computing, okay, they need same operating system. If they are using different operating systems, they cannot connect. Okay, so observe the point. They must be homogeneous. Whereas here they can be either homogeneous or heterogeneous. I hope it is clear. Yes, next one. In a cluster computing, all nodes are dedicated to perform the same task. Is it clear? In a cluster computer, if you have 1 lakh nodes, all the 1 lakh nodes must be dedicated to the same work. Whereas in a grid computer, is it clear? For example, in a grid computer, okay, let us suppose you have 1 lakh processors in one institute. There is one laboratory. Here you have a supercomputer. In this supercomputer you have 1 lakh nodes. But now the workload is less. Now the workload is less. You need only 50 nodes. Okay, I mean you need only 50,000 nodes. Is it clear? Then what happens? The other 50,000 nodes, can be used for other work. I hope it is clear. You have a laboratory. In that laboratory you have a supercomputer. It is having 1 lakh nodes. But at this moment the workload is very less. 50,000 nodes are enough for the time being. So other 50,000 nodes are now free. So they can use that their free capacity to do the other work. I mean when you have a free time, okay, you can do some other work. Then guess the name of it. Is it cluster or grid? Is it cluster or grid? Depending on the content in this, you tell me. In a lab, there is a supercomputer, 1 lakh processors are there, but now the work is less. 50 nodes are free. When they are free, they can do the other work. What kind of a system it is? Very good. This is grid. Is it clear? Computers in a grid contribute their unused processing resources to the grid computing network. But computers in a cluster must be dedicated to the same work and perform no other task. Even if these 50,000 nodes are free, they just have to stay free, but they cannot perform the other task. Such a computing system is called as cluster compute. Both are supercomputers, but that is called as cluster computer. Is it clear? Next one. In the cluster computer, computers must be located close together in the same room. Look at this image. Meet Mr. Pratyush. Pratyush is the name of a supercomputer. And look at here. What are they? What is this? This is like a CPU. It is like a processor. Means it is like a node. And you can see, okay, several nodes. in the same room. Okay, so Pratyush is a cluster or a grid? Everybody, Pratyush is a cluster or a grid? Here all the nodes are located in the same location. So is it a cluster or a grid? Very good, cluster. Computers are located close to each other. Computers may be located at a distance, but there is no rule that they should be located at a distance. They can be connected even when they are at a distance. So obviously they can be connected even if they are close to each other. Is it clear? Okay, so this is close or distant. This must be close. Is it clear? Okay, right. This is like, okay, different work. Okay, but this is only one work at a time. Okay, next, grid computing here. Okay, they can be same or different operating systems, but here same operating system. Is it clear? Same or different operating system. Done? Okay, so that is cluster and grid. Pratyush is a cluster. Okay, with theoretical peak performance of 1102 petaflops. Okay, what is a petaflop? To know what is a petaflop, first of all, what is a flop? Flop means, okay, floating point operations per second. Okay, flop means floating point operations per second. It means how many tasks it can do in 1 second. Is it clear? The number of operations. For example, if you give the supercomputer some problems, how many problems it can solve in 1 second? How many tasks it can complete in 1 second? That is called floating point operations per second. Is it clear? Okay, and when it comes to this, okay, observe this point. Okay, you will have like, just 1 second please. Yeah, okay. So 1 teraflop means what? 1 trillion. 1 petaflop means 1 quadrillion. Is it clear? You will have kilo, mega, giga, tera, peta. That is the order. Kilo, mega, giga, okay, tera, peta. Okay, tera means 1 trillion operations. Okay, 1 peta means 1 quadrillion operations. Is that clear? 1 teraflop means 1 trillion operations in 1 second. 1 petaflop means 1 quadrillion operations per second. But United States of America's frontier supercomputer, okay, can do 1102 operations, not 1102. 1102 petaflops, I mean 1102 quadrillion operations it can do in 1 second. Imagine how speed it is, how fast it is. I hope it is clear. Right? 1102 quadrillion operations in 1 second. With that capacity, this is the fastest supercomputer in the world. Clear? That is the fastest supercomputer in the world. Okay. In India, what is the fastest supercomputer? Aarambh is the fastest supercomputer in India as of June 2023. Is it clear? As of June 2023, Aarambh is the fastest supercomputer in India. Okay. Next. The supercomputer is a single system with more than 285,000 CPUs, 10,000 GPUs. What is a GPU? GPU is a graphics processing unit. GPU is a graphics processing unit. Is it clear? Okay. Graphics processing unit is also like processing unit but more faster. Like we use it for AR, VR, gaming applications, is it clear? VFX, graphics, for such purposes we use graphics processing unit. Okay, it is much more capable when compared to a regular processing unit. Okay, a supercomputer can have 2,85,000 CPUs and 10,000 GPUs in one particular unit. Is it clear? Okay, next. What are the applications of supercomputer? Okay. Right. Supercomputers are designed for calculation intensive tasks. Is it clear? So, I have given a list of applications of supercomputers, but to be short, what are the applications of supercomputers? Fast computing. Wherever you need faster computing, there we can use supercomputers. Is it clear? Wherever we need faster computing, we need supercomputers. Wherever you have to process enormous amount of data in a short span of time, then we have to use supercomputers. Is it clear? Or you want to analyze huge amount of data in the real time. Is it clear? As the data is accumulating, you want to analyze it. Okay. So for such purposes, we use supercomputers. Clear till now? We can use it in quantum mechanics. Okay, why quantum mechanics? What do you mean by a quantum particle? What do you mean by a quantum particle? Quantum particles means electrons, protons, neutrons. Is it clear? Okay. In a small matter, for example, in 1 gram of some matter, you will have trillions of quantum particles. Do you agree? If you just take 1 gram of any matter, it will have trillions of quantum particles and they interact in various combinations. Okay, if you have to study these many things, if you want to analyze these many quantum particles for quantum mechanics, you need supercomputers. Is it clear? Next, weather forecasting. How do we forecast weather? How do we predict rain? Okay, based on the previous data. In what kind of situations it rained earlier? How much was the cloud cover? How much was the percentage of humidity? Is it clear? What was the temperature? In which combination of conditions it rained earlier? The more data you have about when it rained earlier, the more easily and more accurately you can predict when next it will rain. Is it clear? So weather forecasting. Is it clear? Then climate research. For the same, more data you have to analyze for climate research. Is it clear? Molecular modeling. Okay, I mean you have to predict the structure of a protein. You want to predict the structure of a drug, or you want to design a new drug, or you want to design a new protein. In such places also supercomputers will help. Plus modeling the spread and prediction of pandemic peak. Like for example, okay, in the COVID-19 pandemic, we had to predict the peak. I mean, when do we reach the peak? Or during the peak, because in pandemics usually what happens, the cases gradually increase, they reach the maximum and they will start decreasing. So in which month we will get the peak, and at the peak how many cases will be there per day, when the decline will begin, and when the peak will actually end. All these things can be predicted using supercomputers. It means, okay, roughly predictions from the data, analysis of data, complicated calculations. Okay, when you want to study the interactions between a large number of particles, in all such cases we can use supercomputers. I hope supercomputers is clear for all of you. Okay, thank you so much, Pavan Kalyan. Now focusing on this, Pratyush and Mihir. Okay, by the way, who will develop this supercomputers? Center of Development for advanced computing. CDAC is the organization which will develop these supercomputers. Next, about Pratyush and Mihir. Okay, what is Pratyush capacity? 6.8 petaflops. Okay, but okay, Pratyush plus Mihir together is 6.8. Okay, only Pratyush is 4 petaflops, only Mihir is 2.8 petaflops. Okay, Pratyush is, see, Pratyush 4 petaflops, Mihir, that is another supercomputer, 2.8 petaflops. Pratyush is a cluster, Mihir is a cluster. But when Pratyush and Mihir are connected through grid, the together is 6.8 petaflops. Is it clear? Pratyush is a cluster computer, Mihir is a cluster computer. And Pratyush is located in Indian Institute of Tropical Meteorology, whereas Mihir is located in National Center for Medium Range Weather Forecast. So Pratyush is in Pune, Mihir is in Noida. So if Pratyush and Mihir are interconnected, should that be by cluster or grid? Okay. If Pratyush is located in Pune and Mihir is located in Noida and they both are interconnected, is it a cluster or a grid? That is a grid. Okay. Primarily be used for supporting research for improving the weather and climate models. Pratyush, Mihir both together are working on climate modeling and weather forecast. Is it clear? Pratyush and Mihir both work together for climate modeling and weather forecast. Okay. Then a recent news, that is in September 26, 2024, the Prime Minister launched 3 supercomputers, 3 Paramrudra supercomputers. Is it clear? 3 Paramrudra supercomputers. Is it clear? Possible Antara. Okay, possible Antara. Okay, that is the reason why I am giving you the dates. I have given frontier by November 2022. Okay, and Antara saying that November 2024. Okay, there is another one called as Captain that crossed the frontier, possible. Okay, see, the data is enormous. If there is any small update like that, you can add it to it. That is the reason why, to avoid confusion, because this data is quickly changing. Is it clear? To avoid confusion, I have given you the dates. Is it clear? By November 2022, this is fastest. Okay. So in the later date, if there is something, yes, definitely that must be the faster one. I hope it is clear. Okay. Yes, you can just cross check regarding that. Okay. Recently, okay, Prime Minister launched 3 Paramrudra supercomputers. The first one is located in Giant Meter Wave Radio Telescope in Pune. This is 1 petaflop. Second Paramrudra supercomputer is located in an institute in Delhi. Is it clear? Okay. That is 3 petaflops. That is 3 petaflops. Next, S.N. Bose National Center for Basic Science in Kolkata. That is the third Paramrudra supercomputer. Okay. And that is 838 teraflops. Okay. And one more thing. Because some of you are giving the information about this Captain, what I will do is, okay, US Captain, I will update in the notes. Before I post this notes to you in my Telegram channel, I will update it and then I will post it. Okay, right. Next. Now, I hope it is clear. 3 Paramrudra supercomputers in 3 different institutes all over the country. This is their capacity. This is their speed. Also, PM inaugurated Arka and Arunika. Use both the words once. Arka and Arunika. Okay, what are those two? Okay, Arka and Arunika, they both are high performance computing systems. High performance computing systems. Okay, what do you mean by high performance computing system? Usually this is a grid. Is it clear? One supercomputer word we use for cluster when many supercomputers are connected together. If a few supercomputers are connected together, just 1 second please. Okay, just 1 second. Okay, I'm sorry, you're able to hear me? Yes. Okay, you're able to hear me? Right. Usually we use the word one supercomputer for a cluster. That is, if all are located in one particular place like this Pratyush. But if a few supercomputers are connected together, if few supercomputers are connected together in a grid, then that is called as a high performance computing system, HPC, Arka, Arunika. That is the reason why if you see its capacity is 11.7 petaflops. Is it clear? Whereas only Pratyush only 4 petaflops. Okay, only 2.8. How 11.7 petaflops is possible? Here a few supercomputers are connected together in a grid. Such a system is called as high performance computing system. Is it clear? Okay, I repeat. If there is one supercomputer in one single location, that is usually we use the word supercomputer. But if a few supercomputers are connected together in a grid, that will be faster than a single supercomputer. Such systems are called as HPC, high performance computing systems. Okay, example Arka, Arunika. Okay, so Prime Minister launched 3 Paramrudra supercomputers and 2 high performance computing systems. I hope it is clear for all of you. Right? Okay. Yes, current affairs are also included in this. Next, the fastest supercomputers in India, Aarambh, 13.7 petaflops. But if you observe, Aarambh 13.7 petaflops. Okay, and then Paramsiddhi 5.27 petaflops. Pratyush 4 petaflops. Mihir 2.81 petaflops. I hope it is clear. Any doubts in this? Okay, very good. Next we are discussing about National Supercomputing Mission. Let's just have a small recap. Okay, supercomputers are a group of processors working together. Okay, and then we have seen about Pratyush, Mihir, okay, and Aarambh, okay, Paramsiddhi, like that. And also we discussed about, okay, some recently launched supercomputers called 3 Paramrudra supercomputers and a high performance computing system. Is it clear? Okay, and then applications of supercomputers. Next we are discussing the National Supercomputing Mission. Okay. Yes, National Supercomputing Mission. This mission was launched in 2015, duration of 7 years with a cost of 4,500 crores. The target of the mission is to increase India's supercomputing capability by 45 petaflops together. Okay, what is this? All supercomputers together capacity must be 45 petaflops. Also, I think I will update this once. I think this number may be, okay, increasing, right? Next, this mission is implemented by Department of Science and Technology, okay, and the Ministry of Department of Information Technology. Is it clear? Which departments are implementing this mission? National Supercomputing Mission is implemented by Department of Science and Technology and, okay, yes, right, very good. Then the leading organizations are CDAC and IISc, okay, Indian Institute of Science Bangalore and CDAC. These are the two leading organizations who are implementing this particular scheme, mission. Is it clear? Scheme is implemented by two leading organizations, Center for Development of Advanced Computing CDAC and Indian Institute of Science Bangalore. 34 supercomputers have been installed across the nation with an aggregated computing capacity of 35 petaflops. This is as of March 2025. Is it clear? As of March 2025, in India, we have total 34 supercomputers with a cumulative capacity of 35 petaflops. Clear till now? Right. So, with that, we completed the supercomputers. The next topic we are discussing is artificial intelligence. The next topic is artificial intelligence. Okay. Let us see about artificial intelligence. Clear? Okay, let us continue related to artificial intelligence. The mic is troubling a bit because it's been so long. The charge is getting completed and we are getting some other technical issues. So sorry for the brief okay, disturbance in between. Yes, we can continue now. Okay. So now we are discussing about artificial intelligence. Okay, right. So what is artificial intelligence? The ability of the machines to perform the cognitive tasks. I mean, if a machine can think like a human being or a machine can solve a problem like a human being, then that is called as artificial intelligence, right? Ability of the machines to perform cognitive tasks like thinking, perceiving, understanding, learning, problem solving, decision making, etc. Okay, that is called as artificial intelligence. The total artificial intelligence is based on neural networks. Let us try to understand this neural network. First, I am explaining the natural neural network. Okay, use the word natural neural network. Natural neural network. Clear? What is a natural neural network? How the human brain works. Because here in the artificial intelligence, the machines should imitate the human brain. Machines should imitate the human brain. Is it clear? So, first let us try to understand how the natural human brain works. In the brain, we have, okay, the nerve cells, right? Okay. What are these called as? Dendrites. Dendrites. Okay. This is called the cell body, cell body, and this is the nucleus, this one is the nucleus, okay. And then one dendrite will be little longer, and that is called as axon, that is called as axon. Then axon also will have dendrites at its end, okay. Then what happens, the dendrites of the next neuron, is it clear? The dendrites of the next neuron, right. And this junction between the two is called as synapse. Synapse, that is the connection between two neurons. Is it clear? The connection between the two neurons, okay. This is called natural neural network. Now let me explain the artificial neural network. In the black color whatever I am writing is artificial neural network. Is it clear? Artificial neural network. In the red color what I have written is natural neural network, okay. So here each cell body is compared to a node. Is it clear? In our brain, a neuron, a nerve cell, okay, is comparable to one node in a artificial neural network. Is it clear? Neuron cell compared to nodes, okay. For example, here 2 lakh nodes connected, okay, 2 lakh nodes connected. Here, okay, billions of neurons connected. Is it clear? Billions of neurons connected. Got the clarity? Right? So neuron cell is compared to node, okay. Dendrites are compared to input devices. For example, what are the different input devices we have? The pen which I am using here, this is an input device. I am giving some input to this particular system, okay. This mic which I am using is an input device. So like that, dendrites are compared to inputs of the supercomputer. Axon is compared to, dendrites are compared to inputs, axon compared to output. Is it clear? And synapse compared to, okay, like synapse compared to, okay, like trinetra, that is a, okay, connection between the nodes. Trinetra is not a supercomputer, that is a connection between the nodes. Let me just, okay, I will just have to, I think, recheck this name once. But I think Trinetra mostly is a connection between the nodes that can be compared to the synapse. Interconnections between the nodes is called as synapse. Done? Internode communication, okay. Internode communication. So like this, in the black color I have written artificial neural network, in the red color I have written natural neural network, okay. The artificial neural network will imitate the human brain, it will imitate the human brain, okay. So like that, the machines will be, okay, intelligent like the human brain. Like human brain, they can also solve a problem, take a decision, think about something, all these cognitive tasks they can perform. I hope it is clear, okay. Artificial intelligence solve problems that are even impossible or difficult by the human beings. In the artificial neural network, nodes contain the data and link to the other nodes like neurons in the human brain. Is it clear? In the artificial neural network, nodes contain data. Each node will have data, okay, and all the nodes are interconnected just like the neurons are interconnected in the human brain, okay. We have seen the comparison between the two. Then in the artificial intelligence, we will have 3 levels, 3 layers. First layer is called as, the very first layer is called as deep learning. The first layer is called as, okay, artificial intelligence. What is this? Development of smart systems, smart machines, okay, that can carry out the tasks that typically require human intelligence. Is it clear? If the machine can do a task which actually require human intelligence, then we call it as artificial intelligence. Is it clear? If machine can do any task which originally require the human intelligence, means a machine is mimicking, imitating the natural intelligence, then we call it as artificial intelligence. I hope it is clear. Next, what is machine learning, the second layer or the second level? Here, okay, this creates algorithms that can learn from the data. You tell me, artificial intelligence is based on what? Artificial intelligence is based on memory, based on data. For example, okay, your Myntra app will suggest, okay, what dress you want to purchase next. Your Amazon app will suggest you what more item you want to purchase next. Or it will give you advice. For example, if I purchase one object, the app will suggest me that, okay, the customers who purchased A also purchased B. So there is a chance that you may also wish to purchase B. Like that it will suggest. On what basis it is suggesting? Based on the previous data. For example, your Netflix app, okay, if you, or your YouTube, I mean, if you keep on watching one type of videos, you will keep on getting suggestions for the same type of videos. I mean, the machine is learning based on the memory, based on previous experiences, based on the data. The more data you give to the computer, the more intelligent it will become. I hope it is clear. Okay, then, right? Same, whereas the natural intelligence, human intelligence is based on emotions plus of course data and memory. Okay, of course it will also take decisions based on the memory. For example, I touched the fire once, my hand got burnt. Next time obviously I will not touch the fire. I have the data stored in my brain, in my memory. Of course that is there, but it will also, what is lacking in the artificial intelligence and what we have in our intelligence is emotional intelligence. In your ethics paper you also study about emotional intelligence, EQ, just like we have IQ, we also have EQ, emotional coefficient, okay, that is another parameter. So what is the primary difference between artificial intelligence and natural human intelligence? Artificial intelligence is purely based on memory. It will not take decisions based on the emotions. Is it clear? Okay. Whereas natural intelligence is of course based on the data and memory, plus it will also take decisions based on the emotional intelligence. Is it clear? Okay. So, right. Machine learning is learning from the data and make decisions, okay, based on the patterns observed. Patterns observed means the Netflix app observed a pattern in which your videos watching. So it will suggest similar videos. YouTube learnt a pattern from your previous views. What kind of content you are enjoying. It will show you the same suggestions. It will observe a pattern. It can also solve crime cases based on the patterns. We have serial killers. You know, modus operandi. A criminal will usually have a mode of operation, we call it as modus operandi. Like for example, some people will have a habit of smoking after the crime, or eating something after the crime, like that. Based on the pattern they will follow in doing the crimes, it can even solve the crime cases. There are many cases solved using artificial intelligence in the recent days. It can also solve the cases like cyber crimes. Basically, it will observe a pattern. Okay, and based on that pattern, it will take decisions. Like it will suggest you a video. But did it happen several times that you got suggestion for a video that you do not like? It happens several times. It means the machine learning is not 100% accurate. Is it clear? Sometimes it needs human intervention when its decisions goes wrong. In the machine learning, the machine's decisions can go wrong. For example, you are chatting with a chat bot. Okay. Then what happens? You will type a question. It will give you an answer. For some preset questions, it can give you the answer. But if you give a more complicated question to it, then it cannot answer. Then a human being will come into the picture. Is it clear? That is called machine learning. It can identify a pattern and it can take a decision. But the decision can go wrong. And when the decision goes wrong, okay, then what you have to do? You need human intervention. This is not completely free of the human intervention. Third level is deep learning. It uses artificial neural network to reach accurate conclusions without human interventions. For example, if a chatbot is using deep learning, then you don't need a human intervention. Even if you give a complicated question, it can intelligently think and answer. So these are the three levels. Okay, so first level. Okay, first level, artificial intelligence. If a machine can do a task which require human intelligence, artificial intelligence. And if a machine can observe a pattern and take a decision, okay, but it can sometimes go wrong. In that situations, you need human intervention, machine learning. Okay, and if a machine can observe a pattern and take a decision and it will not go wrong, you do not need the human intervention, then that is called deep learning. These are the three different levels in the artificial intelligence. I hope it is clear. Okay, right. How they observe the pattern. For example, one person is killing only females, or one person is killing only babies. Like that, the artificial intelligence will observe a pattern in the crime. Based on the pattern, they will identify the criminal, like that. Okay, right. So with that, artificial intelligence is completed. Next concept is called deep fakes. Okay, what are the deep fakes? Deep fakes are the fake digital content. I mean photos or videos. Is it clear? Fake digital content like images, videos, audios, like that. A heroine's video, like say for example in the context of this India-Pak war, okay, there are many deep fake videos as if, okay, Trump is giving some statement about the war, something like that. So all these are called deep fake content. Is it clear? Right. Okay. So sometimes it will be confusing. It looks so realistic as if really Trump is giving that particular statement. Okay, or really some Amitabh Bachchan is giving a statement, or really this is a video of so and so heroine. Okay, it will be like that. They are called as deep fakes. Deep fakes are made using, okay, a technique called generative adversarial networks. Generative adversarial networks. It uses two competing neural networks. Two artificial neural networks will be used. Is it clear? Okay. One is called generator. What is its role? It will generate the video. Second artificial neural network is called discriminator. Is it clear? So, generator will generate a fake audio and video of Trump. Is it clear? Okay. Discriminator will try to distinguish it between with the original video of the same person. Is it clear? Okay. For example, generator will generate my photograph. Discriminator will compare it with my original photograph. Okay. Generator will generate my video and discriminator will distinguish, compare it with my original video. Is it clear? If the discriminator is successful in distinguishing between the two, then the generator fail or success, fail or pass, tell me. If the discriminator can distinguish between my original video and my deep fake video, in that case the generator is successful or failed, failed. So it's a feedback. Then the generator will edit this video, okay, to more resemble my original video. Then again it will go back to the discriminator. Discriminator will again try to compare it with my original video, and again it became successful in distinguishing between. Discriminator could say that this is original video of Bindu and that is deep fake video of Bindu. Then again the generator fail. So generator to discriminator, discriminator to generator, again generator to discriminator, again discriminator to generator. So this cycle continues so long that the discriminator finally failed to distinguish between my original video and my deep fake video. Okay, so it says that the deep fake is original. This is the fake video of Bindu. This is the original video of the Bindu. Okay, then what happened? The generator pass. This is how the deep fake videos are, okay, created. Is it clear? Okay. The generator learns from the feedback given by the discriminator and improves its output until it can fool the discriminator. If it can fool the discriminator, then it means it became successful. Next one. What is data synthesis? Data synthesis means creating the deep fake. Data synthesis means creating the deep fake. Is it clear? Synthesizing the data, creating the data. Okay. Then AI deep synthesis is also called as deep fake. It is a technology that can generate or manipulate image, audio, video content that appears realistic. It is not realistic. It is not authentic. But it appears as if it is realistic, it appears as if it is authentic. I hope it is clear. That is data synthesis, deep synthesis. Data synthesis means just creating a video. Deep synthesis is creating a video as if it looks realistic. Data synthesis is creating a video or an image. But deep synthesis is creating a video or an image which actually looks like the original video or the image. Is it clear? It looks like the original one. Next. Now let us see some AI models. One very famous one is ChatGPT. GPT means generative, okay, pre-trained transformer. That is the full form of GPT. Is it clear? It is an artificial intelligence chatbot which is created by US based body called as OpenAI. It is a chatbot. It is called generative pre-trained transformer, okay, and it is made by OpenAI, okay. Another example is Bard, now which is called as Gemini, This is also generative AI model. It is built by Google, okay. Similar to ChatGPT but built by Google. Is it clear? Gemini multi-model. What do you mean by Gemini multi-model? This is ability of Gemini AI model to understand, process and generate different types of data. For example, if I ask it a question in the form of, okay, text, image, video, audio together, still it can answer. Is it clear? I am giving a question to Gemini multi-model. The question contains one image, one video, some audio, some text. Okay, it can understand all of them and can respond to me by giving the answer. And the answer also contains some part of the video, some audio, some images and some text. Then it is called as a multi-model AI chatbot. Is it clear? Right? Okay. Next. This one is developed by IIT Bombay. It is a multi-model AI tool. It is a series of large language models that can respond in several Indian languages like Hindi, Tamil, Marathi, etc. Is it clear? Similar to ChatGPT, but can respond in multiple languages, multiple Indian languages. Is it clear? Can we continue? Next one. Okay. The next one is, okay, Sangam Digital Twin Initiative. Interesting. Listen this carefully. Sangam Digital Twin Initiative. Look at this video. I hope you have seen the video. Okay. What is this? This is a digital twin to an original road, okay, and bridge. I hope you understand. There is a city. In the city, okay, you have original road, infrastructure, road, bridge and all. This is also road bridge. Is it clear? But it is not the real one. It is the digital twin of the original road. I mean, we create an identical digital structure similar to a road and a bridge. I hope it is clear. Is the digital twin concept clear? Look at here. This is also digital twin. What is she doing? She is planning a city development. Is it clear? In this screen, she is planning a city development. But where is she planning the development? On the digital twin of that city. I mean, okay, on the earth, I will really have that city. But on my screen, I have a digital model of that city, which is like a replica of that city. Is it clear? Okay, that is called a digital twin. Is it clear? Got the clarity? Right. Okay. A digital twin is a digital representation of a physical asset. Physical asset, road. What is digital twin? The digital representation of the road. Car. Physical asset is a car. Its digital representation is called digital twin. Is it clear? Okay. Such as a car, bridge, building, etc. Clear for all of you? And for example, if you observe how they are using the digital model, initially they kept the bridge at a lower level, but it is obstructing the traffic. Then they increased the height of the bridge. Did you notice? So they are using this digital twin for, okay, designing a city development, planning a city's development. I hope it is clear. They are using it to plan a city's development. Digital twins learn, update and communicate with their physical counterparts, okay, throughout the asset's life cycle. I mean, for example, so long this road and bridge stay, as long as this road and bridge stay there, so long its digital model also will be there. So for example, okay, if the road is becoming old, needs maintenance, you need maintenance in the digital model also. Is it clear? So these digital models will help us not only to design a city, but also to maintain the city, to predict when the repairs will come, what we have to do, what kind of changes we have to do, how to upgrade this, all these things we can do in this digital model. Is it clear? Okay, right. In India, the state of Andhra Pradesh, new capital city Amaravati Smart City is being created using the digital twin. Okay, however, they wanted Amaravati city, the capital city of Andhra Pradesh, they created a digital model of that. They will do the designing, editing, just like I told you here. The bridge was at a low height, but it is obstructing the traffic. So they increased the height. What should be the height of a bridge? What should be the width of a road? How long this bridge can be? Okay, all these things they can do the designing in the digital model, implement it in the real infrastructure. That is called as Sangam Digital Twin Initiative. Okay, first time it is being used in Indian city of Amaravati Smart City in Andhra Pradesh state. Clear for all of you? Okay. Then next, AI, artificial intelligence in India. In June 2018, the Indian government defined national policy on artificial intelligence. Okay, national strategy for artificial intelligence, that is AI for all. This paper identified 5 focus areas. Okay, is it clear? I repeat, in June 2018, Indian government identified the national policy on AI, defined the national policy on AI. That is called National Strategy for Artificial Intelligence, AI for all. This paper identified 5 areas where artificial intelligence can be used. What are those 5 areas? Healthcare. How can we use artificial intelligence in the healthcare? We can predict a heart attack. Is it clear? I mean, if your heart is working like this today, after 1 year, what can be the condition of your heart? If your kidney is damaged this much now, after 10 years, what will be the condition of your kidney? Okay. If you are taking alcohol at this rate now, in how many more years your liver may be damaged? All these predictions it can do. Also, it can gather the data, like for example, we have genome sequencing, if you remember. Genome sequencing. For example, Genome Asia 100K, 1 lakh genomes, 1 lakh genomes means it is large quantity of data. Is it clear? More data. How to process this data? We use artificial intelligence to process and analyze this data. Is it clear? We use artificial intelligence to process and analyze this data. Clear for all of you? Okay. Next, agriculture. In agriculture, we can do predictions like this is the soil, what kind of crop will be better. Or you can predict how much yield can come. Or you can predict when you have to give the next fertilizer, when you have to spray the next pesticide. Okay, like that you can do all predictions. Is it clear? Okay, in the education we use artificial intelligence, obviously. For example, sometimes when you want a model answer for some exam, what you will do? You will ask a question to ChatGPT. Are you using artificial intelligence in your education? Yes. Smart cities development. I just told you Sangam Digital Twin Initiative. You can develop smart cities using artificial intelligence. Is it clear? Okay. What is a smart city? Let us see what is a smart city. A smart city will have a smart electricity meter. Okay, a smart, okay, water meters. Okay, or a smart dustbin. And I am not joking, we really have it. a smart dustbin that will send you a WhatsApp message that I am full, come and clean me. Is it clear? It can sense that it is full. It will send you a WhatsApp message asking you to empty the bin. Is it clear? In the municipality we use it. Is it clear? Smart traffic control. Traffic control. Okay. If you are using all these things, not only that, we also use Internet of Things. Internet of Things. Okay, we call it as IoT. What is Internet of Things? Okay, your smart TV where you can watch YouTube, where you can watch any video from the internet. Okay, or if my AC is connected to the internet, okay, I can switch on the AC half an hour before I reach the home so that the room will become cool by the time I reach my home. Okay, this is called Internet of Things. If the things like your washing machine, your air conditioner, your rice cooker or your television, okay, if they are connected to internet, that is called Internet of Things. Okay, in a city where you have all these things, that is called as a smart city. Is it clear? Okay, smart transportation like Tesla car, driverless cars. Is it clear? Okay, where the car will take a self decision whether to take right or take left or stop, okay, or you have to slow down or speed up, the car will take the decision. Okay, smart transport. In all these areas we can use artificial intelligence. Is it clear? Next. On March 7th, 2024, Union Cabinet approved the India AI mission with an outlay of 10,372 crore for the next 5 years. Is it clear? Right? Next. What are the challenges in artificial intelligence? Lack of broad-based expertise. We don't have much expertise in the artificial intelligence. Okay. Absence of enabling data ecosystems. Where to store this much amount of data? Okay, so we don't have efficient storage facilities. We don't have a data ecosystem. Okay. And high resource cost and low awareness about artificial intelligence. Privacy and security. Okay, you are saving this enormous amount of data, but how can you keep this data secure? Privacy and security. Absence of collaborative approach in the applications of AI. It means all departments should work together for artificial intelligence, which is not happening. So these are some challenges in artificial intelligence. Is it clear? I hope the artificial intelligence topic is clear for all of you. Okay. Right. Now, the next topic we are discussing is, okay, robotics. This is about the robots. This is about the robots. What is a robot? Robot is an electromechanical device. It's a mechanical device plus it is also electronic. Electronic mechanical device. Electromechanical device. It can accomplish a variety of tasks. Okay. Then there are 3 types of robots. The first type of robots are called as fixed robots. Fixed robots means they are basically industrial robots. Okay, what they will do? Repetitive task. For example, there is a drug manufacturing company. Okay, in that, okay, it is manufacturing some tonics, syrups. Then they will use a robot. What the robot will do? Same task. Okay, close the lid, close the lid, close the lid, close the lid. Is it clear? Repetitively, it will close the lid of all the bottles. It cannot do the other work. It can do only one thing. Close the lid, close the lid. 1000 bottles, it will repeatedly close the lids. Such robots are called as fixed robots. Is it clear? They are industrial robots. They will do, okay, specific repetitive tasks, same task repeatedly, okay. Second type of robots are mobile robots. These robots can move, okay. They can move in terrestrial, they can move on the land, they can move on the water, they can also move in the air. Is it clear? They can also move in the space. Is it clear? With legs and wheels. Can you give me one example for mobile robots? They can walk with legs or wheels on the space. Yes, exactly. Rover. Rover used in Chandrayaan 3. The rover used in Chandrayaan 3 is an example for mobile robot. Some drones also can be called as mobile robots. Is it clear? Unmanned aerial vehicles, drones can also be called as, okay, mobile robots. Okay. What about Vyomamitra? Okay. Tanu and Param asking what is about Vyomamitra. That is not mobile, that is third type. That is humanoid robots. They are built to mimic the human motion and interaction. They look like a human being. They can talk to you like a human being. They can interact with you like a human being. Such robots are called as humanoid robots. Is it clear? Such robots are called humanoid robots. Okay. Now let us see different applications of robots. Is it clear? Various applications of robots. First, healthcare. In the diagnosis. For example, COVID-19 pandemic. Very risky. Okay. But some frontline workers were collecting the samples in the diagnostic centers. Is it clear? It is risky for them. What if we keep a robot there? Robot cannot catch COVID-19. It can safely collect the sample from the human being. Diagnosis. Is it clear? Okay. Treatment. For example, you have to give some medicines to COVID patient. If you send a nurse, okay, it will be risky for her. What if you send that particular medicine or a procedure using the robot? Or we also have robotic surgery. Is it clear? We also have robotic surgery. Is it clear? We have something called cyber knife. Okay, it can do robotic radiotherapy. Is it clear? It can do robotic radiotherapy for cancer. Is it clear? Like that, robotic radiotherapy, robotic surgeries, surgery assistance by robots. For example, sometimes there will be a surgery. The junior doctors or the nurses will stand behind them to give some like scissors or some equipment to the senior doctor. But you don't, that will contaminate the patient, isn't it? By chance that particular assistant is having some common cold or some infection, okay, then the patient may also catch it. What if you keep robots in the surgery assistance? Caregiving, give water to a patient, medicines to a patient. Is it clear? Change the bedding of a patient. All such things the robots can do. Is it clear? Okay. Restaurants, cooking robots, serving robots, bartending robots. They will make the cocktails, they will mix the drinks like that. Hotel, at the reservation counter like that. Public infrastructure, we can use robots in surveillance, cleaning, etc. Retail, in the point of sale, I mean like a shopkeeper, the robot will sit. Is it clear? Shopping assistance, a robot will give you assistance, will give you advice about what you may wish to purchase. Or homes, okay, cleaning, okay, like one of you is rightly pointing out pocha lagane wala robot, is it clear? Robotic vacuum cleaner, robotic mopping device, is it clear? It will automatically, okay, clean. You don't have to clean the vacuum cleaner. It will be like a disc shape, is it clear? It will automatically clean. It will first, okay, like clean the room, and then it will also mop the room. Maintenance, cooking robots, space research, is it clear? In all these places we can use the robots. I hope it is clear for all of you. Okay, right. Next, Sophia. Sophia is developed by Hanson Robotics through artificial intelligence. It is the first humanoid robot to get full citizenship of a country, Saudi Arabia. Sophia became a citizen of a country. First robot to get citizenship. Is it clear? Humanoid robot. Done. Next one. This is Sophia. First humanoid robot to get full citizenship in a country. Okay. Next. Vyomamitra. As you are asking, look at here. This is Vyomamitra. We call Vyomamitra as a half humanoid robot. Vyomamitra as a half humanoid robot. Why it is a half humanoid robot? Because we have only torso, I mean no legs. Why Vyomamitra is sitting here? To hide the fact that it has no legs. Is it clear? Okay. Then why are we using it? It will fly into the space in Gaganyaan mission. What is Gaganyaan mission? In Gaganyaan, we are planning to send our astronauts into the space. Is it clear? In Gaganyaan, we are planning to send astronauts into the space. But directly if you send astronauts into the space, it will be a risky experiment. So the plan is we will send the Vyomamitra first. Okay. In the unmanned mission, we will first send Vyomamitra. If that becomes successful, later we send the manned mission. We actually send the astronauts. I hope it is clear. Clear for all of you? Then, Vyomamitra can respond to the environment. It can generate warnings. It can operate switches in the Gaganyaan spacecraft. Okay, switch on, switch off. It can monitor the crew module. What is crew module? The room-like place in the Gaganyaan spacecraft where the astronauts will spend their time. Okay, Vyomamitra will keep monitoring this crew module, whether everything is perfect or is there any defect. Okay, then receive the voice command. It means from the ground station, if you give a voice command, Vyomamitra can listen to it and respond. Respond via speech. Vyomamitra can talk to you bilingual English and Hindi. Is it clear? Right? Okay, that is Vyomamitra, half humanoid robot. Next, there is another robot. I hope it is clear for all of you. Right, there is another robot called Feder. It is a humanoid robot sent into the space by Russia in 2019. Is it clear? Russia spent this robot in 2019. That is also humanoid robot. Another example, Robonaut 2. Robonaut 2. Developed by NASA. This is also humanoid robot. So, Sophia, Vyomamitra of India, Vyomamitra India, okay, and then Sophia, okay, developed by Hanson Robotics, and then you have this Feder, which is a Russian robot, Robonaut 2, which is made by NASA, okay, it was sent into International Space Station in 2011. Is it clear? Okay, Robonaut 2 was sent into International Space Station by NASA in the year 2011. It can take over simple, repetitive and especially dangerous tasks. We use robots to perform certain tasks which are proven risky and very tedious, okay, for the human beings. Is it clear? We use robots to perform the tasks which are dangerous and very tiring, okay, for the human beings. Clear? Next, one more robot, Bandicoot. Okay, this is not a humanoid robot. Okay, it is a spider shaped robot. It will be like this. It is used to clean the, okay, sewers and manholes, because you know that manual scavenging is prohibited presently. We should not employ human beings for manual scavenging. Is it clear? To clean the manholes, sewers and all that. So after that is banned, okay, we invented this robot to clean the sewers and manholes. Is it clear? Okay. Yes, definitely we need robots for border security also. Okay. But they can be hacked, that must be kept in the mind. Next, during COVID-19, what robots were used? Maitri was a robot to give food, water and medicines. Maitri was a robot used to give food, water and medicines in the hospitals. Drones are also robots, unmanned aerial vehicles. They are used to decontaminate the places. For example, a drone will spray the sanitizers, okay, disinfectant liquids on the roads, disinfectant liquids on a hospital. Is it clear? Okay, or it can rush the samples to the laboratories. For example, you collected the samples, okay, for diagnostic test in some remote place like a forest, village, like that. Is it clear? Then you have to quickly send that samples to a city diagnostic center. We can use drones. Like this, different robots are used, okay, in different tasks. So that is all about the robots. Is it clear? First we understood what is a robot. It is an electromechanical device. Then we have seen types of robots, fixed robots, mobile robots, humanoid robots. Then we have seen various applications. Then examples, Sophia, Vyomamitra, Feder, Robonaut 2, Bandicoot that will clean the sewers and manholes, and Maitri which gave medicines, food, water to the patients during the COVID-19. Okay, and drones are used to carry the samples, okay, from the remote places to the cities, to spray disinfectant solutions on the roads or hospitals, okay, when there is like when there are more infections in that area. Like that we use robots for. Even rover we discussed, mobile robot example, rover used in Chandrayaan 3, okay. All these are the good examples for robots. Is it clear? Okay. Then next we are discussing about blockchain technology. Is it clear? Blockchain technology. Okay. Right. What is a blockchain technology? Okay, it is, complete focus please. It is a digital, basically it is a ledger. Is it clear? What do you mean by a ledger? Ledger is like a record of transactions, isn't it? You can have accounts ledger, or for example an apartment entry and exit ledger. I hope it is clear. An apartment entry, exit ledger, or an account ledger, right? Like an account book. But this is not on the paper. This is digital. First point about it is digital. Second point about it is distributed. It is distributed. Means what? For example, okay, if there are 100 users in a blockchain, if there are 100 users in a blockchain, okay, all 100 users will have a copy of this. Is it clear? Therefore, it is called a distributed ledger. I mean, yes. Third point is it is decentralized. Decentralized. Okay. No single centralized authority. All users, okay, all users will control it together. No central authority. Is it clear? Okay. So complete my sentences. Okay. Basically, what is a blockchain? Blockchain, it is a ledger. What are its features? It is a digital ledger and it is distributed. It is distributed or decentralized. I mean, if 100 users are using this blockchain, all the 100 people will have a copy of this ledger. Is it clear? Okay. So, it is maintained on multiple computer systems controlled by different entities. Is it clear? Look at this. Controlled by different entities, decentralized. All users will control it together. Okay, observe what I have written here. All users will control it together. Is it clear? Okay, multiple computer systems controlled by different entities. Is it clear? Identical copies exist. All 100 users will have copies of it. I hope this is clear for all of you. Okay, and it is digital ledger, that is blockchain. Okay, now Satoshi Nakamoto, a pseudonymous person. It can be a person or a group of people or a company. We don't know who is that. No identity. Okay. The name used is Satoshi Nakamoto. We don't know who is it. Created a digital cryptocurrency called as Bitcoin. Okay. Created a digital cryptocurrency called as Bitcoin. Is it clear? Okay. So, now let us see how this blockchain technology works. To this decentralized database, new information is added in the form of a block. Okay. Now, look at here. I will explain how the blockchain technology works. Right. So, for example, this is, okay, a block. Basically, what do you mean by a block? Block is like a file. Is it clear? For example, in my computer, I have different files, a biotechnology file, a space technology file. I have different files. So, in this particular topic, we call the file, okay, as a block. Is that clear? Basically, what is a block? Block is like a file. Is it clear? What do you have in a file? For example, I have some biotechnology file. What do you have in that? I have some data in that. Same way, in the block also we have data. I hope it is clear. Okay, keep responding. Right. So, this is the first block. It will have 3 components in it. First one, okay, it's data. Right. Second one, it's hash. Hash is something like a password. Is it clear? Hash is like a password. Right. Okay. Third one, okay, actually first one only these two, but look at here. Now the second block. The second block will have 3 components. Okay, it's data, the data in the second block, plus it's hash, plus the hash of previous block. Is it clear? I mean, it will also have the password of the first block. Is that clear? The hash of the previous block. Okay. Then, okay, like the next one will have its own data, its own hash plus hash of, okay, the second block. Is it clear? The hash of the second block. Okay, like that, if you have, let us suppose, then the last one will have its data, then its hash plus the hash of 999th block. If this is 1000th block, I hope you understood. If this is the 1000th block, it will have its own data, its own hash plus the hash of the 999th block also. Is it clear for all of you? Right. Then, okay, the data in the block depends on the blockchain is used for which purpose. For example, okay, if you are using this for bitcoins, for cryptocurrency, if you are using it for cryptocurrency, okay, then the data in this will be something like, okay, you will have the data like this. For example, A gave 10 bitcoins. A gave 10 bitcoins to B. Okay, it will have data something like this. Clear till now? A gave 10 bitcoins to B. That is the data. Clear? Okay. Now, let us suppose, for example, I have only 10 bitcoins. I have only 10 bitcoins. Okay. Then, I have already given it to B. I already transferred it to B. Right. It means now my bitcoins are completed. I have zero balance. I have zero balance. But what if I try to change this data as B as C now? It means I am trying to use 10 coins as 20 coins. Is it clear? I am using the same amount, same 10 bitcoins I transferred to A, okay, but I am trying to manipulate, just change the B as C so that I can send the same 10 bitcoins to C. I hope it is clear. Okay, can we do that? It is not possible. Why it is not possible? Because if I try to, complete focus here, okay, if I try to change anything about the first block, listen carefully, if I try to change anything to the first block, then the first block's hash changes. Is it clear? Each block contains some data, a unique ID called hash plus the hash of the previous block. If you change something in one block, it will change its hash. If its hash changes, it will make all the next blocks in the chain invalid, because actually, okay, now the second block, listen this carefully, the second block will have the hash of the first block, but now the first block's hash changed. First block's hash changed means now, okay, the second block is having hash of the previous block, right, but it is having the old hash. Now the hash changed, first block's hash changed, so the first block disconnected, the second block is not connected to the first block. Because how the second block is connected to the first block? Through this hash only. Okay, just observe this. It will have the hash of the previous block. But now, when I tried to manipulate the content in the first block, the hash of the first block changed. Okay, this hash, okay, the hash of the first block changed. Is it clear? Then the first block will lose its connection with the second block. Is it clear? So total blockchain will get wasted. I hope it is clear. And okay, then some people got the same doubt like you, that is, what if you change the hashes of all the next blocks? Okay, like for example, I tried to manipulate the first block. First block's hash changed. Okay, so first block disconnected to second block. Okay, no problem. Go and change the hash in the second block also. Is it clear? Okay, here change the hash of the previous block. Keep the new hash here. You can do that. But if you do that, then the second block's hash will change. So the second block will lose the connection with the third one. Okay, then go to the third one and change the hash there also. Okay, then the third block's hash will change. So it will lose connection with the fourth one. So like that, it goes on. But what if you change hashes in all the blocks? You can do that. But for that reason only, what they do is, we need something called proof of work. Proof of work. It means, whenever I make any change to the content in a block, then I have to, this is like the CAPTCHA code. I have to prove that I am the human being. Like that, here I have to prove that I only did it. For that, I have to solve a mathematics puzzle. Is it clear? The chance of changing the hashes in all the next blocks is minimized by a mechanism called proof of work, where a mathematical puzzle is to be solved for adding each new block. Whenever you want to make any change, after making the change, you have to solve a mathematics puzzle. Then you may ask me, ma'am, I am very poor in mathematics. I don't know maths. So can't I use blockchain technology? Yes. It is not testing your mathematical skill. You take the help of a friend, you take the help of a computer, you take the help of a calculator, okay, or you can bring another friend and make her do the problem. The point is only to slow down the process. It is not to test your mathematical skills. You don't need mathematical skills. The purpose of this proof of work, why you have to solve a puzzle after doing every change in these blocks, is it clear? That is only to slow down the process. I mean, it is not possible to change the hashes in all the blocks. So in essence, okay, what is blockchain technology? In blockchain technology, everybody will get access to your content, but nobody can change your content. That is a summary of blockchain technology. Is it clear? Right? Whatever content you have in the blockchain, if there are 100 people, 100 users in the blockchain, all 100 users can get access to your content, but nobody can change your content. In such places, you can use blockchain technology. For example, everybody will know how many bitcoins you have, but they cannot change the number of bitcoins in your block. Is it clear? Everybody can know how many bitcoins you have, but nobody can change the number of bitcoins you have. They cannot manipulate the content. Okay, so that is a summary of blockchain technology. Right. What are the applications of blockchain technology? First one, cryptocurrency, Bitcoin, Ethereum, etc. Is it clear? It allows cryptocurrency to operate without the need of central authority. I mean, no bank. Okay, what is the problem if there is a bank? There are many cases where the bank is closed and the people got cheated, isn't it? For example, you make some savings in a bank and they close that private bank tomorrow. It means all your savings are gone. If there is a centralized organization, that centralized organization can cheat the users, misuse the users. Is it clear? Okay. So, that is the reason why we do not want a centralized organization. We don't want a bank to control our transactions. We will control our own transactions. Okay, so that is the purpose of cryptocurrency. It will reduce the risk and eliminate the processing transaction fees. We don't have to pay transaction fees to the bank. Okay, this is related to cryptocurrency. Okay, can a single person destroy the blockchain in this way? Theoretically, yes, but it will take a long time for doing it. Is it clear? Meanwhile, people can realize that something fishy is going on. Okay, I hope it is clear, Gaurav. Okay, right. So, cryptocurrency. Example, Bitcoins, Ethereum. They both are Bitcoins only, cryptocurrency only, but different brand name, that's all. Next, can we use Bitcoins for voting purpose? Yes, because for example, if I voted for one person, nobody can rig my vote. Is it clear? Nobody can rig my vote, or nobody can change. I voted for A, but nobody can change it as B. Nobody can manipulate. Very good. But what is the problem? Everybody knows to whom you have voted. Is it clear? So, our political leaders can kind of blackmail you that you have to vote for that person only, or may come to know that you did not vote that person. Okay, such things are possible. That is the reason why we are still depending on EVMs, but instead of EVMs, we can use blockchain technology for voting purpose also. I hope it is clear. We can use blockchain technology for voting purpose also. Next, healthcare. For example, I have so many diagnostic test reports. Okay, I can store them in the blockchain. Okay, secure transfer of patient database, prescriptions, medicine supply, etc. Diagnostic test reports I can store in the blockchain. Land registrations. For example, I purchased a flat or I purchased a land. Okay, then we do the registration. Okay, and we can also do land registrations through blockchain technology. Is it clear? But remember, everybody will come to know which land I purchased, where did I purchase, how much land I purchased. Is it clear? But nobody can manipulate. Okay, for example, nobody can change the name on the registration document from my name to their name. Is it clear? You cannot change the name, you cannot manipulate, but you will know. Other people will get to know that I have purchased this much amount of the land in this place. So, now finally, okay, the conclusion is like, okay, we can use bitcoins we can use blockchain technology in all the situations where privacy is not needed, okay, but security is needed. For example, if you want to write a love letter to your girlfriend secretly, don't keep it in the blockchain. Is it clear? I mean to say, when your requirement is not privacy, secrecy, but your requirement is security, in all these places you can use blockchain technology. Clear for all of you? Next, non-fungible tokens. Non-fungible tokens means what? Okay, the word fungible means exchangeable with equivalency. Okay, exchangeable with equivalency. Okay, what are the examples for it? Dollar. Okay, is my dollar and your dollar have the same value? My dollar is equal to your dollar. My rupee is equal to your rupee. Is it clear? Okay, so that I can exchange my 10 rupees with your 10 rupees. They are called as fungible tokens. Example, currency like dollars, rupees, etc. Bitcoin, Ethereum. What are they? They are cryptocurrency. Cryptocurrency. Is it clear? Okay. My Bitcoin is equal to your Bitcoin. My Ethereum value is equal to your Ethereum value. So they are fungible tokens. I can exchange my Ethereum with your Ethereum, my 10 rupees with your 10 rupees, like that. But what is non-fungible tokens? For example, can I exchange my home, okay, with some Ambani's home? Not possible. Not possible with equivalency, because my home, their home are not having equal value. I hope it is clear. So they cannot be exchanged. For example, okay, my painting, can I exchange it with some Picasso's painting? No, that will be more costly, more valuable. My painting will not be valuable. Who will purchase my painting? Is it clear? So, okay, house, property, art piece, they are called non-fungible tokens. I cannot exchange. For example, I cannot say, okay, you give me your artwork, you give me your painting, I will give you my painting. Or you give me your house, I will give you my house. It is not possible. Such things are called non-fungible tokens. So basically, what are the non-fungible tokens? They are cryptographic assets on the blockchain. NFTs can represent real world items. They are the digital version of art pieces, real estate, or okay, any other music album. Is it clear? Your flat, your land, your art piece, your music album. Is it clear? They are NFTs, but not the real ones. Digital version of all these things are called as NFTs. Clear? The digital version of my flat is an NFT. The digital version of my art piece is an NFT. Is it clear? By the way, and this is also on the blockchain technology only, please. Blockchain technology. Is it clear? For example, I kept my artwork, I kept my painting in the blockchain. Will everybody see that? Yes, they will see. I don't have any problem. I don't want privacy here. I don't want secrecy. Rather, I want publicity here. I want everybody to see that, appreciate it, so that I want them to purchase. For example, why did I keep the digital version of my flat in the blockchain? So that everybody will see this digital version. They may appreciate it. They may wish to purchase it. Marketing, is it clear? That is the reason why we keep these non-fungible tokens on the blockchain technology. Is it clear? This is one more application of blockchain technology. Is it clear? Yes, Gaurav. Okay, blockchain technology accommodates fungible tokens like Ethereum and Bitcoin, non-fungible tokens like real estate, artworks, music albums, digital versions. Why? Because I'm keeping them on sale. But if I want secrecy, privacy, it is not blockchain technology. Search some other method. So, in one word, what are the applications of blockchain technology? Where your target is not secrecy, privacy, but your target is security. Nobody should steal it, nobody should manipulate it, but everybody can see that. Land registration, health records, voting, okay, cryptocurrencies, okay, that is fungible tokens, non-fungible tokens, votings, health records, land registrations, okay, all these things you can do on the blockchain technology. Okay, then solve these questions, all of you. Okay, with reference to the non-fungible tokens, consider the following statements. Non-fungible tokens, they enable the digital representation of physical assets. Yes, it is a digital version of my flat, it is a digital version of my painting, correct. They are unique cryptographic tokens that exist on the blockchain. Do NFTs exist on the blockchain? Yes, I am keeping the digital versions of these buildings, okay, on the blockchain. Is it clear? Why am I keeping it on the blockchain? For sale purpose. Is it clear? I am keeping them for sale purpose. They can be traded or exchanged at equivalency. No, they cannot be. My artwork is not of same value of a Picasso's artwork. I hope it is clear. Okay, which of the above statements are correct? 1 and 2 are correct. Next one. With reference to blockchain technology, consider the following. Blockchain technology is a public ledger. Will the public have access to it? Yes. Where everybody can inspect. Okay. Nobody can control. This is what I said. If you want secrecy, privacy, don't use it. But if you want safety, security, that nobody is controlling it, use it. Correct. Okay. Everybody will get access to it. Everybody can inspect to whom you are voting. Everybody can inspect how many bitcoins you have. Everybody can inspect your art piece, your music album, your digital version of the buildings and all, but nobody can manipulate them. Is it clear? The structure and design of the blockchain is such that all the data in it is about cryptocurrency, correct or wrong? Wrong, because we use it for NFTs, we use it for health records, we use it for voting purpose, we use it for land registrations. Use it anywhere you want security but not secrecy. So wrong. Next, applications that depend on basic features of blockchain can be developed without anybody's permission. Yes, Satoshi Nakamoto took whose permission to make the bitcoins? We don't even know the identity of Satoshi Nakamoto. Is it clear? We don't even know whether it is a man or a woman or a group of people or a company or what it is. So without your identity, without anybody's permission, you can create a basic feature on the blockchain. Is it clear? Applications that depending on blockchain technology can be created without anybody's permission. Okay, so 1 and 3 are the right answers. Is it clear? 1 and 3 are the right answers. Okay, then next topic. Next topic we are discussing is about, okay, the evolution of internet. Okay, but this is not 5G, 6G, 4G. Is it clear? This is Web 1 to Web 5. Is it clear? Web 1 to Web 5. Is it clear? This is not 3G, 4G, 5G, 6G. This is Web 1 to Web 5. Okay. Let us see and try to understand what it is. What is Web 1? It is read only. Okay. We used this, okay, In my childhood, like some 1980s and all, we used this. Okay, read only. It means people are passive users. People are passive users, right? Okay, I mean, for example, I can read the content. I cannot upload the content. I can watch a video, but I cannot upload my video. I can listen to a song in the internet, but I cannot sing and upload. I am a passive user. Okay, that is called Web 1, no interaction. So static, and it is onymous. Onymous. Okay, onymous means having identity. Having identity. User will have identity. Is it clear? Okay, here user has identity. Next, okay, Web 2. Web 2 is social media. Is it clear? This is current, present. Right now the internet, you are watching this video on the YouTube. Okay, so okay, when you are watching this video on the YouTube, you are not a passive user. You are chatting with me in the chat box. Is it clear? You are chatting with me in the chat box. You are interacting with the content. Is it clear? You are asking me a doubt. I am responding by giving you an answer. Okay, so that is called, it is read and write. It means I can read a document, I can upload a document. I can watch a video, I can also upload this video in the YouTube. Is it clear? Okay, so I am not a passive user. I can also do like, share, subscribe, forward, all these things we can do. I can upload my videos. Okay, and it is centralized. Centralized means what? Okay, centralized means, example, Facebook, Twitter, okay, YouTube, Instagram, all these are Web 2. Is it clear? Right. Why this is called centralized? For example, okay, if you are using the Facebook, then what happens? The Facebook organization, the Meta organization is controlling your data. Is it clear? Okay, the company owns your data. You are giving your data to the Facebook organization. They can sell your data for advertisement purpose. Is it clear? They can misuse your data. A centralized organization like Facebook, Instagram, YouTube, Twitter are controlling your data. That is the present internet. Is it clear? Then this is also onymous. It means the user has identity, isn't it? For example, in your Facebook profile, you have your identity. You will keep your name, okay, your display picture, your profile picture, about information, that is your address or your marital status. So you have an identity when you are using the Facebook. Is it clear? When you are using Facebook, or for example, if you make a comment on this video, you are onymous. I mean, I can see your like your mail ID or something from which you are giving this comment. I hope it is clear. So you have an identity in Twitter, Facebook, Instagram, YouTube. The user will have an identity. Is it clear? Okay. Name and other details, even profile picture also, like that. So which internet we are using currently? Currently we are using the, okay, Web 2. Okay, Web 2 we are using currently. Then what is Web 3? Web 3 is read, write, okay. Read, write like Web 2 plus own. Okay, I can read the data. I mean, I can watch a video. I can write the data. For example, I can upload the video. Not only that, I will own my video, I will own my data. There is no centralized organization like Facebook, Instagram, Twitter, YouTube. All users together will own this data. Is it clear? Okay. It means Web3 is like a social media on blockchain platform. Is that clear for all of you? Yes, it is blockchain based. Just like social media, just like present YouTube, Facebook, Twitter, Instagram. But such centralized organizations will not be there, I mean to say. Okay, all the users will control it together. So this is just like social media, but the only thing is, in the Web 2, we only read and write, but here we read, write also. Okay, we own our own data, decentralized. But here, the main problem is, negative point here is anonymous. User has no identity. Is it a good point or a bad point? Is it a good point or bad point? Bad. Because why do you post a video or a music album? Why do you post something in the social media? For fame, for publicity. Is it clear? For identity, isn't it? Okay. So when you don't have identity, it is meaningless. Okay. Then coming on to the last one, that is Web 5. Is it clear? The last one, that is Web 5. Can we proceed? Web 5 is Web 2 plus Web 3. What we take from Web 2? Okay. From Web 2, the feature we have taken is, it is onymous. Means like Web 2, in Web 5, you have identity. Is it clear? From Web 3, the feature you have taken is, okay, decentralized. I mean own the data. Is it clear? I mean taken the plus points of both Web 2 and Web 3. Is it clear? If you club the, what is the best point in Web 3? Decentralized. Read, write and own. What is the best point in Web 2? Onymous. Is it clear? Okay, means you have an identity. Anonymity, anonymous means what? No name, no identity. Okay. So, the best feature in Web 3 is we can own the data, decentralized. The best feature in Web 2 is onymous. Club both the best features in one, that is Web 5. Then you may ask me, where is Web 4 in between? Is it clear? Web 4 is also like Web 5 only, but Web 5 is a little bit more advanced when compared to Web 4. Okay, there are no unique features in the Web 4 which is different from the Web 5. So, separately it is not mentioned here. Web 4 is also like Web 5, but little less advanced. Is it clear? I hope you got the difference between Web 1 to Web 5. Then solve this question. With reference to Web 3, consider the following statements. Web 3 technology enables people to control their own data, correct. In the Web 3 world, there can be blockchain based social network, yes, correct. This is like a Facebook on blockchain. This is like a Twitter on blockchain, like that. Just like Facebook, Twitter, Instagram, but that organization will not be there. Your profiles will be there in a blockchain, that's it. Web 3 operates by users collectively rather than an operation, rather than a corporation, okay. Battery down, full down, okay. Right, D is the right answer. Is it clear? D is the right answer. Done? Next. Okay. Now moving on to the next topic, that is virtual reality. Virtual reality. Okay. What is a virtual reality? What is a virtual reality? Thank you so much for your concern. Okay. And also thank you so much that you are awake, okay, and active and quite actively participating, answering all the questions throughout the session. Thank you so much. Now look at here. What is virtual reality? Okay. What is virtual reality? Look at here. This is a brain. But is it the real brain? No. It is a three-dimensional virtual reality. It is a three-dimensional virtual reality. Is it clear? Okay. Right. No problem. Let us just continue for a few more minutes. I will stop somewhere around 11 o'clock. Okay. Continue this. That is virtual reality. Look at here. Okay. We will just complete AR, VR, metaverse and all those things. Okay. Right. So, virtual reality refers to computer generated simulation. Simulation means what? Imitation. It just looks like a real brain but not a real brain. Okay. Which a person can interact within an artificial three-dimensional, it is artificial, three-dimensional environment using electronic devices such as special goggles. For example, you can see that they are wearing some goggles. They can see this virtual brain only if they wear those goggles. Is it clear? And they are also wearing some haptic gloves. You know haptics, for example, touch screen, mobile touch screen. What is this? This is haptics technology, isn't it? Haptics means touch, right? Okay. Haptic gloves. You know something? They can even feel the brain if they wear those gloves. Not only they see the 3D structure of the brain, they can, how soft the brain is it clear? Like that they can actually feel the brain. I hope it is clear. Okay. So, these made, and if it is a heart, they can feel the heartbeat. Is it clear? Okay. Like that. And they can wear the like some headset. If they wear the headset, they can listen the heartbeat. Is it clear? Okay. Like that, you can interact with a total virtual world. Okay. Haptic gloves fitted with sensors. In this simulated artificial environment, the user can have a realistic feeling and experience. You will feel as if you are touching the brain. You will feel as if you feel the heartbeat. Is it clear? Okay. You will even feel the warmth of the heart. The heart temperature also will be little high because it is pumping the blood. So, the sensors also will increase the temperature and show. Is it clear? Like that you can actually feel. I will give you another example. You can go to a virtual tour to solar system. Okay. You can feel the earth. You can go touch the moon and come. Okay. You can go sit on the Mars and come. But don't get shocked, not the real Mars. Virtual image of the Mars. Okay, you can get the real experience of the solar system. That is virtual reality. Is it clear? Okay. Now, what are the applications of it? Virtual reality has several applications like education. For example, usually medicos, when they learn the anatomy, they have one major problem. They need a dead body for dissection. Is it clear? There is a scarcity for the dead bodies. So they will keep fighting that this hand is mine, this leg is mine, this heart is mine, like that they will keep fighting for the dead bodies. Now with the virtual reality, no need. They can learn on the virtual brain. Also, when you do the dissection, you cannot experience a working heart, a working brain. But in the virtual reality, you can see the working brain, working heart. Is it clear? Okay. So, like that, okay, for education purpose, schools, especially children with special needs. For example, I want to explain the solar system to an autistic child. Difficult to explain. But if I can take him to a virtual tour to the solar system, he can better understand it with experience. Is it clear? Okay. Children with special needs, medicos. Okay. For example, a dentist will try to remove a virtual tooth first. Later will try to remove the real tooth from a real patient. Is it clear? Okay. Medical, consequence-free learning. For example, first I will do surgery on a virtual heart. Then I will go to the direct surgery on the patient's heart. Is it clear? Sports. For example, I want to practice batting. Is it clear? Okay. But who will keep on putting a ball onto my bat? Nobody. I don't have the other partner to practice. So, I can play with a virtual bat, virtual ball, sports practice, military practice. For example, recently in this India-Pak consequences, okay, the government called for youth, okay, to work in the forces, and very nice that several young people actively participated. They really went and they are ready to participate in the army. Is it clear? But now they don't have training. Okay. Or say for example, soldiers only. You are appointed as a soldier. Can I directly send you to the battlefield? Not possible. First, you will practice in the virtual battlefield. Okay. That is consequence-free learning. For all these things we can do. Now, next one is augmented reality. Not virtual reality, augmented reality. Okay. What is augmented reality? Look at this video. In this video, this man is real. This landscape is real. Is it clear? Okay. These mountains are real. But just this dinosaur is virtual. It means something is added. Augmented means what? Augment. Augmented means added. Something is added to the real world. What is the difference between virtual world and virtual reality and augmented reality? Okay. In virtual reality, the real world is completely shut down. In augmented reality, real world will be there, but something will be added to the real world. That is called augmented reality. This is one example. Another example is, yes, Lenskart app is a good example. Your face is real, but the specs you try on your face are virtual reality. Reality is not shut down, rather something is added to the reality. That is called augmented reality. Okay, Dulux company, okay, is providing an application through which you can apply a virtual paint to your wall and see if that color, okay, is actually looking nice on your wall or not. Because sometimes when we see the catalog, is it clear? Okay, we see one color, but when you actually apply it on the wall in a large area, it turns out to be a different color and quite disappointing. We all might have faced it sometimes. So you can try it. That is augmented reality. Is it clear? Okay. 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 1 second. My Okay. So that is also called as augmented reality. Okay. Tourism also. Let us suppose you went to a museum. Usually when we go to such monuments and all, what we do? We appoint a guide. But there are some problems. You don't know the language in which the guide is talking to you. You don't know the language in which the guide is talking to you. And you have to pay huge amount to him. Plus, you don't know whether the guide is giving you the real information about the monument or he is telling you his own fantasy stories. How to trust that person? Is it clear? So in such situations, what we can do is, for example, I want to know about this monument. The monument is real, but the information about this monument is superimposed on that. I will see that painting. On the painting, I will superimpose the digital version of who painted this, when painted this, all those details. Same way, heads up display. Look at here. This is the real display. Real view of a pilot. Is it clear? Real view of a pilot. And on that, you can see that these directions are superimposed. For example, when you are driving a car, when you are driving a car, if you want to check the direction, you will check the direction, in Google Maps, beside you in your mobile. But for example, you are driving a fighter jet, I will just check my mobile whether I have to go right or left. I will just check the mobile to know whether I have to take right or left. But it is a fighter jet. It's not a normal car on the road. Is it clear? So it can lead to a huge problem. So what happens? The directions will be directly superimposed on my view itself. So I don't have to turn my head to see where I have to go in the Google Maps. Is it clear? Something like this. This is all called as augmented reality. And now lastly, the last topic in today's class, that is metaverse. The last topic in today's class, metaverse. What is metaverse? It is, I'll tell you a small story. I will explain metaverse in a small story. You know this? Look at here. This is H&M store. This is H&M store in metaverse. Is it clear? Where what happens here is, I hope you all are following. Where here, have you ever played these games like PUBG and all? In those games, you will create your virtual avatar, isn't it? I mean, you can keep moving your virtual avatar will shoot, your virtual avatar will dance, Depending on your commands, your virtual avatar will keep moving. Is it clear? So, you will create your virtual avatar. Step 1. Your virtual, listen this story very carefully please. You will create your virtual avatar. Your virtual avatar will go to virtual H&M store. The one you are seeing in this picture. Then your virtual avatar will try virtual shirt and you liked it. Is it clear? You will purchase it using cryptocurrency, and you will come back. That is metaverse. Is it clear? It means in this you are using AR, VR. What is AR? Your virtual avatar is AR and this is AR. Is it clear? This H&M store is AR. Your virtual avatar is AR. What is VR? You are trying it onto that. That is AR also. NFT. What is NFT? That H&M shirt is NFT. Can I exchange H&M shirt with Zara shirt? No, both have different value. So they are not exchangeable with equivalency. NFT, all accessible in the digital space. It is a digital space. Do we have this store in reality? No. It is a digital space. Sharing the same digital economy. It means you will purchase using bitcoins. All these together is called metaverse. Is it clear? All these together is called AR, VR, NFT, okay? FTs, that is fungible tokens, bitcoins, okay? Digital space, all these together. Example, JP Morgan opened first bank in the metaverse. H&M opened a store in the metaverse. And there is another topic called metanomics. Metanomics means metaverse economics. Economics in metaverse. You know, we have meta crimes also. In the last class I discussed about it. You know, recently rape happened in the metaverse. Cyber crime. Can you expect a rape as a cyber crime? Yes, it happened. I mean, the virtual avatar of a girl raped by the virtual avatar of a boy. Of course, no consequences like sexually transmitted diseases or pregnancies, but it will also create a psychological trauma to that girl. So, we need special laws to deal with such crimes. Is it clear? This is all about the metaverse. I hope it is clear. For example, COVID-19 lockdown, the people were getting bored. All the kids can send their virtual avatars to a virtual park where all the virtual avatars of the kids will play together in the park and come back. Then you may ask me, ma'am, in what way this is different from Myntra app? In Myntra app also you can try it. If you don't like it, you can return it. Is it clear? Okay, then why this one? See, we don't always go for shopping to purchase the things. We also go for window shopping. We like that experience of shopping. So in this we can get the experience of shopping. Is it clear? Then the good thing is you can get the realistic experience of shopping. Realistic experience of why the games like PUBG are so addictive. Because it will give you a realistic experience. Why Ludo is not that much addictive? Is it clear? Like that. So that is a plus point. But minus point is new type of crimes are possible in the metaverse. Is it clear? Okay. Next. Right. There is one previous question on metaverse in the year 2024 which we discussed in the PYQ marathon. So I did not keep it here. The next topic is quantum computers, but okay, I think now even I am not like in a condition to continue this. Though I am in a condition to continue this, my gadgets are not permitting. Okay, so the charge is completed in many of my gadgets like mic and the mobile in which I am checking your chat and all those things. So I will have to stop this session here today. Okay, let us try to have one more session to continue the revision. Thank you so much, and okay, you can pour all your affection in the form of the comments that can recharge my batteries. Thank you so much. Okay, take care. Bye bye. This class ends here. After the class, you can all pour your affection in the form of comments in the comment section, right? Then, thank you so much. Yes.